had to look at the breed prediction probabilities. What's happening here was, like you can see in Kankraj, the range is quite high. Even if it belongs to 10% of Kankraj, it was being assigned to Kankraj. So we thought, like, what should be the cutoff among the set of breeds that we have predicted, and how to predict whether a breed actually belongs to that breed or not. So what we have done is, like, looking at this, we looked at the interquartile range, and first looked at the interquartile ranges of those breeds wherein they are true to breed, like Ongol, Siri, Ambalachari. You can look, they are close to around 90%. So we fixed a cutoff around 90% and only selected animals with those breed prediction probabilities of greater than 90%. So this is what we have, this is what I was talking about. And then we looked at the admixture of those animals that were finally selected with the breed prediction probabilities of greater than 90%. This is the pattern that we got. And once again, this is not admixture, this is only the breed prediction probabilities. And then what we did is, like, we took this data that we have, divided the animals into milk, dual, and draft. And the premise basically was based on identifying the short ROH that were common among the animals of that particular group. So what we did is, this is what is the basic diagram. Among the animals of a particular breed, we tried to identify regions that are common across the animals of that particular group. Means, we are trying to identify a region in the milch animals, probably which is positively selected and is present in almost all the animals. That's why we didn't name it as an ROH, but we named it as a CRLD, that is conserved region of low diversity. Then we went in to identify this, and we found out that out of 150 animals, out of 182 animals, we have got in 150 animals a particular region that is quite specific, which had around 39 SNPs, which is highly conserved in this milk group. Then we were wondering, like, fine, this is the grouping of the milk breeds that we have. These 4 breeds were classified as milk breeds. Then what we had was, like, then we did a milk draft comparison and all the comparisons that we have done for the different groups that we had. And we did a categorical comparison, 0 and 1 and 2 and 3, like milk, draft, and dual. And you can look at the peak that we got in chromatics. And then we looked at the linkage disequilibrium of this region. The region that is very important here is what I was talking about. You look at that linkage disequilibrium, and the CRLD covers this region, and there is an overlap between the region that was identified from the CRLD with the region that was identified with that of this FST and GWAS. And we looked at the allele frequencies. The allele frequency was quite different. And then finally, we looked at, see if you can appreciate the color difference between the allele frequencies. We have actually looked at the genotypes that we had, and we had this particular heat map to identify how different are these different groups with respect to the alleles that they harbor. And there's a clear-cut difference in the color that you identify here with respect to the major allele frequency that is present within this region. Then the most important thing that we did was, like, we thought, like, we have to prove whether this region is selected or not. 2 more minutes. Whether this region is selected or not. So we looked at, and we did an extended haplotype homozygosity, and we found out that IHS for this particular set of breeds, which is in blue corresponds to milk, it's around -2. And then we validated the results again on an independent set of samples with 815 independent set of animals. And then we were not further satisfied with this result. We went into this and looked at the validation HapMap 770K data. And again, we got the similar pattern. And this is the HapMap GIR data that we have already present in the database that is already available freely. Yeah, this is quite interesting. If you can look at, further we looked into how the haplotypes would actually differ at the genomic level. You can see the percentage of difference in the milk, almost like the percentage, the homogeneity among those haplotypes is quite high, with around more than 60%. And we looked at the haplogroup frequencies, and then we finally proved this with Tajima's D. And finally here, like, what we now currently think is, we have proven that this particular region is highly conserved in the milk group, because even if you look at the Tajima's D, it is negative over here. If Tajima's D is negative, means it's positively selected. The same thing was done in height. I'll just run through because it is similar, but the only difference is in the height we had to compare each individual breeds with the individual breeds of height, and then we took all the chromosomes that yeah, chromosome 19, and this is Vechu here, and we again further proved this with the validation of the results, and then we have got a haplotype frequency. You can have a clear look at the haplotype difference. Vechu has got T here, and Malnadi get T, and all the tall animals had C at the correlate. And then the haplogroup differences are quite, and again the Tajmas T. The same thing was done for adaptability. The paucity of time, I'm just running the slides. This is again chromosome 13 that we have identified a clear-cut difference here. And this is a broad idea of what we have for adaptability in hand. And then finally looked at whether breeds, do they have signatures? And like to our dismay, probably is the right word, we identified in 18 different breeds specific regions, but Vechu and Siri are the only breeds where we had that this particular region is positively selected. And the regions that were identified in all the rest of the breeds, they are there in those breeds, but they are not only positive selected on those. And they are only present in Vechu and Siri. Thank you.
Okay, thank you. So there is time for one quick question, if anyone has. Okay then, thank you. Thank you, Dr. Ravi. Now I can invite the next speaker, Dr. Asid Jain from Jabalpur. He is going to present on capsaicin innovation. Please.
Good morning to all of you. I am Dr. Asid Jain and USC Jabalpur. It's my great pleasure to present our work on capsaicin innovation by E64 due to oocyte maturation. Due to oocyte maturation, enhanced developmental competence of buffalo embryo via apoptosis suppression. So this is the overview of my the first presentation. So first we start from the introduction. So oocyte maturation in livestock is a complex process and it is a very sensitive. There are many factors affect the oocyte maturations, and one of them is the capsaicin. So capsaicin, as the follicle develops, gradually capsaicin increase inside the follicles. However, its level become excess, then it induces the apoptosis and they get the important protein inside the oocytes. And this will lead to weaker maturation in terms of the nuclear maturation as well as the cytoplasmic maturation. So this will lead to decrease the developmental competence of the oocyte as well as the embryo. So controlling the capsaicin activity during maturation might be the key to improve the oocyte health. So E64, one of the molecules which inhibits the capsaicin expression inside the oocytes and other cells also. So this E64 helps reduce the capsaicin activity, it reduces the apoptosis, and it prevents the cytochrome C release from the mitochondria. So overall it helps in the maintaining the internal stability of the oocytes. Ultimately it will lead to development of the competence in terms of the blastocyst as well as the quality of the blastocyst. So there are several studies which have shown that inhibition of the capsaicin by the E64 enhance the developmental competence of the oocyte as well as the embryo. So buffalo it has a special sensitivity for the oocytes because its oocytes contain the high lipid contents. It has a lower number of the primordial follicle, high incidence of the follicular atresia. So improve the oocyte protection could bring a major breakthrough for this buffalo species. There is a gap. So as we know, capsaicin has already been explored in terms of the maturation, but it is not studied during the embryo development. So on the basis of the literature, we have hypothesized that if we inhibit the capsaicin with E64 during the in vitro maturation, it will help preserve the integrity of the oocyte, it will reduce the apoptosis health, and it will not on the oocyte, but it will ultimate effect on the during the development. So this will lead to increase the developmental competence of the buffalo oocytes. So on the basis of the literature, we have designed 3 objectives, and these 3 objectives integrated approach to understand cathepsin function in buffalo reproduction, bridging a molecular mechanism with practical application in assisted reproductive technologies. The experiment what we designed, we have selected 2 grades of the oocytes collected from the buffalo strata of the we have culture in the presence of the E64, and we developed in vitro embryo from these 2 types of the oocytes. Here we combine the molecular, cellular and developmental analysis to get the complete picture. We assess the mRNA expression by the RT-PCR and the protein popularization by the immunocytochemistry and DNA fragmentation by the analysis. And we produce the in vitro embryo as per our standard protocol in the lab. And we have assessed the developmental competence in terms of the oocyte maturation, cleavage rate, blastocyst rate, and ICM and T. These are the experiments what we done. First, we have optimized the E64. So we tested different concentration of the E64 and choose that concentration of the E64 which inhibit the maximum cathepsin expression. So this is the first technique, RT-PCR. We have followed as per the method described by the Genomics in our lab. And we choose the total 11 genes which cover the cathepsin, including intrinsic pathway of the apoptosis. So this is the immunocytochemistry followed by the conducted in our lab as per the method described by the Bolwala and co-workers, and analysis as per the manufacturer instruction and Petagora and co-workers. In vitro embryo produced as per the protocol of our lab. We statistically analyze all experiment result. So here we can see that it is a dose-dependent reduction of the dose-dependent reduction in the cathepsin expression. So we choose the maximum dose 10 micromolar in the subsequent experiment. In experiment 2, we can see that there is a higher expression of the cathepsin and pro-apoptotic genes like BID, BAX, CAS, and cytochrome C in the B grade oocyte as compared to A grade. But it was the vice versa for the A grade oocytes. If we see A grade oocyte as well as the B grade oocyte, we found that there is an increased level of the during maturation, either A grade or B grade. When we culture these in the presence of the E64, then there is a reduction of the pro-apoptotic genes. But there was no significant difference in the anti-apoptotic genes like the BCL2 and ISAGB2. This mRNA data was further confirmed at the protein data, and we have found that there is a reduction of the cathepsin B expression in the presence of the E64. Finally, mRNA data and protein data confirmed by the DNA fragmentation apoptotic cells, and we found that there is a reduction of the apoptotic nuclei in the presence of the E64. These are the control. If we look at the embryo, so we found similar finding as we have seen in the oocyte maturation. We have observed that there is an increased level of the pro-apoptotic and cathepsin genes in the absence of the E64. But when we culture in the presence of E64 molecule, there is a decreased level. Here we have found that there is an increased level of the cathepsin and pro-apoptotic gene at the X cell stage of the embryo. It may be because of there is a genome activation takes place at particular stage. So this again we can see in embryo same thing, the mRNA data with protein data, cathepsin mRNA, cathepsin protein reduced in the presence of the E64. Here we can clearly see again apoptotic nuclei found lower expression in the presence of the E64. Now, important outcome of the result on the embryo development, and we found that there is a significant increase of the blastocyst yield in blastocyst derived from the A grade oocyte as compared to B grade. And last important finding, here we can see total cell number increase in the blastocyst derived from either quality of the oocyte, particularly tophoectoderm cells. And tophoectoderm cells are important for the implantation process. So on the basis of the result, we can conclude that cathepsin B expression is closely linked with the oocytes quality and development potential, serving as a marker for the competence. And inhibition of the cathepsin by E64, it will decrease the cathepsin activity, ultimately apoptosis. It will not have impact on the oocyte maturation, but also have an impact on the embryo development. So we can use E64 as a supplement for the improving of the in vitro embryo production. Thank you for highly acknowledgement for NAIP and Dr. Honorable Vice Chancellor of the University, Dr. Hidatta sir, and thank you all.
Thank you, Dr. Asad.
Thank you.
So with this, we come to an end to the Mola, Dr. Kadiravan, Dr. Gautam Sahana, and two best abstracts from Dr. GPVS Ravi Kumar and Dr. Asit Jain. So I thank the organizers and hand over to them.
Thank you, sir. Now this is time to felicitate our speaker. So may I request Dr. Rudy and Dr. Jain, please come on dais, and I request chairperson to felicitate them. Thank you, sir. Now we start our session 3, exploring IoT and artificial intelligence in animal food system. So for this session, the chairpersons are Dr. N.H. Mohan, Dr. R.K. Rawat, and Dr. S.K. Chaturvedi. And rapporteur, Dr. Sanjay Kumar. So I request all the chairperson to come on the dais. Dr. Mohan, Director ICR, NBAGR Karnal, and Dr. S.K. Chaturvedi, former Principal Scientist, ICR, CIRC Virat. And rapporteur, Dr. Sanjay Kumar, Principal Scientist, CIRB Hisar. I request all the chairperson and the rapporteur to come on the dais. So this session we have 4 lead papers and 2 oral papers. Lead paper will be presented by Dr. T.K. Mahanti, Dr. Somen Mandal, Dr. Srivasa Kiran Ambati, and Dr. Suresh Raja Nethirajan. And this 4th one, Dr. Suresh, it will be on online. So I request Honorable Chairperson to start the proceedings.
Hello, good morning, everyone, esteemed delegates, honored guests, and all of our. This present shows how ISAGB has performed so well to organize this event this time. So this is the session, which is the application of tools like AI and others to advance the livestock production and healthcare. So with this now we have the first speaker. We'll go to the Dr. Suresh Raja Nethirajan from online talk from, this is from which, Canada? He's from Canada. So this is the first talk because his time is coinciding with this at present now, as we are left. Hello, Dr. Suresh. Hello. Not audible, huh? Hello.
Sir, are you listening us? Hello. Dr. Nithiranjan, sir.
Hello.
Your slides are visible. Can you hear my voice? We are not getting you, sir. Your voice is not coming here. Your slides are showing welfare by design, harnessing AI and IoT for compassionate and climate-ready livestock systems. We can see your slides, but your voice is not coming, sir. No, it's not coming. Okay, after some time, is it possible?
We have to speak with them.
Sir, Dr. Nithiranjan, are you hearing my voice from the hall?
Hello? Hi.
Hello. Yes.
So is it audible now?
Yes, yes. Go on telling, sir.
Okay, all right, perfect. A very good morning to everyone. Thank you so much for this opportunity to connect with you. Thank you to the conference organizing committee and the Vice Chancellor for this invitation. It's great to be here. We published 3 books just a year ago. These are available in Amazon. If you had a chance to look at it, you could perhaps consider reviewing them. It talks about a variety of the applications of artificial intelligence, how one could go about using AI as a technology, as a tool, as a platform for solving problems. Sometimes we think that is not practically feasible. Recently, in this year, 2025, just a few months ago, we also launched 4 mobile apps. These are free mobile apps available globally for both the Android users and also for the iPhone, iOS users. So maybe I will give 15 seconds, leave the slide here. If you have a phone, please go ahead and use your phone. If you have an Android phone, on the right side you see the green color QR code. If you scan them, you would be able to access these 4 mobile apps. For the iPhone, iOS users, there are the red color QR code. You can scan with your phone and access these Diary, Air Canada, Moonlog, Club Talk, and Cluckify. There are 4 different apps one could download. So the Moonlog is about decoding the language of cows. How do we understand the bovine vocalizations in the way we humans can interpret and understand the context behind it in terms of the emotions, in terms of the stress. Cluck Talk and Cluckify uses natural language processing and artificial intelligence to better interpret and understanding the cues and the dynamism and the meaning behind the context of these different calls. For example, we tend to shrug up, there is some making noise. But actually, each of those sounds, calls have specific meaning behind it, and we can actually create a way in terms of understanding them at this moment. Our long-term goal is to create a 2-way communication toolbox so that we humans can talk and then the black box, the tool can record them and convert using signal processing, semantics and linguistics via natural language processing with the help of deep learning and talk back to the animals, either cow or the chickens, poultry. So that's a long-term goal, but for now, we have the ability, we also have scientifically demonstrated that we can indeed decode and understand the meaning behind them. The app Diary Canada is basically what we have done is we used satellite data from 3 different agencies: NASA, European Space Agency, JAXA, Japanese Aeronautical Space Agency, and collected 15 years of weekly methane emissions across coast to coast of 1,300 dairy farms in Canada, all the way from Victoria to Newfoundland, and come up with a way. So when the farmers go to a farm, he or she, they stand in their farm, open their mobile app, it will automatically geotag, locate that particular location, and they can know their methane emissions. They can also select the past historical record. They can compare within 150 kilometer radius how they have been doing within the province and within countrywide. So it's more of a benchmarking tool. We are giving free for the farmers based on the quantification. For example, if a particular farm has 200 cows right now, but during COVID season they have only 125. There is a difference of 75 cows between that period and this period. How the farm has been doing in terms of emitting methane. Can we be able to measure the milk equivalent of CO2 and the milk equivalent of methane? To be able to produce 1 liter of milk, how much of methane the particular farm has emitted or the carbon dioxide has emitted? That kind of understanding and possibility is available via this app. In addition, we also provide the option of creating information in terms of benchmarking and carbon credit pathway. So as a country, globally, every country has net zero goals. So Canada has 2030, and then we 2050. By the year 2050, we want to offset whatever is being produced in agriculture, especially in livestock. So this is a national level benchmarking tool we have developed and made it as a free tool for the farmers. So please have a look at it and review.
Dawn breaks. Each gentle moon becomes visible, floating like whispered secrets.
Ancient sounds hold meaning. Through artificial intelligence, the secret language of cattle reveals itself. Real-time insights flow. From comfort to distress, the herd speaks to those who listen. From moo to meaning, AI gives every cow a voice, transforming care itself. Introducing Log, real-time cow communication, right in your pocket. Every move, now a message.
What if every with Log, our AI-powered cow translator, it does. Using bioacoustics, Log analyzes vocalizations, grunts, bellows, and hums to detect emotional states like hunger, pain, or social longing. Farmers get alerts when something's off, even before physical signs show. It's like Shazam meets doctor. The log helps you check if your herd can respond with care. Whether you do 5 cows or 500, the log makes sure no moment goes unnoticed. Cow communication is real. The log just gave it a voice. And farmers, a way to listen. What if every
Developed a tool called Diary DGD. The inspiration is from facial recognition technology for humans. But we went one step ahead. The idea here is after we install cameras in the farm system, can we be able to not only identify this is Lotus, this is Jessica, this is Jenny, but can we be able to classify based on their pregnancy status, whether she is a dry cow or a milking cow, and potentially say something about the age of the cow based on the biometric features, morphology, the hunching, and other parametrics. So this is a proof of system tool we have developed as a way to the idea here is right now cows wear ear tags and neck collars. Can we be able to come up with digital phenotyping and developing digital ID and biomarkers without having to touch the cow? Because once we handle them manually, the welfare is being impeded. So the computer vision technology provides additional information, but with the help of artificial intelligence, we can go multiple steps ahead in terms of determining resilience parameters, in terms of phenotyping and so on. Here is another tool we developed for pain check. Pain is an interesting phenomenon. The idea here is it's a protection mechanism. Cows have the ability to express their pain. They may look alike in terms of their facial morphometric, but when we look at the emotion indices, the mental makeup of these cows, we will be able to precisely say something about how do they feel. Because at the end of the day, a happy cow is a productive cow. As long as we keep the cows much more in a positive indicator, we can expect them to be live longer, have an enriched quality of life. But how do we use scientific way in terms of understanding whether they are happy or not? What are the ways we can quantify them? If they go through emotions such as pain, can we be able to tell that in real time? Yes, it's possible through the facial biometrics using 30 key point identification system, but also look at what we call as arbitrary units. How the squinting of the eye that happens from the upper eyelid and the lower eyelid, how the muscle pattern changes. Because they inhale and exhale, there is a clenching happens if they are inserting pain from the upper jaw and the lower jaw. Using artificial intelligence, we go using mathematical modeling and look at the fine scale changes which are not visible for human eyes, but with the help of computer vision and AI, we can actually measure them and quantify and classify in a binary way, pain or no pain. And then we go one step ahead in terms of coming up with multiple scales of pain. For example, at a scale of 0 to 10, 10 being the highest, 5 being in the middle, 0 and 1 being no pain at all. How does this pain scale happens with respect to the context? Pain could be due to n number of reasons, mastitis, lameness, and other related issues. So the context also plays a role in terms of determining the pain. Here is a simple flow chart that shows how we develop this mathematical model along with the AI to be able to quantify and classify pain as a way of measuring emotion from the faces of the cows. Here we can see these two small video clips from our university campus dairy farm. One is pain and one is no pain. So at the end of the day, the goal is, can we create a tool where farmers can use their mobile phone systems and quickly do a check before they call a veterinarian? Because sometimes the rapid detection, how do we go about bring down the time to detecting the pain so that the intervention can be a bit more quicker? So the goal is not to replace veterinarians, but to overcome the subjectivity in terms of assessing pain. A seasoned farmer may be able to say very clearly, using their intuition and wisdom and years of experience, they can say clearly about the pain. But a person from a city or non-experienced person may not be able to say something in a much more accurate manner. So how do we overcome the subjectivity and move towards objective way of measuring pain as a way of emotion? So this tool comes quite handy in determining pain from the faces of the cows.
It started with a blank, not a screen, not a limb, just a blank.
Cows are masters of disguise. They hide pain like a porcupine hides their tail.
But the body always whispers before it screams. You just need the right eyes to listen. We built a machine that watches, not to control, but to understand. We filmed thousands of cow faces across Canadian farms. Each blink, ear flick, and twitch revealed.
From thousands of cow faces, AI learned to read the pain signals hidden in every twitch. With 99% accuracy, we found micro expressions aren't noise, but nature's secret pain signals. We learned the language of tension, flicks, blinks, and clenches. The invisible Morse code of distress. Before pain becomes suffering, a silent alert becomes a gentle touch. Chaos to calm, from guesswork to precision. Animal care just grew a neural cortex. One blank started a revolution. This is not surveillance. This is empathy, automated.
The future, where no animal suffers in silence, starts today.
So we are also currently building a tool called Facebook for dairy cows. The idea here is once we install the multiple cameras at different locations in the ceiling and walls of the dairy barn, can we be able to do a predictive analytics in terms of their behavior? Sometimes a bunch of cows, they are happy, get along with each other. Sometimes they have stick behavior. They demonstrate that aggressiveness, very mild aggression through licking and headbutting and their unique way of behavior. When she goes in heat, she tends to mount on the other cow. That causes a disruptive in terms of on daily management. So we were interested in how do we use this as a way to improve the husbandry system and the housing conditions. So here is an example of proof of concept to look at the different key points. Without touching the cow, we were able to quantify the different, for example, here we have the brown, which is a Jersey breed. On the right side, we have Holstein breed. Jersey is a little more playful, little more dominant compared to Holstein. When she goes for milking to the robotic milking system, Jersey tend to push away Holstein and go in front, although Holstein is in front of the queue. So we were able to observe and quantify and come up with a different way of, okay, this cow number 1 is a little more dominant, cow number 13 cannot get along with cow number 14. So that need to be moved to a different housing system based on the observation and the social network analysis acquired with the help of artificial intelligence. So there are a lot of end-use applications here. We can see here a mounting due to e-stress deduction. Here there is in the water feeding station, there is a little bit of dominance being displayed. There is also, sometimes the cows say, hey, this is my spot, move away, I want to lie down here. So depending on the housing area, we can actually group them, classify them via hotspot-based applications. So what we go through right now is called VUCA, volatile, uncertain, and complex and ambiguous. But from the genomic perspective, resilience can be an indicator of welfare. You know,
Suresh, you have 1 minute to actually summarize the presentation.
Yes, I'll be wrapping up in a minute. Thank you so much.
Thank you.
When we talk about resilience in farm animals, we are really talking about something intuitive. how quickly and completely an animal bounces back after a challenge. Farmers see it every day. Some animals barely flinch at heat stress or social disruption, while others struggle for days. For decades, farmers know how to recognize this difference. But if there is no way we can measure resilience or adaptation capacity with real precision, so with the help of artificial intelligence, now we have the ability to quantify the resilience. Machine learning, with the help of neural and hybrid models, can detect the disturbances early and can map the shape of recovery with accuracies up to 80 to 90%. So, what does this mean? This means, for example, if the animal is going to go sick, we can do things way ahead, at least few days to weeks before, in terms of taking intervention decisions as we move forward. So, Internet of Things and artificial intelligence technology has a lot of applications currently coming up in terms of understanding the emotional makeup, adaptation physiology, health and welfare of these cows. These are not practically possible without the enabling and the adoption of AI. So we are just starting to surface scratch, but there is much more to come. For example, there is a technology called digital twin we have been working on in terms of developing a avatar or a twinning technology to say something in terms of real-time assessment of their digital biomarker and phenotyping that's currently happening as we speak. So with this, I would like to end my presentation. Thank you so much for this opportunity to connect with you.
Thank you, Dr. Suresh Nitirajan. It was a extremely nice presentation, and you have highlighted so important points, because once we have a very productive and very nice animal selected, and the animal also should be happy and to be very productive, so stress-free. So that is he highlighted some points. So if some queries are there, means we can have one or two quick questions from the audience. My coaches. So I have a question. How we are going to implement it? What is the minimum size of the herd which we can implement this program?
So when we look at globally, in terms of scalability for commercial adoption, the context matters. So we used to think that it is not possible for small-scale farmers to adopt. But when I was traveling in India a few months ago, already there is a lot of good adoption in terms of the animal body wearable sensors. For example, there is a sensor you can put in the tail of the cow. It can tell within few hours before the cow is going to call based on the movement of the tail and the acceleration due to gravity. There are also low-cost approaches that are also coming. For example, we developed a technology using paper, just as a paper as a technology. Using a reagent solution, you dip the paper. Paper is made of an origami structure. Based on the color change, we can say with certain quantification whether the Mycobacterium paratuberculosis is present in the urine sample to find out about Johne's disease.
Thank you.
Depending on the context, we can develop the technologies to meet the needs. So it's not necessary that a large capital investment is needed, but of course, government support, subsidy is absolutely essential to be able to avoid the zoonotic possibility and to come up with the predictive capability to keep the animals healthy and enriched manner during their lifetime.
Thank you very much for the wonderful insights. It is indeed true that it's not the question of salary and money, and it is the question of, there is a, 1 second, I was just telling that it's made a practice. The practice has to be implemented in the farm. That's the thing, the practice has to come. Then comes the cost factor. Thank you. I think there is one query from the director NRC, International Camel Research Institute, Dr. Rasool. Sir, please.
I am really fascinated by your talk. My query is whether this approach is made for raising animals, mainly horses. I think where there is they using this approach in raising young horses?
So some, not in a commercial way, but in research and development, here at our university also we look at the race horses and also for breeding horses we do use. When we use the word emotion, we have to carefully look at the word. It starts with the letter E, motion, emotion. The letter E means energy in motion. There is a lot of energy that is constantly moving. So heat energy, thermal energy, blood has specific biomarkers like the neuroendocrine, cortisol, non-esterified fatty acid, metabolic markers. There is also certain electricity, we call it bioelectric energy, constantly movement. For example, the heart rate, the respiration rate, and so on and so forth. So sound energy, vocalization is also an energy. So when we combine all of these energy, we cannot look from one modality. We have to combine all these modalities, sound, thermal, acoustics, the bioelectric, respiration, heart rate, the movement of the animal due to locomotion, point A to point B. When we combine all of this, we can clearly say very good about the mental makeup or the emotion. So we divide that into three different classes: positive, neutral, and negative. Most of the time, in the as of past few years, we have been focused only on removing the negative element of emotions. How do I avoid freedom from hunger, freedom from thirsty, freedom from stalking density, so the animal has better environment to live? And then slowly we transition into, as long as they are neutral, they are okay. But now the focus is on how do we enrich their quality of life? Can I provide some playful material for the horses, even for the chickens or for the cows, such as brush, so that we change their mood, the mental makeup into more exploratory in terms of positive quality, positive behavior and experiences. So that completely changes the quality in terms of the emotional makeup. The different energy, the values and threshold also changes. That is directly proportional to the resilience, adaptation capacity, and eventually the longevity and the length of the life. So in terms of application of sensing technology for measuring emotions, yes, there has been some work currently been happening in our lab, and also the private small industries, they have been using a variety of technologies to quantify and measure emotions from horses and other animals.
Thank you.
Thank you very much. So, if not, no other queries are there, and we appreciate you joining the thing. Thank you. And now we would like to invite Dr. Tushar Mogundi for the lead presentation on the topic of precision livestock farming using sensors and artificial intelligence, optimizing resources and productivity. This is a very, very important lecture we are going to have from the distinguished scientists. And as we might be having a highly productive animal, but the other resources for supporting the production also have to be there. So one of the way of, if we are not able to generate the resources, we conserve the resources. So the precision farming in fact helps in the rationalizing the or assessing the actual needs of the productive needs of the animal and based on our resources. So it's a very important lecture, sir. Floor is yours.
Good morning to all, honorable chairpersons and reporter. I will be talking about precision animal management. Okay, so this, you have heard this, there are 2 lectures already being done. So I will be talking about what are the requirement of Indian context, because we are a very vast country with multiple breeds and large number of animals, and data collection is not systematic. So whether we are ready to adapt to the system or not, I will be talking about this thing. So if you are talking about Indian livestock sectors, per animal productivity is low compared to global standards. Effort to improve genetic did not succeed as expected. Means we are trying to do a lot of things to improve the productivity. genomics selection platform, protein testing programs. Now, in last 10 years, it is being implemented, and not to all the breeds. It is very, very in nascent stage. I will talk about this. Scope of commercial dairy farming is encouraging because the smallholder farmers are leaving now, and our demand for milk is increasing. So definitely we'll go for commercial farmings. It may be 10, 20, 50 animals, and it will be a transition we'll have to adopt. Otherwise, we'll not able to get. And the small farmers, can we take this technology to the small farmers so that they can also have all the technology in their home with 5 animals also? What Sarani was asking for, that whether we can reach to these persons, those who are having small farmers. So the profitable business ventures requires proper planning and careful execution. The planning and execution in our small farmers are not happening. So why you cannot do this thing? This animal performance recording in the field condition is not happening. So if you are talking about data, AI, algorithm, or any type of machine learning, so this requires beginning of data, then we can make our health management good. We have gone up to vaccination and preventing diseases, but not other type of problems which we are having, because we don't have everyday data from the animal. Feeding, because the input costs are the major input costs are the major things. So can you optimize feeding so that the productivity related to feeding, if you are not doing, then it is not profitable. Then only you can do this productivity and genetic improvement. Whatever you are doing, you are not able to express the genetic improvement because you are putting very good quality semen, but the calves which are coming, they are not producing as per the expectation, because we have problems with health management, feeding management, and all these things. These are the problems with us. So that problems performance are recording because large number of animals scattered in rural areas, animal holding is low, cow and buffalo multi-breed with different economics parameters, non-availability of IT tools and infrastructure suitable for farmer success, as well as vet and planners. I am talking about the 3 people they need, because whatever you are doing, you are not reaching to the farmers, they can utilize this data. Low computer literacy animal workers and the investment of performance recordings. The government has to invest, it requires its requirement is there. It is a very vast country, multiple animals, it has to be sectorized. My point is that we have to go for the animal performance recording first, then only all other things will come. So problem in service delivery in animal science, this is fire brigade response when damage is already done. We are doing a lot of things to salvage the animals, salvaging the animal when there is outbreak or any disease, dystocia, repeat breeding, all these things, but we are not going to preventive things, we are not going. Farmers perceive heat, pregnancy and incentive as 6 animal treatment is the everything for them. They are not knowing what are the other things, can be preventive things are there. So, and it is a discontinuous, they never get all these things at every time. So this is not continuous, they are discontinuous, they are not continuously available for them. So I will give you an example, we are very late, because these are the countries when we have started performance recording, you can see these things, and we have started 2015 only with enough, and now with other programs we are now trying to do this thing. So what are the importance of records? Proper management is key to start any enterprise. Requires sound decision making, a record that helps farmers pedigree, production, reproduction, feed, health, cost of inputs, selection animal with better genetic producing ability, better future heart. So when you talk about genetic selection, without optimum productivity, you cannot do the genetic selection properly. This data-driven dairy production system, when you talk about now this precision farming, so it is a data-driven. Without this thing, it cannot be precision. Data dealing with increased information, increasing the complexity of decision. That is the point coming. When without data, we cannot do this thing. When the data will come, it has to be increased complexity in decision. There this AI will come, there the algorithm will come, there this decision support system will come, there all terms are there that people are using now this thing. It is actually the decision support system or data analytics, applying professional approach. My point is that the data which are coming, it has to be looked after by professionals. There is a lot of requirement of will be professionals, whether they are ready to do such type of things in in our veterinary curriculum and all these things, they are ready to use such type of data for decision making. So that is a challenge. Applying to management, decisions of artisan and useful and fulfill expectation of the farmers. We have to do this thing. Then only we will be successful. So these are why data is important, because traceability, people are asking that where from the milk is coming, where from the meat is coming. Precision services predict the management action to be taken in the providers and the farmers. Diagnostics, which is coming, that diagnostic is the things lead to the identification of fertility, production, health problem, well-being, clinical signs, preventive actions are all possible. But if you are collecting such type of data, it can be collected by human being or by collected by people or by sensors. What we are talking about, sensors mean here camera, video or wearable sensors are there. That preventive service has to be implemented when we are talking about sensors and all these things. Then only we can improve the use of drugs, hormones and supplements. We are more dependent on drugs, hormones and supplements to make all the problems solved. But my point is that it can be solved if you have a good data collection and the optimum use of this thing can be done. So these are the forecasting we can do these things, high quality product we can make, problem tracking we can do these things, service quality appraisal, evidence-based service, all these things can come when the data are there. Without this thing we cannot do this thing. So these are the very hard management softwares. I am talking about that enough and all these things are very big softwares. They are not useful for the farmers. We need softwares for the small farmers, software for the medium farmers. They should collect the data and the genetics or the breeding data that can be sourced to the other big organization. But the basic requirement of the farmers is daily work, what to do, what are the vaccination schedule, how are the treatments, what are the feeding. All these things are required for having such type of very hard management software. We are not talking about these things. Once we can develop such type of things and give to the farmers now, Android-based such type of programs are available. My point is that we have to do these things then only. So when you are getting data, that data visualization makes people more interesting to use. Without the data, if you see, then you cannot predict anything. But when you represent in a graphical form, that gives a very good insight that what is happening and all these things. So these are the type of things I want to show these things, that when you have such type of data, every detail about the animal, when it was vaccinated, when it was made AI, when it calved, when it is expected to come to heat, all these things, if it is in front of you, then only you can do all such type of things. So this is my first part, that I go for this. What are the things we are doing in NDRI? So we have done this software after collecting the data. For growth module, it is small things, but this is the most important things. If your animals are not performing or growing in right things, whatever the genetics you are putting, it will not be expressed. Can you do these things? Small thing. The animal should grow minimum 400 to 500 grams per day, means around 12 to 15 kilograms. Can you measure all these things, and it can give an insight that yes, your cows or heifers are growing fine, nicely. Lactation car modules, and you can do these things. There is a lot of research and all these modules are available. But can you utilize this thing, that whether my animal next lactation is following the lactation which is followed before or not, or if there is any problems because the animal is having mastitis, whether it is predicted, means standard lactation, how it is deviating from there you can predict. This is followed by the abortion, how it is coming down. So these are the things when the biometra is there. So such type of things can be correlated, that the predicted model or this animal's predicted model and what is happening from there, we can judge what type of diseases animals are there. These are all, my students have worked on these things, that original data, and we have predicted all these things. Metritis, so this is how it is happening from the beginning of things. So then we have collected a lot of whole year data. Can you predict mastitis from pH, EC, collected from the animal with milk yield? So we have collected all this data, and we could make this thing physiology-based module we have developed, where you can predict that beyond this thing, because it is a huge set of data of near about 400 animals for whole 1 year. We have collected all this data, then we have developed these modules which can predict from this only pH, and because for every individual animal, you cannot predict. If you are having 7 days same animal data and the particular day with any change is there, then you can predict that the animal is going towards mastitis or not. Absolutely you cannot do these things. So these are the things we have developed. So this is another one is that healthy and mastitis use of ANN models. This is the neural network models has been developed, pH and conductivity. So then how to collect the pH and conductivity. So we have developed an instrument, it is a smart bucket, we call this thing, and Dr. Bandapadhyay sir has given this idea. So it is a smart bucket, it can collect all the three data, temperature, pH and conductivity and weight at a time to collect all this data, and you can have this basic algorithm behind, and it can predict if you can collect such type of data. So this is another model we are now developing so that it can be marketable. Our things can be marketable by somebody can take. This is also going on. Our engineers from there. So then we have gone for doing this thing, thermal imaging. The thermal image data can be used for prediction of mastitis. So under temperature in Mura, we have done all reads to find out that how it can predict very accurately, 93% accuracy you can predict all data. So, but here the problem is you have to collect two-side data because the others are having 4 reads. So you have to collect these quarters-wise photographs. Then only you can do this thing. So these are the things required. So automatically, when the animal is passing in a parlor row, you can collect this such type of data automatically. So the algorithm can be developed to predict automatically that yes, this animal is going towards mastitis. So these are the seasonal also because we have a lot of variation in the temperature. So the thermal image when it was taken, they are affected by the season. So you have to collect data for different season to develop algorithm in different for season. So then only it can work. So these are the things we have done. So these, then we have thought of why you will take the other temperature, why not these milking tubes which are attached to a milking machine, because after sometimes the milking tube get heat up in a different way in a mastitic animal because milk temperature increase by around 2 degrees centigrade. So that can be captured by such type of things. So short milking tube thermal analysis in the mastitis in Mura buffaloes, you can do this thing. So that identification is stress from this thermal camera because thermal camera is little bit costly, but it is a one-time investment in a farm and it can be used for heat detection also. So this data we have collected for near about 11,000 data set we have collected, and stress, non-stress it can be differentiated because these are the small farmers. I am talking about where there is a small farmers, they are approachable. We can go to touch that animal. You can take such type of data. You can make true positive, false positive, and all such type of. This prediction model we have developed, confusion matrix, and you can see this thing 91% precision can be done. So then you can do this thing pro-stress, stress, stress by using the activity parameters. Activity also, these pedometers we have developed with IIT Delhi. So we are utilizing this thing for collecting the activity of the animals, and we can, the major challenges with us is the battery power because it is working only for 21 days. So you have to change the battery or you have to recharge this thing. So that is a problem. Still now we are not able to solve this thing. You can collect this data wirelessly without disturbing the animal, and you can see this thing in stress. There is an increase of around 200% in the activity, and from there you can predict the stress. So what are the threshold? Because when the activity increase, at what threshold you can find out to get this accuracy is more? When you are talking 20%, so you can see the sensitivity, specificity and accuracy. So when you are going 30%, 40%, so it found that in 30% is almost best things that if there is an increase in 30% from the 3 days average before stress, it can predict the things. And on that basis, that stress is called this onset of pedometer stress, which is almost around 3 to 4 hours before what we say this thing, the animal coming to stress. So that you have to, on the perimeter stress, you have to synchronize at when to do AI. It is around 5 hours, little bit late. If your perimeter stress is giving you the data, so you have to do AI little bit. So these are the behavior indicators. We can do this thing by feeding behavior. You can predict metritis. Then you can do this thing, feeding time. Then lameness also, it is a very big problem. Mastitis, then lameness also, we have detected by IR camera, we have detected all these data we have collected, and we can predict the animal and mastitis. The activity from this, we can have this activity. From the activity, we can detect the lameness 10 days earlier, and you can see this thing, how much milk loss are happening because of things. You can predict the pasteurization time, because in a large farm, when the animal are calving, it is a very difficult things to predict. So by image analysis, we can do this thing. In buffaloes also we have done these things. So we have developed a model, DSS model, to predict also. It is 4 hours before we can predict that yes, animal is coming to things. Then we have done this thing, phenotypic, similar type phenotypic animals can you predict? For my idea is that the AI technician they can do this thing, right AI. If the semen is there, they can predict this is a sahiwal, so they can use sahiwal animal or tharparkar, because they don't know all these things, it can be used. So these are the image processing part. We were able to predict around 86%. So work is going on with more data sets. Citizen requirements is there. Evaluation of testicular blood flow. So this is also the body weight can predict the things. You can temperature, humidity, data collection. You can do this misting, that milking, water intake can predict the diseases. So this is the power BI-based method of predicting the stress. So these are conclusions. My thing is, many technologies are available, and to dairy producer and researcher for detecting stress, illness, calving time, and rumination time. Technology must also be economically feasible. My point is there that economically it should be fit within the management styles, and how a research and producer perception, detection accuracy, and economically feasibility of on-farm daily health monitoring through the precision dairy technology are in demand, but the cost is a major factor in Indian context. So they are available, but they are not able to use these things. So these are my projects. Through this thing I have worked, and I thank my directors and all our farms workers for doing this thing. Thank you.
Thank you, Dr. Mohanty, for a very informative lecture. In fact, the talk has opened so many discussion points, from stress control to mastitis, lameness, different diverse topics are there. But I can, before I make any comment, it is open for the House to make any questions or any queries to the speaker. If the House doesn't have means, then I request questions.
I will repeat my question that I asked in the morning, is that you are doing all of this to diagnose, for making your management system easy, but are they heritable?
Yes, I will talk about two. We are now working one very heritable trait. We could find out.
No, that's just your quick comment.
Yes, it is heritable.
Can we select the animal against lameness?
Yes, it can be done.
We can select the animal
can do
against
this.
lameness?
Yes, lameness and heat, postpartum heat when it comes. It is most heritable reproductive trait. The animal which come to heat.
That means the service period.
Yes.
Service period.
No. First postpartum heat.
First postpartum heat.
When it is very heritable, it is around 2.28.
28%.
0.28, yes, 28%.
28%
Yeah.
Oh, that's very good.
So these are the things coming up. So the sensors are.
Others might be having questions.
Okay,
Thank you very much. Yeah.
Thank you, Dr. Mohanty. Very small query I have. Many of these softwares we have been shown, it is Android based, or can we?
So now, till now, we are developing the front end now. We are going front end for these softwares. Now that's for IR camera. The front end has to be developed, because our database, we have near about 25,000 database for heat detection, and for mastitis detection and lameness detection. But the front end has to come because you have to upload the photographs. It goes to the server, then that work is now pending. It is only 4 years work we have done all these things.
Any suggestion to the audience regarding the storage of data, because all this will generate large quantity of data. So where do we propose to store that? Any suggestions?
So I'll give you this reply. The ICR server was our main data centers, and it is
we don't discuss that.
So we don't have that type of data set. It is very difficult. It is costly. I'm talking about, that's why I'm talking about need a lot of infrastructures. Need a lot of infrastructure.
We might need a data structure and the storage facility as well as a high computing facility for the quantum of the data. Thank you. I think we have a announcement from the organizers.
Good morning, sir.
Proceed.
Thank you, sir. We have an announcement that we are very happy to announce that Dr. P.V. Salim, Secretary Government of West Bengal, is with us, and we as organizer want to felicitate him. So in this connection, I request our Honorable Vice Chancellor, Dr. T.K. Dotto sir, to come on the dais, and Dr. P.V. Salim sir, please come.
Well, friends, it's a special occasion when we get one of our earlier colleague, in fact, a veterinarian, Dr. P.V. Salim. He is a specialist in animal genetics, so he has a context of coming to this particular conference. But Dr. Salim thought of trying his luck in IAS, and now he is a kind of a who's and who in the West Bengal government bureaucracy. And his work touches on very difficult subjects like power generation and minority management and this and that. Dr. Salim, we are very happy to have you here. And maybe
no, I'm extremely grateful. I didn't expect this. I didn't want to be on the stage. I just thought my colleagues are here, my college mates are here, my friends from IBR are here. Mohan is a batchmate, colleague. Dr. Dev was my guide mentor at IBRA. So I'm here as an audience. Thank you so much.
Thank you, sir. So I request chairperson to start the proceeding again, please.
Thank you. We shall receive the session. Now we have eminent speaker, Dr. Somen Mandal. May I request Dr. Somen Mandal, PS Human-Centered from Human-Centered Robotics and Cybernetic Group, CSR CMRI, Durgapur, please. He will be talking on the leveraging AI to IoT powered wearables for small cattle holder systems. So it will be very interesting to hear from you, and the floor is yours. Dr. Shauman Mandal, please.
So, a very good morning to each and everyone present here, and I am extremely thankful to the organizing team for giving me an opportunity to interact over here. And today I am going to talk on leveraging AI IoT powered wearables for small cattle holder systems. So the previous two speakers had given us a very nice introduction regarding various kind of systems that are available related to AI and IoT. However, in my talk, I will be specifically focusing on small cattle holder systems. The reason is that in Indian context, this actually small cattle holder system is extremely important, because large number of farmers, they rely for their actually occupation on these actually small cattle holder systems. Next slide, So I come from CSIR CMERI Durgapur, and this CSIR CMERI is an apex R&D organization for mechanical engineering under aegis of CSIR, that is Council of Scientific and Industrial Research, which has 37 number of laboratories all over India. And being only the national level R&D institute in this field, CMERI's mandate is to serve industry and to develop technology, so that India's dependence on foreign products is substantially reduced in strategic and economic sectors. So at actually CMERI, we do agricultural machinery, we do industrial consultancy, we do manufacturing and material processing, we do mechatronics, we do energy and environment, and we also work on these kind of things like actually development of some IoT devices for cattle activity monitoring and for actually other contingency. So this work, this is the acknowledgement, and this work was conducted in actually collaboration with WBUAFS, because we are the developers of the technology, and they have actually deployed and tested it, and after that this technology has long gone into the market, and we also have given this technology to two different industries who are currently the users, and we do sell these actually technologies to NDRI Bangalore right now for their purposes. And regarding the affordability of the technology, we do sell these actually wearable devices for cattle at just 10,000 rupees. So yes, it is actually affordable right now. So coming on to the objectives, it is all about understand the requirements in small cattle holder systems and what are the IoT enabled cattle wearables. I will give a very brief introduction, and regarding that I would also be talking regarding cattle health, cattle activity, location and stress behavior determination in cattle. So coming on to the introduction, I will not speak more about the cattle, small cattle holder systems, and I would directly go to the requirements. So in actually small cattle holder systems, since individuals or families, they actually manage relatively small number of cattle, primarily using family labor, they have to leave these cattle for grazing. So the cattle location and the cattle safety is an important criteria in this case. And also, as the previous speakers already spoke, that the cattle activity, rumination and the standing behavior, these can be an indicator of the stress behavior and other diseases. And the cattle body temperature is also an indicator of cattle diseases. So we actually require a cattle wearable device that can measure the physical parameters like cattle location, as well as actually body temperature and actually cattle activity. So these are the considerations that are required for design of any kind of wearable cattle holders, actually wearable for small cattle holder system. Cows usually move constantly during grazing, and they can cover around 13 km per day. So regarding the location and transmission, what kind of modules we are going to use over there? Selection is extremely important. Gestation lasts approximately for 9 months, and for the stress behavior identification, we have to take the consideration in our actually sensory systems. And apart from mooing, cows also use the placement of their head, leaves and tail in order to communicate. So using the sensors and the physical parameter, we can easily actually determine what is the state of the cow. So coming on to the existing cattle wearables, these are few of the cattle wearables in actually market. So these actually existing cattle actually wearables, they do real-time monitoring of cattle health and behavior, and they enable early detection of disease, stress and other issues. And they also contribute to the reproductive management. So the first device that is moved on from Stellabs Technologies, and actually this is an Indian technology. It is a startup in Bangalore, and actually this is a kind of pedometer for cattle which can detect heat and actually various disorders based on their activities and resting behavior. And one more actually device, it is actually smack stick. This is actually a device which is usually inserted in the rumen of the cattle, and it is actually for accurate actually rectal temperature and pH. Then there is actually another device that is cowlar. This is a kind of strap
that is designed to comfortably fit around your cow, and actually box that can take the actually cattle activity and cattle behavior. And one more system is there, that is a wearable ear tag for the cattle, which is called the smart paddock. And these all actually technologies are developed in USA and Australia, except the first one, which is being actually developed in India. So these are the few gaps which we found in these actually technologies. The first gap was limited primary range. Most of the devices, they can be used in organized farms. And as I said, that cattle can graze up to 13 kilometers per day. So we need something by which we can do some kind of universal transmission. And hence actually what happens that in actually current systems, there is requirement for repeaters. And these makes the system extremely costly. And actually regarding Indian context, the current devices are not very effective in Indian context, where location stress and FMT are the actually main concern. So coming on to our problem statement, which is on the development of an intelligent IoT powered cattle wearable targeted to small cattle holder systems in India for determination of cattle location and activity classification that are indicators of stress identification and detection of foot and mouth disease. So coming on to the major, actually why we are doing this kind of R&D, because AI and IoT is an emerging area, and this actually smart wearable research is currently being practiced all over the world, and there are a number of government objectives like Rashtriya Gokul Mission. So we are able to grasp actually some kind of funding for that. And coming on to the R&D publication, the first publication that my group made in this actually domain was actually 2022, and that publication reached a citation of 70. This shows that this kind of activity or work has some kind of global reach. And also there are opportunities towards startup creation or technology development. And for Indian context, since nobody has actually focused on FMT till now, and these kind of actually devices for actually unorganized farms are not available. So this actually gives us more impetus. So actually coming on to the method, this is the block diagram of the actually wearable. We have a GPS model, actually temperature sensor, sound sensor, the inertial motion unit. Then there is BMS, the battery module microcontroller. And finally there is a SIM-based GSM module which can transmit the actually data to the server from anywhere. So we actually usually make two types of modules. One is the neck module. So the first one is the neck module, and the second one is a leg module or the pedometer module. So this is the kind of prototype development, since what happens that whenever we are going to develop some kind of technology or prototype, it has to go under various number of stages from development to testing. Because there is a term called technology readiness level, which is called TRL. So in our actual technology development, we usually focus on these TRL levels. So TRL 1 to 3 is we are conducting some kind of actually basic research in the laboratory. 4 and 5, it is all about the lab testing. 6 and 7, we are going to the field or operational actually environment. And actually 7, 8 and 9 are actually launch. So here actually we have reached up to a technology readiness level of 6 or 7. So initially we have developed all the actually parts in India only in our laboratory, and you can see that there is a neck module and actually leg module, and this can be charged within a standard actually cell phone charger, and we have got a run time of around actually 16 hours and a charging time of 4 hours. So coming on to the experimental location, and these experiments were conducted in WBUAFS Mohanpur campus, as well as some of the experiments conducted in Namkhana and some in actually Durgapur. And we have a dedicated server at CSIR CMRI Durgapur, and these are few of the housing conditions. So coming on to the design of the experiments, initially the raw data was stored in the server, and for these experiments we have logged the activities by a field supervisor, and we were also capturing the time-stamped video for our experiments. And we have actually classified the activities of the cattle. The first one is standing, lying, standing and ruminating, lying and ruminating, walking, walking and grazing. And of note, I can tell with 100% confidence that no other technologies are available which can distinguish between very close activities like standing and ruminating. Because here the features are very, very close and near. So coming on to the activity classification, since I said that we have classified the activities into 6 numbers and we have 5 different sensory data and we have collected these actually data and then we have tested using XG based and RF based actually classifier and we have done actually classifier based performance comparison. So as we all know that whenever we are capturing the data, so after that we have to find out some of the significant features in order to implement the ML model. So few features which we found were actually standard deviation, the mean and the RMS. So for this actually acceleration, the temperature, the speed which we got through GPS, we actually clubbed this. So it's a kind of multi-sensory approach in which a number of sensory actually data were taken into account in order to classify the cattle activities. So this is the classification report and here we actually found for the standing and the ruminating, the classifier have actually performed very well. And then actually coming on to this thing, we have shown here the construction, working and development of a neck-mounted intelligent IoT-aided device for cattle activity monitoring inside the barn as well as the pasture. And the device is capable of recording acceleration, temperature and walking speed information of the cattle and transmitting the data directly to the server over SIM-based GSM module. And we can discriminate nearly similar activities such as standing and ruminating. And we have found a classification accuracy of around 97%. And standing behavior of the cattle can actually assist detection of the stress behavior in a more effective way. And also the health conditions such as FMD, lameness, mastitis, etc. can be detected. So these are the selected references. And today it's actually Children's Day and that's actually 14th November. So I put this actually picture. So just for the interest of the audience, I have shown that what we are actually currently doing or after that what we have done. That how we have taken care of the stress and FMD. We first went for the actually observations and in stress we found that the rumination decreases, the standing behavior increases and the cattle becomes actually restless and the body temperature increases. Similarly for FMD, we found that the rumination decreases, the standing behavior decreases, the restlessness also decreases and the temperature actually increases. And from this, we have actually taken the data from our wearables. That what is the variation of the data from pre-stress to stress. And we have found that there are statistically significant variation in the neck acceleration whenever we are going for this actually stress behavior. However, whenever we actually went for the temperature variation, that was the actually the body surface temperature, we couldn't find very statistical actually this actually variation. And then we went for the stress and actually FMD evaluation using actually ML. So the actually determining parameters were the rumination time, the standing time and the cattle activity index. And from here we have found the actually classification reports for the stress and we also conducted FMD experiments. And from there we found that from stress to non-stress, for our actually population, the standing time, there was an increase of 44%, the rumination time, there was a decrease of 22%, and the activity index of the cattle increases by 42% for stress to non-stress change. And for FMD to non-FMD change, the standing time, 71% there was a reduction, and for rumination time, it was 31% actually reduction, and the activity index, it was 51% reduction. So we have actually determined the threshold for stress and FMD. So we have concluded that for our sample experiments, 20% decrease in rumination and 40% increase in standing behavior and actually cattle activity index can be an indicator of the stress behavior. And for the FMD, reduction threshold of 30%, 70% and 50% for rumination, standing behavior and activity index could be an actually indicator of FMD. So thank you. We still have one actually minute. I would be taking some questions. Yes, sir.
Yeah, thank you, Dr. Mandal, for finishing before time.
Not a question again, this is a curiosity.
Yeah. And yeah, it is open for the house for any queries, quick questions we can.
I am doing it, Mohan. And you are talking about the rumen sensor, right?
Yeah.
So this is some kind of a eatable sensor.
Yeah.
Where probably the life of the battery, that sometimes matters.
Yeah.
Because I make it eat and it survives there for 3 months, 4 months, 1 year, whatever. So you are an engineer.
Yeah.
So can you tell us that how long the battery can survive?
Yeah, actually the battery can
Is
go ahead.
it lifelong?
It can go up to 4 to 5 years. Okay.
4 to 5 years.
That has been tested and also reported. Okay, in their actually seat.
And this
Company actually seat.
This transmits signal.
No, it actually doesn't transmit signal always. Okay.
Okay.
You actually need to take your cell phone, the device. You have to turn on the Bluetooth. You have to come near. Only then you can get the reading because
Come
it is
near. I have
yeah,
to come near.
Yeah.
So then that is labor intensive.
Yeah, that's actually labor intensive.
Very ideally, I would like to have a system that is capturable in a distance of say at least 500 meters.
Yeah, definitely.
Somewhere.
Yeah.
So you are doing something about it?
Till now we haven't gone for the pH or the human temperature. Okay, since we are actually currently going for the non-invasive techniques. Okay, because that will be invasive one. Okay, and that would need more amount of studies. Okay, but since you have told us, it has inspired me. So I will actually definitely look into it.
Human sensors are probably not very intensive. There is a device by which you make it to eat.
Okay.
And this go and sits in the rumen for whatever time you want it. You cannot take it back.
Yeah.
But then there are issues there.
But we have to be extremely careful that it actually doesn't pose any problem in the gut of the cattle.
There are many things. For example, this sensor, it actually floats in the rumen.
Yeah.
Right. But the actual methanogenesis, methane, this happens in the layer of the rumen. Right. So these are some issues, and then partitioning the fatty acids, volatile fatty acids into propionic, butyric, and acetic. So they are things available. So please, if possible, enter into this.
Sure, sir.
Very beautiful area.
Sure, sir.
And lot of money. If you talk climate change, the funding agency eats it.
Okay, sir. Okay, sir. I will.
Thank you. Thank you.
Yeah. Oh, there are 2 more queries. Please.
Thank you, Dr. Mandal, for an excellent presentation.
Thank you.
I have some doubt only. See, all these measurement devices you are attaching to the animal. So it is an external device on the body of the animal.
Yeah.
So it's an invasive procedure to me. Can we have any control data measure the same parameters by non-invasive methods so that we can say that this data is authentic? Why I am asking this question is, decades back, when telemetry was introduced in wildlife tracking, they have found initially that the behavior of the animal is changed in the initial phase of this. Whether such things are happening on this device. Can you elaborate a little on this?
Yeah, actually, whenever we develop this kind of devices, there is a term named as RSSI. Okay, we have to see that what is the value of RSSI for the signal which we are transmitting. Because if the RSSI value is too high, okay, it can really lead to discomfort of the animals. Okay, for instance, whenever we are going for some kind of cell phone, we are buying. Okay, good companies or very standard companies like Apple or Samsung, they will definitely write that this is the RSSI for their frequency or the signal. Okay, but actually few companies do not actually note that. Okay, and if we can control that RSSI, since we are also wearing this kind of smart watches here. Okay, which is actually GSM enabled. Okay, so I don't believe that cattle will have much problem if we are controlling it like that.
Thank you. Mrs. please.
Dr. Mandal, I saw that you are working on 2 aspects, right?
Yes.
One is stress and another is FMD.
FMD, yeah.
FMD is a serious disease.
Serious disease.
Affecting the economics of a large area. You said that you have found out a threshold to identify that if it is 20%, that's 70% and 30%.
No, that's the reason that
Just
whatever
I am
we...
not end the question. The question is, how could you come to the conclusion that this is the threshold to identify an animal is having a FMD or not? Why not the sitting behavior, the sitting, standing, and all behavior changes in any disease condition? So how come, what are the threshold, and what are those controls for FMD and non-FMD to come to this conclusion?
Yeah, actually, I would like to mention here that for this FMD based cattle, okay, we have selected one hospital in Asansol region. Okay, during this actually just after the July, when this actually FMD happens during the rainy season. Okay, so there actually FMD sampled cows we have got from there, 12 number, and we have tested on that. Okay. So,
I think I
so
got
it
it.
was
I got
actually
it. So
controlled.
you have to
Yeah.
make statement
Yeah.
on a threshold for FMD,
Yeah.
because this is something will go out. So therefore, all I think precautions, or you have to consult the FMD institute. There are two, IVRI and NISAR, and then there is a DFMD, National FMD Institute at Bhubaneswar.
Okay.
So please consult the experts coming into these thresholds,
Okay.
and conclusion for FMD at least.
Okay.
At least the National Institute of FMD at Bhubaneswar.
Okay. Okay.
Thank you very much for the, any other queries from the chairs? I have one query. He has, in fact, you have very rightly pointed out the various experimental results, but this is really a matter of concern, Dr. Mandal. It's a matter of concern that how much the animal will be able to wear, because the size of the instrument is 10 centimeters into 8 centimeters into 3.
Yes.
So that is not that small. So and in collar, if you are putting it, as you can understand that when we purchase a new shirt or new dress, we also have felt sometimes see uncomfort, no? So that is a really matter of quite a concern. Second is, I'm just highlighting the points emerged. The second one is that there might be several confounding similar signs. Temperature can rise to and many diseases, not only stress, many other things can happen, of course, is it not? So these also might be a factor which can really.
Yeah, that's the reason we are focusing on multi-sensory approaches, so that we can get a number of data from much different sensors. Okay.
Okay.
So
that is true, but the size of the product and RFID also, as he has told very rightly, I remember on tigers, when RFID was put for tracing the animals and all, they faced this kind of issues. Anyway, animal behavior might change. Anyway, thank you very much.
Okay, thank you.
So, thank you very much, and now I have the honor of requesting Dr. Srinivas Kiran Ambadiputti, Professor of Biosciences and from IIT Roorkee, and he will be talking on the smart wearable sensor system for monitoring buffalo health. Dr. Srinivas Kiran Ambadiputti, please. The floor is open and it's for all yours.
Thank you very much.
Thank you very much.
Firstly, I'd like to thank Dr. Datta, Honorable Vice Chancellor, for the invite. And up until now, we have been talking a lot about cattle, and Dr. Mandal showed about smartwatches. So along with Fitbit, smartwatches, smartphone, there's been a lot of talk about how we can identify, for example, I'm sleeping, talking, and cardiac activity. And more recently, Gabbit. Ranbir Kapoor has been doing a lot of ad about that. So, but this is a fitting talk. We just pivot from all about cattle to buffaloes. They are no way similar in terms of their behavior as we compare cattle or cows in particular. So I'm going to talk in the next 15-16 minutes and try and impress upon the policymakers sitting in this room how different are buffaloes and what we have been doing as a journey for the last 14 months. And Alfred is here. He's the person who has funded us. So to CIRB, in fact, here is the co-PI sitting here. All right, so buffaloes contribute significantly to the Indian dairy economy, in terms of milk production. They are pretty rugged animals, and they live in diverse climatic conditions. And I'm sure they convert low-quality roughage into high-value milk, including protein, fat, macronutrients, as compared to a crossbred cow. But unfortunately, they have received much less attention scientifically and technological innovations. But considering their resilience and their contribution to the Indian dairy, as well as the sustained milk production to the farmers, now I think is the right time for the policymakers as well as the scientists, not only in India but around the world, wherever the buffalo population is, I think to focus on buffaloes and how we can promote and sustain their milk production, including their sustained reproductive efficiency. All right, if you look at the publications coming out from different parts of the world from scientists, and if you look at take an informed decision about the buffaloes using the analytics, there's a lot of data being thrown out at scientists around the world. It has been said that buffaloes are superior in terms of their disease resistance, stress management, and extended lactation cycles. However, there is very, very limited behavioral and physiological data that is available to the scientists so that we can optimize their feeding, breeding, and health perspectives. So it is very important that we use the empirical data, whatever the farmers observe or humans observe in terms of how they are behaving, and to convert into an actionable and quantifiable data so that we can take informed decisions. So here we are. Here I'm going to talk about why we want to use sensors. In spite of all these advantages, buffalo rearing has lagged as compared to a crossbred cows. As you can see on the slide, there are a lot of disadvantages, a lot of limitations in terms of poor heat tolerance, longer calving intervals, higher initial cost, and in terms of milk, people prefer cow milk as opposed to buffalo milk. But in the interest of the presentation today, I'll particularly focus on delayed heat or the estrus, or this is a very common problem in buffaloes. It's called as the silent heat. So that is why I think it is imperative that we use the modern technology, whatever is available to us, to see how we can improve or detect this delayed or silent heat. There is a perceived complexity and lack of decision support tool particularly to work on buffaloes and see how we can detect or how early we can detect based on their activity the estrus or the silent heat, so that we can use the artificial inseminations. All the veterinarians, when we talk about AI, the first thing that comes to mind is artificial insemination, not artificial intelligence. But now I think we'll have to relearn and say that yes, if we miss a heat cycle, then our breeding program or whatever the timely, we want to ensure artificial insemination gets pushed by many weeks. And there is a calving delay, and eventually the milk production gets affected. And most importantly, accurate identification of estrus is very, very important and pivotal to maintain the reproductive efficiency in case of buffaloes. So considering all these backgrounds, Alfred here, along with Dr. Datta, actually that is where we came on board along with the co-PI sitting here from IIT Roorkee to work on or to develop a sensor-based technology that is not similar. Most people think it is similar to cows and other animals, but not so similar to buffaloes. So I'll quickly take you all the basic of a little bit of basic understanding of how a sensor and how we are progressing in terms of the sensor. And every sensor has a brain, the microcontroller, and the communication is very, very important between the microcontroller and through the gateway to process the data of the behavior of the animal. And at IIT Roorkee, talking about the space we have, that's why, as one of the points that was raised was a lot of infrastructure is necessary, and that is where the Gates Foundation has actually gave us a lot of money to CIRB and to IDROG, as well as to University of Adelaide, Dr. Khatkar earlier said, which actually is very, very useful to process the humongous amount of data that you're going to generate from the behavior of the animal to classify their behavioral patterns and eventually to help us detect stress early. So this is a 3D model. You use computer-aided design to have the CAD device 3D printed circuit board with the different parts that I just mentioned earlier. And this is the final sensor. The final sensor that will go onto the, you kind of make it very small, compact, so that it can, the animal can wear it. Now, one of the major problems with all the sensors is how far you can transfer, because one of the speakers said about 13 kilometers. SIM enabled, but for us it is long range. The LO of the long and RA of the range gives you the LoRa, where we are able to transmit the data up to 500 meters, as Dr. Datta pointed out, because he was part of the architects of this project. So based on that, along with Dr. Ballara, who is the PI of the project, finally we came about saying, like, okay, we will limit our transfer data to 500 meters, where it can actually help a small buffalo holder as well as a relatively big farm. So, and also someone asked a question. We initially started off by placing the sensors on two different places. One by the ear, and the second one was by the tail, because we don't want to disturb the normal physiological activity of the animal. And we found out that when it is placed next to the ear, it is getting aggravated. So we had it on the tail. This is one of the 6 prototypes that we had to work on to get to the final product, what I'm going to show you in a bit. So finally we finalized on the tail, and these are the two pictures showing the sensors on the tail. So now that we have the sensor, which is about 4 centimeters long, to answer one of the questions of the chairperson, it's a pretty compact that goes on the tail. And this is something that we get out of the sensors. These sensors, or this data here, contains not only genuine behavioral patterns, but also interfering high frequency sound from the environment, as well as the muscle twitch of the tail. So how do you make sense, or how do you delineate the different signals that is coming out from 3 different sources? So this is something that, when it goes through a Butterworth filter, I'm not going to go into the details of the filter, but then once we get the data, you filter, and then something at the, you can see the x-axis is the time, and then the acceleration and the gyroscope, because we have acceleration XYZ axis and the gyroscope fit into the sensor. Once we have this, now we start to get the signal. But if you zoom into a particular small segment of the signal that we get on the top, and you see three different significantly different kind of the oscillations of the amplitude. And you can see on the far right, as the speaker, Dr. Mandal, pointed out, there is a lot of activity. There's a lot of activity going on. Rumination decreases, and there's a lot of animal gets restless, and there's a lot of walking. What we have observed here, and the middle one is absolutely no movement, and the far left is the animal just standing. So you can delineate or differentiate between the 3 different stages, like whether it is walking, standing, or actually pretty active, because we want to use these signals, and not just the ability of the animal to walk in XYZ axis or stand and pivot wherever the animal is standing, but we want to use pulse and temperature, which I'm going to talk in a second, to integrate into the movement of the animals. And this is, as you can see here, just look at the green and the yellow. It is a walking and standing. Now, as the animal walks, Now the standing part, the yellow bit, keeps on decreasing. Eventually, you want to make sense of this as we go along and closer to the estrus. As Dr. Monty pointed out, you got a very, very small window, the 5 hours leading up to it, and then post the time. So you want to pick up those cues to get information to the scientist and to the farmer so that we are ready to do the artificial insemination. So again, zoomed in image is the same thing what I just showed you a little while earlier. 3 different. You can see we can divide it into walking, standing, and no movement. As you zoom in into the little amplitude oscillations of the signals that we got from the sensor. So to improve the diagnostic precision of detecting estrus early, we pointed out the temperature goes up, the speaker earlier mentioned. So along with the temperature, we want to include pulse. So there are 3 different parameters we're going to integrate into the sensor to be able to figure out the small window where we can use it for the betterment of the animal. So eventually, we want to end up at this place where we can start to see the high burst activity. For example, if the animal is not eating, can be the animal is full, or there is no activity, means that probably the animal is sick. But then if the animal is super active and the rumination is less, but it's super active, and the temperature has gone up by a wee bit, then the pulse has gone up. We're going to integrate all this data into the prediction model, and eventually the prediction model where we can detect the estrus early. And then how do we detect? And Dr. Monty has pointed out, we need the front end, where the person sitting in IIT Roorkee, along with the folks in CIRB Hisar, can actually tell which animal is in the heat or is coming to heat. Puneet, the guy sitting there, he's again the guy who has done this tail tally. Why it is tallying, we'll get to that in a second. But this is the dashboard. The people probably in University of Adelaide, people in CIRB, probably Albert sitting in Seattle. We'll be able to look at the animals in Somalia, or it is in India. He will be able to do it. He'll be the person who will be able to look at all the animals from here. But the next one, the figure on the right is the individual farmer. He will be able to check on his phone. And this is what we have developed using Android as of now. iOS needs a lot of security clearances, so we have not gone about that. This is now freely downloadable using Play Store perfectly. But once the sensors are ready, we're going to CIRB on 18th to install our first batch of sensors, the 5, and probably by 15th of December we'll have sensors on 25 animals. That is the first for the tail, and then 25 for the rumen bolus. So this is our journey. To conclude, this is our journey. As we go from the left and right, we came on board, and since yesterday we have been talking about a lot of traditional methods of using different techniques, but then it's like an old wine in a new bottle. We need to use the modern technology, and absolutely it has to be a collaborative effort between the engineers, as Dr. Mandel is a mechanical engineer, who have known mechanical engineers, electrical engineers, electronics, and the scientists and the veterinarians to come together under one roof and then solve a problem that has been a perennial problem, to be honest, that has been there. And now that we have funding, a lot of money, we should be able to get through and reach the final digital dairy ecosystem, where we should be able to do everything from a click of a button. Now this is the team, and as I say, the tail will flick, we will get the data, and it'll live to tell a tale. So thank you very much. And this is the team. Puneet is here. Sorry, he's Dr. Puneet Kumar, not just Puneet. And Pranita is my spouse. She's also an immunologist. She's a person working on the biosensors. Professor Pradhan, he's an electronics guy, and Arpit is the graduate student who is again, he's partly probably a wizard on electronics along with Puneet. So any questions on electronics, they are the guys to answer. Anything with the biology, yes, I can answer. And thank you very much.
Thank you very much for precisely finishing the on time and a very informative lecture. Can you, any questions from the audience? Any queries?
Just a query. So, as for these three terms, whatever we observe, so are we developing any best practices for the data collection? Suppose we are telling tail movement. So tail movement is due to physiological or is it to a fly sitting on there, or ticks or mites. So what is the, before using those technologies, so whether you are developing any best practices to collect this data for sensor evaluation or whatever it may be, or this is using this technology?
The question is like, what are the best practices to use this? So what I'm trying to tell you is like, there's actually a live video of the animal. We have video cameras all across the farm. So at the end of the day, whatever data we get, we'll have to correlate with the video, whether it's a swatting of a fly by the tail or it's actually a real movement of the tail. So when these guys are there in CIRB, because there will be absolutely a crosstalk between IIT Roorkee and CIRB, they should be able to actually physically go and check at some point. It's pointed out. It is not at all possible to actually tell a swatting of a fly to a normal switch of the switch moving, but then this is probably, I wouldn't say the trivial thing, but then it's a worth considering.
Not exactly about your adoption of technology or generating your data, also adoption of the technology in the field, that is much important. So how to go ahead, that you should think also. All the researchers who are working in this field, they should think this aspect.
Yesterday, Dr.
All
Mahanti,
those things, whatever I told, observe on these all these talks. So we have to develop a guideline to support this novel technology, using of sensor technologies and others, how it can be better implemented for field as well as improving animal. Basic objective is the animal health, production and welfare. So these so we have to develop guidelines, also you have to develop best practices. Otherwise, working and doing research on aspect, developing guideline and best practices, that should be our, and again, how to share your co-worker, others people are working with you and other groups you are working. So how are you communicating? So you have some definition, they have some definition, they have some definition. So these are varied. As I observed, this is varied. Tail clicking is something different for you, something. So clearly you have to define what should be the definition for this tail clicking. Dr. Mahanti is there. So developing a guideline, it should be better. So interconnectiveness, and we have to discuss amongst ourselves how to develop this field of research.
he is one of the external experts on this.
Yes, that's what I'm telling, because all those research is one way and other aspect is application aspect. I don't think it will be able to apply this sensor data at this condition with this type of research data.
Dr. Vikas Divakar, followed by Dr. B.N. Shahi. Dr. Divakar and Dr. Shahi, 8 minutes presentation, please. We don't have any time. Vikas Divakar. Slide deck of Dr. Vikas Divakar.
Kindly for proceeding only the very brief introduction and the very brief materials method, please go to
Dr. Vikas Divakar is
so that
Animal
message is passed
Breeding
on.
Division, NDRI
Please.
Karnal. He is going to tell comparative analysis of artificial intelligence-based prediction algorithms for breeding value estimate in Pura Buffalo. Please continue.
Good afternoon, all of you. Myself, Vikas Devar, PhD scholar, under guidance of Dr. Vikas Vora. Today I am present my PhD work, and that is comparative analysis of artificial intelligence-based prediction algorithms for breeding value estimate in Murrah buffalo. Now, for breeder, the estimation of breeding value is crucial for enhancing genetic gain. So it's typical to estimate the breeding value, predicting using a complete lactation milk records, while partial records were seldomly utilized. So we are thinking we are using machine learning approach for estimation of breeding value, because machine learning having a non-parametric approaches that can be used for advanced data analysis on a large scale of phenotypic data for estimation of breeding value in early lactation stage or early lactation records as compared to the traditional methods. So we are using a few machine learning algorithms like artificial neural network, random forest, and extreme gradient boosting. For that, we are collected 42 years data from 1982 to 2022, and then after we are estimate the 305 days breeding value with the help of Bombed software. After that, we are using that breeding value in a training data set as an input for the estimation or train the machine learning algorithms. So we are split the data into two sets, like first the training data set and secondly stress data set. And the training data set setting up to 70 for 75%, 80% and 90%. After the training data set evaluation, we are considered 80% data set for the further analysis. So we develop machine learning algorithms models for random forest training, random forest, extreme gradient boosting, and artificial neural network. After the evaluation after the optimized hyperparameters of the models, like learning rate of the models or hidden units of that for the artificial neural network. So we are optimized on the basis of correlation coefficient, mean absolute error, coefficient of determination, root mean square error. For this, we are used 1250 models at one time, and out of that, we are choose best five models out of the 1250 models. So that 5 models we are used on our test set for the estimation of breeding value. And this is the structure of data, where the total number of animals are 1679, and in case of multiparity, the total records are 4,379. And we are divided 42 years period into 14 groups, and each group have 3 years. And for the milk yield, we are collected cumulative monthly milk yield for 42 years records. And for the training data set, as I already told, we are use estimated breeding value as an input only in training data set for the train the machine learning algorithm, along with pedigree information, monthly milk yield, parity, season and calving, and period of calving. And after that, I train the machine learning algorithms, and then the best model are used on test data set for the estimation of breeding values of the animals. So these are the hyperparameters for the different machine learning algorithms, like for extreme gradient boosting, that machine run rate ranges from 0.00001 to 1, and the tree depth range is 1 to 9. I optimized that for my data set. And for artificial neural network, we are use the epoch from 150 to 500, and hidden unit is 2 to 8. Now coming to the results of my work, it is that for random forest, as we see here, K305 500 milk yield, R square is 55.3%, and for 3 months milk yield is 44.6%, and with the correlation coefficient of random forest is 75%, and for the 3 months milk yield it is 66%. And in case of extreme gradient boosting, the R square value is increased up to for 305 days milk yield is 64.5%, and for the 3 month milk yield it is 53.1%, and RMSE of 305 days milk yield, it is 38%, and for 3-month milk yield, it is 45. As the correlation coefficient of our extreme grading boosting and algorithms is 80% and 74% for the 3 months milk yield. And for likewise for artificial neural network, the 305 days R square is 40%, and for 3 month milk yield, the R square is 36%. The overall comparison between the algorithms, we can see here that extreme grading boosting performing better than other machine learning algorithms on the basis of R square, RMSE, MAE, and correlation coefficient of the algorithms. And here we can see the total percentage of deviation of breeding value between BLUP and machine learning algorithms. Here we can see here clearly that for 305 days, the percent of deviation ranges from 0.0 to 0.05 to 4.52, and for 3 months milk yield, it ranges from 1.99 to 8%. And this overall results, like correlation coefficient between the predicted and observed breeding value for 305 days is 80%, 75.7%, 63% for the algorithms, and while 3 months milk yield, it is 74%, 66%, and 60%. And we can say that extreme grading boosting was found to be a best for the prediction of early lactation breeding value with the reasonable accuracy. So it's my recommendation is like extreme grading boosting can be serve as a decision support in the breeding strategies in Morra buffalo by predicting breeding value at early stage of lactation. Thank you.
I have one query, Dr. Divakar. This is you have used separately different models. Is
Yes.
it not? I have two queries here. One is, can we have a mixed model, because ANN, XGB, and random forest, can we have a mixed model developed? Because each one of the model might be giving a much more accurate prediction for a certain type of parameter. So can we have that kind of a thing? Because the correlation for a particular model will be could be suitable for a particular phenotypic parameter or some parameter which we are going to predict. So if we have a mixed model, then that might be a little bit more helpful. Is it possible?
So it's little bit difficult because it's packages I'll run in R package. The packages are totally different for each algorithms, and with the hyperparameters also different. So at a time, the tuning of hyperparameter is also important for the machine learning analysis. The most important part, if the machine learning the algorithms is tuned better, then result will be good.
Yeah, that's what I am telling, because it is good that we have used 3 of them separately. So
Yes,
someday there is a scope to develop of our own a model which can be used for training. That's what it is being.
I'm not sure, sir. So
Think about it.
Yes, sir.
Thank you.
Yes.
Someday it has to come, is it not? Because certain random forest will give a better prediction for a certain parameter, XGB will give for a different one. So if you have a combined one or something which we develop, we write a program, then we extract it, then we make our package.
Okay, sir.
Not
We'll
that
try.
difficult.
Yes.
So all the best.
Thank you, sir.
Thank you. Then on behalf of all the chairs, I invite Dr. B.N. Shahi, Professor AGB Bangla. Sir, please. He will be talking on the comparative evaluation of machine learning models for prediction of first lactation 305 milk yield in crossbred cattle.
Thank
Floor
you,
is
sir.
yours, sir. Please.
Dignitaries on the desk and off the desk. My topic is comparative evaluation of machine learning models for prediction of 305 days lactation milk yield. The earlier speaker is having this type of information also, but he predicted the breeding value. But my prediction is based on the lactation milk yield. So for the the total 305 days is still current basis for the genetic evaluation of dairy cattle, and the information is used by the producer to make their management and breeding decisions. The machine learning model prediction allow us to make highly accurate guesses as to the likely outcome of the question based on historical data. So previously, data which was present with us, that data is utilized for the prediction of the models, for the prediction of the different models. The machine learning algorithm is a subset of artificial intelligence, enables software system to improve the propensity of the anticipated outcomes without being space programmed to do so. And in order to forecast the new output value, the machine learning algorithm has the historical data input, the data which we are having previously. So the objective of this study, that is what was done by one of my PG students, to select the most appropriate variable for mapping 305 days first lactation meal yield using feature selections. The number second, to predict the 305 days first lactation meal yield using the variables selected for the feature selection through machine learning models, and to compare the various machine learning models for the best prediction of 305 days lactation yield. So far, the data is recorded from our cattle dairy farm. We have collected the data of 29 years from 567 animals, and the traits were considered was 30 days, 60 days, 90 days milk yield, first lactation peak yield, first calving interval, first service period, dry period, days to attend peak yield, first age at first calving. So these are the parameters which we have taken for the observations. And for in prediction of first lactation 305 days milk yield, the input data preparation in the present study, we use the MLR, RF, XGBoost, Light GVM, and GVR. These are the 5 models which we were used for the prediction of the first lactation 305 days milk yield. And for feature selection, will be applied to the total data set to get the best input to feed into the models. So for the feature selection, one of the most crucial aspect of machine learning is feature selection. In the most real world data set, there are likely to be the numerous features. However, not all the features are required for a particular machine learning algorithm. Using too many unneeded features might lead to the slow of the issue. So feature selection is a technique for limiting the input variable to a model by only using useful data and eliminating the noise. So far we are having the 9 traits or 9 variables, but we eliminate the unuseful data from the unuseful traits for our feature selection models. So the noise is removed and only useful data is collected for our feature selection model. In feature selection, we are using the correlation matrix, and we giving the value of threshold for the correlation is 0.5 for all the traits, and we are using the multiple linear regression that was discussed earlier by one of our previous speaker, that multiple linear regression, random forest model, gradient boosting regression model, extreme gradient boosting package, and light GBM model, that these 5 models were used. So the coefficient of determination and the root mean square value are commonly used to assess the accuracy and performance of the model. So in total, if we go for the material method part or the methodology, the input variable collected from the instruction dairy farm Pantnagar, application of the feature selection for the selected of best input traits for 305 days lactation milk yield, division of data into training and testing data set. For training, we are using the 80% data, and for testing, we are using the 20% data, and apply the development of the different models of MLR, RF, GVR, Light GVM, and XGBoost, and then the testing of development model by using the 20% data set, and qualitative and quantitative analysis of the evaluation of first 305 days lactation milk yield. So, coming to the result, our result is having the 3 sections: selection of best variable trait for the feature selection, prediction of first 305 days milk yield using different machine learning models and comparison of different machine models. So for the selection of best input for the first lactation 305 days milk yield prediction model, we are having the value which is more than 0.5, which is the threshold value for our feature selection. So the 4 traits, 30 days, 60 days, 90 days and first lactation peak yield is having the best correlation, which is having more than 0.5 correlative value. So this is the scatter plot of first lactation 305 days milk yield of MLR for testing and training data set. On x-axis we are having the observed first lactation and y-axis predicted first lactation value. The R-square value is 0.76 for the training data set, and for the testing data set it is 0.56. Sir, for RF model it is 92% accuracy, which is most higher accuracy accurate model for our data set, and for 0.85 likewise GBR, light GBM, XGBoost. So this is our final conclusion, that the RF model is found to be the best model for the prediction of 305 days lactation milk yield on the basis of these RMSE or R-square value, because R-square value is found to higher in case of training and testing data set, and RMSE value is lower in case, both in case of testing and training data set, followed by light GBM, GBR, XGBoost and MLR. So that is the conclusion of my, thank you sir.
Thank you for a very nice presentation. The presentation is open for queries from the audience, please. The chairs, sir. And thank you. And I have a query in this.
Yes.
Okay. So first thing is that exactly by our Organized Secretary sir, VC sir has pointed out exactly that clustering of the data is one factor I have seen in your presentation as well. That should be a matter of really a great concern. And what we sometimes call as noise or outliers, they may not be actually. They also give a lot of insights. So I have seen that you have a lot of noise filtering you have done. That also may not be a good idea, because when we screen a population so there is that variability is a natural variability.
Yes.
So that if we have to incorporate that natural variability, then we should not actually go for too much of elimination. So what I mean to say is the noise filtering should be as minimum as possible.
Yes.
Or you should have a valid reason that animal became sick or ill or something like that. We excluded from the experiment. That should be okay. That's what I feel like. Sure.
Okay.
Thank you.
Okay sir, thank you. Thank you sir.
Thank you very much. So now we have come to the end of the session, technical session 3. We thank the organizers for giving us opportunity to chair the session, and we had 6 lectures, very exciting lectures, and beautifully it has been correlated with the most important hot topics of the day, like artificial intelligence, AI, ML, and all those things, along with the conventional breeding. So it is very seemed. So we appreciate the leadership of ISAGB and the organizing our team of the university for a wonderful session and the careful planning. Thank you.
So, thank you very much, sir, Dr. Mohan, Dr. Rao, Dr. Chaturvedi. I now request Dr. Mohan to hand over the memento to the speakers, Dr. T.K. Mahanti, Dr. Shauman Mandal, Dr. Srinivas Amajipuri, Dr. Vikas Divakar, and Dr. Shahi. Please be ready, one by one. I now request Dr. Mohan to hand over the token of gift and love. सब लोग जाइए एक एक करके दे दीजिए. T.K. Mahanti, sir. Please hand over the memento to Dr. T.K. Mahanti. Dr. Soman Mandal. Dr. Srinivas Kiran Amadipudi. Dr. Nithe Ranjan was in online, so we will send it by courier. Dr. Vikas Divakar, please come. And Dr. B.N. Shahi. I'm now requesting our Honorable Vice Chancellor, Dr. T.K. Datta sir, to come over the dais to give memento to our chairpersons and report here. I now request Dr. T.K. Datta sir, Honorable Vice Chancellor of University, to hand over the memento to Dr. Mohan. Next one, please. To Dr. P.K. Raut and Dr. S.K. Chaturvedi. Dr. P.K. Raut and Dr. S.K. Chaturvedi. And also, First. Dr. S.K. Niranjan, rapporteur of the session. Wait. Dr. Sanjay Kumar. Oh, sorry. Dr. Sanjay Kumar. Sorry, sir. Thank you very much, sir. So, very fast, we are now going to start our technical session 4. ISAGB Young Scientist Presentation Award. So I call upon the dais Dr. Singh, Dean, SGIDT, Basu Patna, Dr. R. Vinu, Registrar, Sri Venkateswaraya Veterinary University, Tirupati, Dr. Pushpendra Kumar, ex-HOD AGB, IVRI, Vijayanagar, and rapporteur Dr. Pandit Nandedkar, Associate Professor, Department of AGB, Barwani. So now we are going to start the technical session 4. This is ICAGB Young.
So Arunachali yak is the first registered Indian yak breed and endemic to the West Kamen and Tawang districts of Arunachal Pradesh. They are reared by the pastoralist community of the Monpa tribe. That is, they are called the Brokpas, and they are considered as the lifeline of these highlanders. However, there still lacks genomic data on this animal, and in order to address this, our team have generated a chromosome level reference genome assembly of this animal using a combination of PacBio hi-fi long-read sequencing, optical mapping, high-C sequencing, and isoform sequencing approaches, which was which was published earlier this year in Scientific Data. So we use this sample of this animal, which is a 6-year-old female from the institutional yak farm, which is located at Lukmadung in Arunachal Pradesh, at an altitude of around 2,550 meters above mid-sea level. And this is a pipeline that we followed for genome assembly as well and for genome annotation. So the initial PacBio sequence read had 1.6 TB of raw subreads, which were processed by CCS and which was used to generate 86.9 GB of hi-fi reads. We also generated 141 GB of high-C data, and optical mapping was also done, in which 99% of the molecules fell into the more than 20 K bin. So the genome assembly consisted of 1,296 contigs with a total length of 3.16 GB, and the largest contig was of 161.96 MB, which after filtering for duplications as well as contaminations, the clean assembly was again so scaffolded on the optical map as well as the high-C sequencing data to achieve a chromosome level assembly. And the final assembled genome consisted of 63 scaffolds with a total 2.8 GB in size, and the N50 was of 104.5 MB in size with largest quantic size of 161.96 MB. So in order to assess the correctness of this assembly, we did a high-C contact map and generated the map that is shown here. So it showed 30 chromosome scale scaffolds, which corresponded to the 30 chromosomes of Arunachali yak. A KMR-based genome survey was also done using genome scope, and the predicted genome size was 2.3 GB with a heterozygosity percentage of 0.21 and duplication percentage of 0.35%, which showed the completeness of the assembly. So repeat content analysis was also done, in which, because we thought it is essential for precise genome annotation, it helps avoid the incorrect classification of these regions as genes and offer a valuable understanding of this genome evolution as well as the possible regulatory functions of these elements. So in order to further validate the quality of the genome assembly, we've performed BUSCO as well as mercury KMR completeness analysis, and in BUSCO we found that 95.7% of the mammalian genomes was present in our assembly, and then in mercury KMR completeness analysis, 95% completeness was shown with a base level accuracy of around 67%, which is within the limit of high quality reference genome assembly as defined by the Vertebrate Genome Project. Gene prediction was done using a combination of both homology-based as well as initial gene prediction method, and in the final list, we included only the complete gene models, and then they were annotated using various functional databases like Gene Ontology, CAKE, and UniProt, and we found that 81.5% of those predicted genes were successfully annotated in at least one of those databases. So we compared our assembly with the already existing two reference genome assemblies of yak, both of them are from China. So, and we found that ours has better contiguity because only 63 scaffolds were used in our final assembly, compared to 343 and 1,313 in the other assembly. So the chromosome representation was also correct since we have 30 chromosomes, and genome size is 2.85 GB, which is slightly larger than the other references, suggesting a capture of novel or previously unassembled genomic regions. In terms of scaffold metrics, although the scaffold size N50 is 104.5 MB, which is a little bit smaller, like marginally lower than the one of the assemblies, but still it is comparable, and combined with the low scaffold number that we have in our assembly and the chromosome representation, our assembly defines a very high structural accuracy. So to complement this resource, we also generated a transcriptome atlas of interest, atlas of Arunachali yak. We did isoform sequencing of 22 yak tissues from different representing major physiological systems, and final data set of around 2 lakhs non-redundant transcripts were identified, and then they were used for further studies. So functional annotation was done based on a multi-database alignment method, and we used NCBI non-redundant database as well as nucleotide, UniProt, geo-annotation, PFAM, KO, and then COG annotation, functional annotation was done. So using COG and functional annotation, we annotated 34,771 transcripts, which mostly fell into the unknown category, suggesting a large number of the genes is still uncharacterized. And for in CAKE annotation analysis, 27,120 transcripts were mapped to 417 CAKE pathways distributed across 5 major functional categories. And using gene ontology enrichment, it was around 31,000 transcripts were categorized into different levels of biological processes, molecular function, and cellular component. So since there's increasingly interest towards long non-coding RNA as they are one of the regulators for gene expression, especially during the stress condition or environmental stress, we predicted the long non-coding RNA using 3 data domain filtering domains, that is FLEX, CPC2, and PFAM, and identified 4,319, which were shown as non-coding in all the 3 data domains. So we also predicted the operating frame to identify the protein coding sequences, and then to identify potential markers for future studies or downstream annotation. So we also identified simple sequence repeats in this in our genome, in our transcripts, those non-redundant transcripts that we had identified. So mononucleotides were the most abundant in this. So in conclusion, I would like to say that this reference genome as well as the transcriptome atlas is a critical genetic resource for elucidating and understanding the genomic architecture of this very, very unique animal, which is adapted in a very high altitude where no other animals are there. These resources are key to understanding the genetic traits like linked to high altitude, adaptation, disease resistance and productivity, and can be further used to facilitate further research, empower targeted breeding, sustainable management of yak populations, and conservation of these species. Overall, this will help ensure the long-term sustainability, productivity, and resilience of yak farming in India, particularly in the face of climate change. Thank you so much. That's all. Thank you. Thank you, sir.
Topic is open for discussion. Questions from the audience, please. You may ask one or two questions.
If there are no more question, I can ask two. Dr. Martina, long non-coding RNA you have identified.
Yes, sir.
Could you associate these long non-coding RNA with some trait also? Trait or function?
Sir, for now.
Number one. Number two, you have done all this genomics and transcriptomics, but as far as I understand, there are no breeding improvement program or genetic improvement program going on in yak because the population is so small and it is reducing as well. So first and foremost thing is to frame out some genetic improvement program. Genomics come later. First, you should proceed with the genetics. What's your opinion?
Thank you, Saxena, sir, for the beautiful questions. Answering to the first question regarding the long non-coding RNAs, for now we haven't started any annotation or analysis on that part yet. I'm just these data are still new to me. I've just received them and then just recently published. So further studies on them will always be I'm planning on that in the future. And regarding the second question, that is breeding, means that there is no breeding plan or something. So we have national tax task force on national breeding program for yak that we have conducted 2 meetings already. So hopefully it will come up in the future. And then, sir, not only the genetics, the most important what we feel the most important reason, because the reason for the decline in yak population right now is the hardships that the yak farmers are facing. Because I'm sure not all of the people know, the system of rearing yak is really different from how the other animals are reared. They are reared under transhuman system. It means that the animals are migrate. They come to lower lying lands during the winter months, and then in summer months they go to high altitude beyond 3,000 meters above mid-sea level. And that transition and then those movement, it happens not only the animal they travel, but they travel along with the herders, and that's one of the most important thing. Why the younger generations, they don't want to rear yak and they're running away from this profession, and keeping them in the profession is according to me personally, is what should be the main focus right now. And regarding conservation, sir, you are right. We are as an institute, as Sarani and yak, we are implementing many of the ARTs, or we are trying to propagate the selective breeding is being done in our farm, and we are propagating those elite germplasm. Sir? Okay, so we're distributing elite germplasm through for cryopreserved in a way through cryopreserved semen, and then distributed to the farmers also. So that's the activities that's ongoing right now. So I think we're working on that area also.
I have two small questions.
Yes, sir.
Whether any yak whole genome sequence is available in the public domain? If it is not available, which scaffold do you use for annotating the sequence?
Also,
QSR analysis, could you find any tetrad sequences could you detect or not?
Sir, answering to your thank you, sir. Then answering to your first question, sir, says I have already shown there is a whole genome sequence available in the database, but that is of our Chinese yak. Both of them are of Chinese yak, and none of Indian yak is available. So this is the first one.
Sufficient is if it is available. Right?
So this is the first one, and as I have shown the comparative table, it's of a very high quality. And then, sir, regarding the SSRs, sir, I have answered earlier. No downstream analysis has been done yet on all this data yet, sir. The transcriptome data is still unpublished. The isoform sequencing data is still unpublished.
But you could not compare tetrad or trinuclear dinucleotide sequence.
Not yet our work on that side.
Thank you.
Thank you.
Martina, thank you.
Thank you so much, sir. Thank you.
Now next presenter, Dr. Madhuri. Dr. Madhuri, you have 8 minutes. Okay, please go ahead.
A very warm good afternoon to one and all. I am B. J. Madhuri. I have completed my PhD from Madras Veterinary College. I feel it a privilege to present my thesis topic, transcriptomic profiling of spermatozoa in indigenous Tamil Nadu goat breeds to reveal the key genes influencing male fertility. Coming to a lot of schemes under the government, we have the NLM scheme. We can say that goat farming has now become a profitable enterprise. So we have a huge local population, and also we have 41 registered indigenous goat breeds, but yet we lack in productivity and reproductivity efficiency. So the government of Tamil Nadu has come up with an initiative to bring the genetic upgradation of the local populations using the semen of indigenous goat breeds through AI. So we all know that AI actually necessitates the selection of a superior male. So superior males can be selected based on both phenotypic and molecular level. Coming to the phenotypic level, we have the breeding size examination and the seminal attributes, whereas the molecular level, we have the DNA polymorphisms and gene expressions. Lot of work has been carried out in DNA polymorphisms, and few literature has been seen in gene expression in case of goats. So we come up with the objectives for this study to evaluate the semen characteristics of indigenous bucks of Tamil Nadu using CASA, and the second objective was to identify the candidate genes influencing vital seminal attributes using RNA-seq, and the third one was to validate the expression of selected candidate genes. Coming to the brief technical program, we collected semen samples from 3 indigenous breeds, Kanni, Kodia and Salem Black, and then we have analyzed both conventional and CASA parameters. Then we classified our bucks into high quality and low quality semen groups using PCA. Then we followed the RNA isolation, which was the toughest part of my PhD journey. And then we used the basic parameters like FastQC, Trimomatic, and we aligned with SANET because it was having the wide genome reference also. Then we did the differential gene analysis, proceeded with the gene ontology. And then we validated using qRT-PCR some of the few genes which we selected from our study. Coming to the semen traits, the conventional were volume, concentration and motility parameters, and CASA has given us a lot variety of 12 parameters for assessing the fertility. And then we did also quality traits and flow cytometry parameters like mitochondrial membrane potential, etc. So coming to the results, Kodiadu was having low volume, but all the other parameters of Kodiadu were high, followed by Kanni and Salem Black. Then the percent of sperms, the progressive sperm is the actual one which will be fertilizing the egg. So these were more in case of Kodiadu, followed by Kanni and Salem Black in all the neat, pre-freeze and post-semen parameters. Then coming to the next part, the progressive sperm, the CASA parameters, we have ALH and VCF, which the ALH, if it is high, then the sperm will be low fertile. So the low ALH was found in Kodiadu breed, and then the linearity and straightness on all the motility parameters were high in progressive sperm in case of Kodiadu, followed by Kanni and Salem Black. And even the host parameter, acrosome integrity and viable sperm, all we found out that Kodiadu was superior. So once we have done the cluster analysis and PCA plot, all the Salem Blacks were clustered into one category, which we call it as low quality semen, and the Kanni and Kodi were falling into one category on the cluster analysis, which we call it as high quality semen. Then we went for the RNA isolation. So we used intron spanning primer to check the purity of RNA so that there is no DNA contamination. And to ensure that it is only from the sperm, we did use some other primers to check whether any other cell contamination is there or not. So this is the image of our gel picture which reveals that it was only from the sperm RNA. Then we checked the RNA integrity, and which was 2.5. Since the sperm is very degraded, the sperm RNA is very degraded, the integrity will be between 2 and 3. Then coming to the fast QC results, we found there are we found the quality is very good, and we got 65 million raw hits, which is a very good number for global gene expression data. And then coming to the mapping statistics, we got around 97%, which was very good mapping rate, and a total number of 19,257 transcripts were found, and after quality filtering, we found 18,871 transcripts, of which 1574 were differentially expressed, and we took a small log fold change value to further analysis. So there were 1083 genes which were upregulated and 350 were downregulated, and these upregulated genes were having function in mitochondria and negative regulation of BMP signaling pathway, which is very important for the fertility of the male. And then we took Cobas and David for KEGG pathways, and we have found out those involving in calcium signaling pathway, NARC signaling pathway, and TGF beta signaling pathway. So this calcium signaling pathway, the genes which are involved are very much for sperm motility, capacitation and acrosome reaction. And even the NOT signaling pathway, the disruption will be leading to decreased sperm motility and fertility. And then there are other pathways which are also useful in the cilia movement and flagellar movement of the sperm, which will be used for good fertility in the males. Then coming to we used cytohubba for finding the hub genes. We found some ribosomal protein genes and MSDR1 genes, and those downregulated we find CCP genes, and there are some genes which we BRWD1 which are very important for the sperm. Then we took 4 genes, INHBC, TARC3, BRDW1 and RBPP6 for our qPCR validation, out of which 3 were actually giving the same results like the RNA sequencing, but only the TACR3 was not having the proper validation. So coming to the conclusions of my study, I actually feel CASA parameters were very important in classifying the animals based on low quality and high quality semen, and our sperm RNA sequencing has unveiled numerous transcripts which are essential in spermatogenesis, sperm capacitation and fertilization, and we have got numerous pathways that are significantly involved in sperm quality and male fertility. And these few genes which we used could be used as biomarkers for selecting the bucks at a very young age. So coming to the future directions of our study, we have had lot number of genes, like almost 54 genes that were differentially regulated. So we could use them for validation and develop some kind of chip, and then we can use for genomic selection. So we could progress our study, and we also went for rRNA depletion approach. So we could also undermine the long non-coding RNAs and miRNAs involved in the regulation of mRNAs. So, and the next step we are planning is, since we are a part of the Tamil Nadu government scheme where we are providing the AI straws, we also want to correlate with the field fertility rate and give a conclusion, best conclusion to our study. And these are my references. Thank you.
Nice presentation. Now topic is open for discussion. Co-chair, 1 or 2 questions from audience. Dr. Rao sir.
Thank you very much for your nice presentation.
Thank you, sir.
I have 2-3 questions. First question, whether this RNA sequencing you did outsourcing or you did in your lab?
It was done in GBRC, sir. I personally went and did in Gujarat Biotechnology Research Center.
Second thing, this showed in one of your slides, there was an STR correlation with the sperm motility. How STR correlation with the sperm motility?
STR, sir.
You showed short tandem repeats. Did you do that?
No, sir. Short tandem repeats I didn't use.
Okay. Did you go for any methylation difference with the sperms?
No, sir. We only went for transcriptome sequencing only, sir.
Thank you.
Jaya.
Sir.
I'm sorry, I did CASA, must be about 20 years back.
Sir.
Don't have lost the luxury of doing on-bench science. So
CASA, there are two parameters. One is BAP,
VAP, average path velocity.
right,
Yes.
and another is ALH.
Amplitude lateral head displacement.
So do you find a correlation among them? I mean, I still remember this gives me a feeling that they are redundant parameters. They are all the same.
Yes, sir.
So do you also feel so?
Yes, sir. But most of the studies in court, they tell ALH is the very good indicator of sperm.
No, I'm not talking. I'm asking Jaya.
Sir.
I mean, Jaya, what do you say?
Sir, what I felt is when I did analysis based on the data, I felt ALH and PCF along with straightness and VSL, straight line velocity, were more important in predicting the high quality and low quality sample.
When a sperm is running and it is running straight,
yes, sir.
It is not a good thing.
Yes, sir.
It should have a VCL.
Yes, sir. Curvilinear velocity.
Curvilinear
yes,
velocity. So that's a desirable phenomenon.
Yes, sir.
So when you have classified the two batches,
yes,
one
sir.
is good, one is bad.
Yes, sir.
So you did it based on which CASA parameter or all
Like
CASA
I took all the parameters, especially the progressive sperm in post or semen, sir.
You should not have taken all. You should be choosing to see that this is the one
yes,
that differentiates between a good and a bad. And then you do all your sequencing and this networks. Thank you.
Okay.
Nice presentation.
Thank you, sir.
Thank you, Dr. Madhura.
Thank you, sir.
Now, next speaker is Dr. Mumtaz. Please come. Dr. Mumtaz. Dr. Mumtaz is not here. Now, moving to next speaker, Ms. Swati Jaiswal. Please come. Topic is already displayed and you have 8 minutes. You have to finish your talk within 8 minutes. Go ahead.
Good afternoon, esteemed panel members and everyone. My name is Swati Jaiswal. I am to pursue master's degree in animal biotechnology from the College Garbasu, Ludhiana. I am here to present a master's thesis research work, and the title is AI-driven cytological diagnosis of canine round cell tumors using YOLO V8 and deep learning architectures for enhanced veterinary and health management. Okay, AI, artificial intelligence. Everyone knows about it. I mean, who doesn't know about self-driving cars? So we can say AI is a broader or a middle term underneath ML and DL comes. Our concern part is for the DL, deep learning, which is some type of machine learning as well as an AI. We're inspired by the human brain structure and its function. So when I said human brain structure and its function, that means our human body is designed to process complex information. And who does that? Nervous system, right? So one unit cell is known as nerve cell, and in DL, one unit cell is known as nodes, and previously it was called as perceptron. Now these nodes are arranged in layers to process the complex information. And these nodes are very important for the model training. Now coming towards the slides. How do I do? Round cell tumors are the most common cutaneous tumors in canines, and microscopic diagnosis depends on the expertise, knowledge, as well as it's time consuming. So we came with the aim to integrate artificial intelligence and computer vision for accurate and diagnostic tool. Knowledge gap in the veterinary field, histopathology work is very common, but cytology work is very limited. And there is no such kind of widely adopted standardized deep learning pipeline that combines detection and classification of canine tumor cytology. Hypothesis: cytology images of RCT have identifiable patterns that can combine with the urine detection, and with serum classification can improve diagnostic accuracy in the veterinary cytology. So we came with the objectives like to design and develop a computer vision powered microscope image analysis approach for the detection of tumors, and to validate the trained model with real experimental data. This is the pipeline and the methodology of my research work. The simplest workflow, I'm using the images. So the input will be the images. I will collect the images, annotation, and then data augmentation if it requires. Then we can train the YOLO model. And after training the YOLO model, we will prop those tumor images and we will send to the classifier for the classification system. If you want to see how the model behaves on the images or the validation or test images, you can see through the breadcrumb visualization, and last, you can evaluate your model. These are tools and computational resources I use for the model training. Data collection. I receive my data from the two sources, Department of Veterinary Pathology, Garbasu, and online sources as well, peer-reviewed research articles. And all the images have been seen and verified by Dr. Kuldeep Gupta, HOD of Department of Veterinary Pathology, Garbasu. Now, I trained my model in 2 phases. Phase 1, object detection. Phase 2, tumor classification. Now, dataset annotation. I annotated my dataset using robopholographical interface, and each tumor was manually labeled by me with bounding boxes. And after manual label, after the annotation, the dataset was exported using the proper compatible format like YOLO format or YAML format. Now, coming towards the YOLO model training, for the model training, we will split our dataset into training, testing, and validation. And here we can see class-wise imbalance. And to treat the class-wise imbalance, we will apply the restrict class-wise data augmentation and transformation have been applied. And after applying all the transformation, we will finally move towards the YOLO model training by setting up all the parameters like image size, hyperparameters, optimizer, and everything. Now move towards the serum classification. Dataset preparation and the processing are the 2 most important things for the model training. Like the enhancements we have made, the expansion, padding, and the resizing and everything, still we can see that imbalance in between the 2 classes. So to deal with that, we require the class balancing using augmentation. Okay, I use the Convenex tiny model, which is a hybrid latest CNN model. It works on transformer model. So it has strong feature extraction for cytology patterns, and remember the stage 1, 2, 3, 4, these are the neural networks I was talking about, which helps for the extraction of features. And this model haven't been used in the veterinary field yet. This is the final data set. Still we can see the imbalance in between the data sets. So we can compute with the compute class weight at the time of model training. Now we will set up all the parameters, we'll train the model and come with the results and discussion. This is the performance metrics, and here MMP, the accuracy is like 92.80, which is quite good because it helps in the strong precision of around all across 4 tumors. Performance metrics or the confusion metrics shows diagonal value shows the correct prediction, and the rest of the values are the wrong prediction, and the minute misclassification can be seen due to the overlapping psychological features and also the class imbalance. Now this is how it looks. It looks on the single image, and see how beautifully detected the tumors with high confidence scores. And comparison between the RCT and RCT can be done with the high detection. This is the validation accuracy I received in the serum classification 96.37 with minimum loss. Classification report with minimum minute misclassification. ROC curve, AOC value is close to 1, that means our model has excellent sensitivity and the specificity. If you want to see how your model behaves, you can use breadcrumb visualization. Okay, I have compared with different models also, and our model outperformed from every other models. Okay, this is the very important part because based on this, our model's accuracy depends. Why? Because total 276 images I took, and these are not included in our training data set or the model trainings. So out of 276, we got the prediction of 265 images, and the accuracy is about 96.01, which says strong generalization on unseen clinical data. This is how it looks like. As I said, the model shows high precision, robustness, and minimum misclassification. Conclusion, integration of YOLO V8 and CNN pipeline give reliable results on unseen real-world data. Societal benefits, we can assess pathologies in routine screening, useful for diagnosis in remote areas, as well as it can reduce the human error-prone microscopy. Future perspective, we can add more diverse datasets with better annotation to increase the efficiency of the model and to deploy as a web-based diagnostic tool. I would like to give my acknowledgement to my team and my advisor, Dr. C.S. Mukhopadhyay, who had been a constant pillar throughout my journey. Okay, at last I would like to say, we support, we don't replace the veterinarians or pathologists. We are just over here to aid them as a third helping hand for reliable, accurate, and robustness and vet diagnosis detecting tool. Thank you.
Thank you, Swati, for a nice presentation. You have already finished your talk within 8 minutes. Before that, I congratulate. Now topic is open for discussion. Our colleagues and audience, 1 and 2 questions you may ask. Audience? Yes.
I think it's nice presentation. And what about how do you compare your this type of study with the human?
Humans. Actually, the work has been done in the humans and histopathology work, but the cytology work is so rare. That's why this work, the pipeline I made is basically for the cytology veterinary. We can include in the human as well.
Has this study has been implemented anywhere in India or in the world? Yeah, here.
Madam, did you apply any statistical model also? I find some anomalies in your analysis.
What's that?
Because such type of work should have false positive, false negative, such type of categorization, and have some specific model. Yesterday we had a presentation on modeling itself. There appears to be no model applied in your work. So this is a critical, observation when you are doing any such kind of work. Model is primary thing. You have to fix what model you will apply, and accordingly, statistical analysis is done. So there is no statistical analysis. There is no model I see. So these are some gross anomaly I observed in your presentation.
Sir, as I said, the work has been done before on the histopathology slides, but cytology work is rare, and I had used 5 different models. The the Convenex model which I have used is the latest model which has not been used in the veterinary field. And if you are talking about the statistical analysis, the confusion matrix what we get after taking the model, ROC curve, and the different graphs we showed, that shows the statistical analysis of our model. I tested my model with the unseen data set also, like 276 images have not been
You
used.
presented your data in percentages, 93% correctly identified, or like that. Percent and statistical analysis are different.
Yes, sir.
She is not understanding it.
Sir, this is Vikas Vora, sir. To answer the Dr. Saxena's question, sir, AI modeling is totally based on non-parametric, non-linear. So that classical statistic what we are looking for is not applicable in this case. Whatever she has presented as a confusion matrix in percentage is okay.
Just to support her.
Thank you. Yes, sir. The statistical analysis in the way it was done. So confusion matrix was developed. I believe if it can be displayed again, Swati, can you please go back to that? Okay, sir. So yeah, it was done.
No, that is not required actually. No need.
Yes, sir. Yes, sir. Actually, here in these cases, we compare using some confusion matrix. Specific accuracy we check, and of course, the parameters have been checked over there.
Is it not? It's not statistics. Yeah. So anything to say in defense? If not, it is fine.
That's it. Accuracy in terms of percentage. The accuracy has been given in terms of
accuracy in terms of percentage.
Yes.
He says accuracy.
Precision and recall was calculated using total true values divided by total number of claims.
Thank you very much.
Okay. Thank you, sir.
Thank you very much. Thank you. Now next speaker is Dr. Marine Amdani. Now you will be the last speaker. Your topic is AI. She will be talking on this AI-based genetic merit prediction in seed.
May I start, sir? Okay. Honorable members of the jury, respected scientists, a very good afternoon to you all. Kolkata eche khub bhalo lagche. So beginning with that, I'm Dr. Amreen Hamdani from the Center for Artificial Intelligence and Machine Learning, Kashmir. So, as we all know, an animal breeder's primary responsibility is accurate identification of superior animals for the scientific selection of animals, which in turn is made possible by the estimation of something called the breeding values, right? So what are these breeding values and why do we do it? We expect every generation of animal should be better than the previous generation, and that is what genetic progress is all about. And this has worked very well so far, standing on the shoulders of giants like Robert Bakewell, Darwin, Fisher, and so many others. And these computationals have largely been data-driven. However, the world is changing today. We are seeing intensification of animal farming, and with the result of that, a lot of data is being generated. We are living in the world of big data. And this data cannot be handled by conventional strategies. Conventional strategies cannot manage this data, and as a result, what happens is a lot of the times, breeding value is not estimated at all on smaller farms. So what is the way ahead? The way ahead is to become a part of the technological revolution. We must also be a part of Industry 4.0. And the logical next step is to integrate artificial intelligence in animal breeding. And as you would all agree with me, that animal genetics and breeding is already a data-intensive science. All we need is one push into the realm of artificial intelligence. So considering all this, we use 12 artificial intelligence algorithms for the prediction of genetic merit as well as breeding body weights in sheep. And my research has been cited by a number of, I was very happy to know that it has been cited by a lot of scientists, new scholars from NDRI as well. And evolution, now then we evaluated the algorithms and we ranked them. So very quickly going over the materials and methods, we had a 52-year-old data from our university farm on the corridial breed, and we appropriately pre-processed our data because it's a real-world data, so it had a lot of MARs or missing at random data as well. And then we went with dimensionality reduction in order to mitigate the curse of dimensionality challenge. And then obviously we use the conventional scoring criteria as well. The data split was heuristically determined, and it was found that this one worked best for us. And finally, we went on to try 12 different artificial intelligence algorithms for this problem at hand. And our experimental findings indicate that imputation worked very good for our MAR data. And then vinsorization helped us get rid of the outliers as well as normalize our data to a very large extent. Then, of course, as I told you, the PCA helped us in finding the variance ratios for important features, and 6 features were extracted for that. And then, like I told you, we made 2 data sets. One was for the principal component analysis, and the other was simple feature selection. So this is for the principal component regression for breeding values. The correlation coefficient was somewhere around 74%. For the ordinary least squares, the R squared was about 50, but not extremely good. And then we have the Bayesian ridge regression. For the Bayesian ridge regression, it was 74%. And we tested a lot of artificial neural networks because we were expecting them to outperform everything else, which you will see it did not work that way. And what we saw was that out of the 3 models that we tested, the third one, wherein we had an iteration of about 2,000, gave us a correlation of about 73%, much less than what we had expected actually. Then we also trained about 100 models for breeding value estimation using artificial neural networks, and here the one with the swish activation layer gave us the best results. And this is the hyperparameter optimization curve. As you can see, after 300 iterations, we could barely see any improvement in the model. And so no matter how many more iterations we did, there was hardly any change in our results. And this is again, as you can see, after 500, no change at all. And these are the graphs for the declining mean squared error, for the declining mean absolute error, and the correlation between predicted and actual values. For the support vector regression, again, the correlation was somewhere around 77%. For the regression trees, the tree-based algorithms outperformed all other algorithms. Here, as you can see, the correlation is about 91%. In fact, for the body weight prediction, it went as high as 99%. This is the gradient boosting algorithm. Again, the XGBoost did a great job. Polynomial regression did a great job. The gradient boosting regression had a correlation of about 91%. Then we move on to the k-nearest neighbor. K-nearest neighbors is a very simple algorithm. So as we had expected, it did not outperform the tree-based algorithms. The Mars was also good enough, but not but not extremely good, as you can say. And this is the final algorithm ranking. As you can see, random forest outperformed all other algorithms, but all the tree-based algorithms, the gradient-based algorithm, the XGBoost, the classification trees, all of them have done a great job in prediction, where the correlation coefficient is almost, for all of them, it is above 90. And the conclusion for the same is that we successfully developed 12 artificial intelligence models. So why is this important in the real world? Let's say we create an interface. Now the farmer can only put in his data and get the results. He doesn't need to know what is happening at the back end. So that is why our models are reusable, they are deployable, and they can, to a large extent, reduce our dependence on high computation that is required when we perform, tasks like BLUP, the best linear unbiased prediction, which is very common for value estimation. So here is the ranking of the top 3 algorithms: the random forest, closely followed by gradient boosting and XGBoost. And we could safely say that artificial intelligence is not just the next big thing in the world, but it has the potential to transform our beloved animal genetics and breeding sciences as well. So this study stands published in scientific reports. And that would be all. It has been a pleasure coming to Kolkata, and I'll be happy to take questions.
Thank you, Dr. Amlani, for a nice presentation. Now, topic is open for discussion. 1 and 2 queries. First, you have already evaluated, already given the breeding value.
Yes.
How it is different if you are using the BLUP, and here you have already used AI.
Yes, that's a very good
Can you compare
question.
it? Which one is the best?
Yes, sir. See, we wouldn't say that any algorithm is better or the other way around. What artificial intelligence does for us is that once we have evaluated a model and we have deployed it, now we don't need any computation. For example, in BLUP, if you have data, you put your data in BLUP, and then there are a lot of processes involved. For example, you make the matrix, then you invert that matrix, and then you perform a lot of other matrix multiplication, etc. calculations. And that is a huge burden on the resources. An ordinary computer would not do it for a lot of animals, right? So here what you do, you train the model on a very expensive, a good computer, and you deploy the model. After that, it will run on your mobile phone. That's as simple as, that's how simple artificial intelligence can make animal breeding today. And that is the power we have today, and we must harness it. The algorithms the algorithms that we will always have will always depend on the kind of data that is available to us. If the data is not good, the algorithms will never be good. So we must work towards improving the data.
Question from audience. But my personal view is very few students, rather young scientists, they are coming in the field of animal genetics and breeding particularly.
Yes, sir, that's
Everybody
a
is
concern.
working in the genetics, particularly molecular genetics. Nowadays, breeders are also required for this country for the genetic improvement of the livestock. I urge you all, the young scientists, you have to put your energy in the animal breeding population genetics also. After some times, it will be very difficult to find out the good breeder in the India.
Sir, can I add a little line to this, sir?
No.
To what sir said, I want to comment.
No need to reply.
No
No
problem,
need to
sir.
reply. That is my.
No problem, sir. Okay, sir.
Any question?
No, not at all, sir. I wouldn't even dream of it, sir.
No need to reply. Any question? Yes. Question answer.
Good work you have done.
Thank you, sir.
So, if we give you the data of 100 cows,
Yes, sir.
and it's blind data, you don't know what is what is the ranking of those cows or bulls, okay, rather. So, will you provide us the correct ranking of the breeding value applying your model? Have you ever tried your model on blind data?
Sir,
Hiding their breeding value and testing it.
Sir, what do we mean by blind data? Like, we don't know which animal is which?
You should not know what is the ranking of those bulls or the breeding values of those bulls, as Dr. Umesh Singh told, club breeding value or progeny tested breeding value, whatever kind of GAV is available for those bulls, we hide them.
Yes, sir.
Don't give you. We give you the data, whatever parameter you require.
Absolutely.
Then you give us the ranking.
Yes,
so
absolutely.
how far your model correctly rank those points?
That's a very good question, sir. So, sir, what will happen if our model is trained once, then it will forever work on your blind data. For example, my model is being trained on sheep, so it will work on blind data from sheep. That is the testing data set that we basically use. When we test a model, we don't tell the model what the actual breeding values are. So that will always be blind. But if you put a cow data set into a sheep model, then it will not work.
Okay, Dr. Andani. Which breed you have taken? Only
Corridel, sir. Corridel, yes, sir. Because the data was more or less intact for this breed.
You have not mentioned in that.
I have, sir, in the material methods.
Okay, thank you for.
Thank you, sir. Thank you very much.
Presentation. Now, we had 4 presentations from young scientists. At the end, they have already covered so many things. They have already, some of the speakers, they have already worked on sheep, goat, and yak. I think it was a very fruitful discussion, and we have heard the young scientists, they are future of the country. At the end, we are very much grateful and thankful to the Honorable Vice Chancellor, West Bengal University of Animal and Fishery Sciences, sir. And also we are very much thankful to the organizers for giving us the chance, opportunity to act and chair the sessions. We are very much thankful to our colleagues. I thank Dr. Venu, Dr. Pushpender Kumar, Dr. Rao sir and Dr. Pandit for making this session very successful. I thank all the delegates and all participants, all delegates. They have already participated actively in the session and asked so many questions from our young scientists. Thank you all. Thank you very much.
Thank you, sir. I now request Dr. V.K. Saxena sir to come over and advise. He is the Secretary of ISAGB. I request him to hand over the memento, our gift of respect and love to the chairperson, judges, as also the report here. V.K. Saxena sir. I just request Dr. Saxena to hand over the memento to Sir Dr. Umesh Singh, Dr. Pushpender Kumar. Pushpender not here. Yes, sir. आ जाइए ना, please. One by one. And also, sir, V.K. Rao sir. क्यों? V.K. Rao sir. No, he's here. आप इधर ही हैं सर, चेयर बैठा नहीं है इसलिए। चेयर वाला दे दीजिए सर। Sir, thank you very much, sir. Thank you very much, sir. Now we are going to start roundtable meet for stakeholders on industry academician interface. The last part of the session now. I now request Dr. Partha Sen. Dr. Partha Sengupta, coordinator of this session. Please wait here. Dr. Alfred Davies, please, very senior. Dr. Alfred Davies, please come forward here. Dr. V.K. Saxena, वहाँ जाना है, यहाँ है, I'm with an NI. Just come. Dr. Thiruven Kadan, Dr. G.S. Konar, Dr. Arnab Seth. So now we are going to start a roundtable meet. The speakers are Dr. Ajay Aswal, Dr. Nilesh Nai, Dr. Shudipta Haldar. All are present here. Dr. Ajay Aswal, please raise your hand. He is here. Yes. Loaded now. Presentation is already loaded. Okay. Dr. Nilesh Nai. Yes. Dr. Shudipta Haldar. Now I'm giving the mic to Dr. Partha Sengupta to coordinate.
Good afternoon, everyone. I'm Dr. Partha Sengupta. I'm working as in charge in vitro fertilization lab at Haringhata Farm under Paschim Banga Go Sampat Vikas Sanstha, Government of West Bengal. So now, as I am a person of in vitro fertilization, so this in vitro fertilization as well as genomic selection and the sex semen, how these kind of emerging artificial assisted reproductive technologies, how they are going to benefit the industry. These are to be talked. These are to be discussed in this session primarily, as well as the poultry genome, poultry industry, how this will be benefited. So may I request the first speaker, Dr. Ajay Pal Singh Aswal, to deliver his lecture regarding how industry will be benefited by the application of in vitro fertilization as well as sexing of semen. So this is Dr. Aswal, please. And I'm very much grateful to Dr. T.K. Datta, sir. Okay, start.
Respected VC sir, Vice Chancellor sir, Chairman sir, reporters. So I have given a talk which is not related to core IVF. So the topic is transforming face of buffalo leveraging the assisted reproductive technology. So we know in the genetics and breeding we have the selection and culling. So generally in India we do not focus on the culling, especially for the females. So we have a selection for the bull. So that is we are using bull for artificial insemination program. Secondly, we are also using female as well as male in embryo transfer technology as a tool for selection of the animals. So we have this priority like sex sorted semen and embryo transfer to be used first in the herd. After that we can use unsexed IVF embryos production and embryo transfer. Third is moite, fourth is sex sorted semen, then conventional semen, then organized breeding and random mating. So this should be the actual way to do the breeding in the country. But we are doing the reverse way. We are first opting for random breeding and at the last we are going for the sex sorted and embryo transfer program. And so here we have the two components in the embryo transfer program. One is the donor, where we use the elite 5% or 1% of the animals for embryo production, and then we are transferring these embryos in the genetically inferior animals. So in general we can produce around 20 to 30 calves in 1 year. And then why the embryo transfer is required? Since we are able to increase the selection intensity, we are reducing the generation interval, and then we can produce lot number of calves from the elite donor and the sire. And sometime also across the world the bulls produce for semen production, most of the bulls are coming through the IVF program or embryo transfer program. So then it's very easy to import and export the embryos rather than importing the whole animal, and it creating a lot of problem. So there are two methods, in vivo and in vitro embryo production. In vivo embryo production, where the embryos are produced inside the uterus flushed and then transferred into recipients, and then the in vitro embryo production, where we do the IVF outside. And the basic difference is that in vivo embryo production, generally in one flush we have 7 embryos, and then if we do 3 to 4 flushings in a year, so we are getting around 30 embryos, and we can get 8 to 10 calves in a year. And then we have to use the FSH. While in case of in vitro embryo production program, the initial cost is high to set up the laboratory, but otherwise the efficiency of embryo production is high in case of IVF program, where we can produce 50 to 100 embryos in 1 year, and then we can have around 20 to 30 calves. So almost the calf production is 2 to 3 times than the IVD program. So there are the differences in the Bos taurus and Bos indicus, but especially for the IVF program, I want to tell that the follicular pool is very high in Bos indicus, so we can produce lot number of calves through the in vitro embryo transfer program. So here what the word is doing, so we can see here the MOET program, 1,96,000 embryos are produced. This is the 2023 statistics, and so we see here the fresh 83,000 embryo transfer across the globe, and 78,739 are the frozen embryos. So the superovulation efficiency is 7 transferable embryo per flush, and the use of sex sorted semen is little bit decreasing, because the sex sorted semen for MOET embryo production program is not giving good results, but it is very good for the IVF program. So what about the Indian status? That almost we are not doing the now MOET program. Earlier the lot of MOET program was conducted in the country, but now it has almost decreased, and very few places I think they are doing the in MOET program. And now the government is focused on the in vitro embryo production program. So here again we can see the country wise. So average number of, this is the for the in vitro embryo production program. So 15 oocytes per OPU, and average 4 number of embryos per donor is being produced. So when we started the program, so it was only 2 embryo per OPU. Now it is increased to 3, then 4 and 5 and 6. Now most of the reports they are coming that per OPU per session per animal they are producing around 6 embryos, and this is quite also achievable in India. And so this is, and then you can see the difference between the in vivo and in vitro production program. So 2015 this in vitro embryo production program increased with much higher pace, and then this activity decreased, MOET program decreased across the globe also. So there is a decrease in the in vivo embryo production program and increase in the in vitro embryo production program. And here we can see the share of total embryo transfer program. So in vitro and fresh embryos are transferred maximum now, followed by the in vitro frozen, and then in vivo fresh and in vivo frozen. So this is the now if you compare the 2023 with the 2004, it was very poor. Most of the embryos were in vivo fresh embryos. So now the country is moving in different way across the globe. And so this is the typical status, and then most of the IVF labs in India, they tend to transfer the fresh embryos, as the frozen embryo transfer pregnancy rates are almost 10 to 15% lower than the fresh embryo transfer in this country. And then regarding the export, from the total global export, we can see 17,000 embryos were sold, which were in vivo, and 11,000 embryos were in vitro sold. And very few numbers of the frozen imported embryos are transferred, because most of the time people are much concerned whether pregnancy will come or not. So most of the time people are using fresh or using the in vivo frozen embryos. So this gives us one sense that when we are importing the embryos, so we must also focus on importing the in vivo embryos rather than importing the in vitro embryos. And this is very good statistics, where this is the where they are calculating the intensity index, means the number of embryos recorded and the population of that country. So we can see the America, the United States is producing lot number of embryos, and when we compare the ratio, so America is number 1 in the embryo index, and then Brazil, which is a 16, though it is producing lot number of embryos, but because the population is also very high in Brazil, so its rank is 16. So with that trend, if we compare the Indian population and approximately nearly in India right now only 15,000 embryos are produced annually. So, but if we go by the trend of the Brazil, we need to produce almost 4 lakh embryos if we want to just match the Brazil. Though we need to do much more. So here, because everybody is talking about the precision breeding model. So here is this is the typical model which we should follow and which will be followed, I think, in the next 10 years in the country or 20 years. One is the genomic selection of the donors and the sires. Then large scale in vitro embryo production through the sorted semen. Third is the genomic selection of pre-implantation embryos. So you have a genomic breeding value of the donor, sire, and then you produce the embryos, and then you get the genomic breeding value of the embryos. And then you transfer these embryos into the recipients. Again, the males are produced for the semen production, and then again evaluate the bull calf based on the genomics, full sieve, half sieves, and pedigree data. I think this is the perfect model that India should look for. So here it's the same as the pedigree selection. Protein testing takes a lot of time, but in the genomics we can take much less time, so we can produce more. And especially genomic breeding values of the pre-implantation embryo is a very, very important program for bull production. So typically like this here, so we are getting the embryos, getting the genomic breeding values, and then transferring only the embryos with the high breeding values. So worldwide, this is the manipulation of the embryos for the genomic breeding value estimation. So over the globe, 13,224 embryos were used for manipulations for genomic breeding value estimations. Then what about the India? How to mobilize the ET in India? So five fundamentals are there. One is the donor, then the recipient, equipment, trained manpower, and financial and administrative flexibility. These are the 5 important points in this country. So model for the donors. So donors, we can use the farm animals which have the records. Then we can have donors which are purchased from the outside, or we can do the donors at the farmer doorstep, or we can do the hiring of the donors, because it is very difficult to get the authentic data with the of the donors, and then donor is producing high, so it's very difficult to get the donors. Embryo production is not a problem in India actually now. The problem is getting the donors and the recipients. Second is the recipient. So in IVF program, the most crucial important step is the recipient, because 80% and more resources and efforts are going for the recipient management rather than embryo production program. So we can go for the large herd animals. Then we can run a heifer rearing farm where we purchase the 6-8 month heifers, we rear them and we transfer the embryos, and then we sell the pregnant heifers, or we get the calves and we sell the calf. And then at the government level, we have a breed multiplier farms, or the farms with every university and livestock farms, so we can do this one. So the training, second component is training. So we can do regular training programs like ovum pickup training on embryo production, embryo transfer. So many of the institutes are providing them training. And the DADH, Government of India, is providing funds under the Rashtriya Gokul Mission. So under Rashtriya Gokul Mission, total embryo produced under this project till date is 25,375. This is the whole India data. And then embryo transfer is 13,799, and the calves produced 2,055. It's very, very less. And then government is also running the accelerated breed improvement program, where government is giving 5,000 rupees to the farmer, to incentive subsidy to the farmer, and rest the farmer has to pay. Though it is very difficult to get that money from the farmer. Third is the breed multiplier farm. So all the breed multipliers farm funded by the Government of India has a mandate to perform the IVF program in the country. So these are the various labs. There's almost more than 30 labs in the country. The IVF program under the livestock boards, NGOs, NDDB, and other ICR institutions, universities. And status of the IVF program in India. So we have a average recovery of almost 12 oocytes, and then embryo conversion rate is 3 or less. Consumption rate on IVP embryos is 30%. Consumption rate for the frozen embryos is still further less, almost 20%. Demand for the embryos is only for the Holstein, because if when you are getting the imported embryos or we are getting the Holstein embryos transferring, so by next 2 years you will start getting the milk. But for the indigenous program, you get the embryos in transfer, and then 3 years it will come to maturity, and then making it pregnant. Almost 5 years it will take to get the money back. So it's not a viable program for the indigenous one. So if for indigenous one especially, it is for the bull production program, but for Holstein and Jersey, so huge demand is there for the embryos, and that is also a viable model. So like private factors are there, like Godrej, Sham IVF, Palamur, there are some of the labs which are coming up in the private sector. So this is SWOT analysis. So what we expect in India? So typically in India we cannot follow the Brazil model because we don't have a SWOT policy here. So what we can do, a moderate approach we can use, wherein we are using MOET programs. In one cycle we are using one lactation, we are using only three MOET programs, and then making the animal pregnant. Similarly, for the IVP, in vitro embryo production program, we instead of going for 20-30 OPUs regularly, so we can go 15 OPUs and then make the animal pregnant. So with the 15 OPU, we can get easily 60-70 embryos, and we can get 15-20-25 calves from that one. And then for the recipients, because recipients at the farmer doorsteps, and our consumption rate has to be high, because at low consumption rates the intercalving will increase very high. So therefore we should not give more than 2 chances to make a cow pregnant with the embryos. And in MOET program we can expect around 45% consumption rates in fresh, and then 40% with the frozen. But similarly with the in vitro dried in vitro embryos, we can expect around 30% consumption and 20% in the frozen one. This is quite low, almost 10 to 15% less than the world average. And in India, if we want to make this commercially viable, we also have to focus on the frozen embryo transfer program. We cannot rely on the fresh embryos every time. So we need to develop our media technology, and we should freeze only the embryos which are the best ones. In India, typically what we do, we get the embryos and we transfer the best embryo and freeze the worst one. And that's why we are getting very poor results. So if we want to increase it, best embryos to be frozen first, and then rest embryos to be transferred one. Of the total expenditure program, as I stated earlier, 80% goes toward the recipient management. And we are least bothered about the recipients. We are focused about the donors, lab should be hi-fi, and everything should be very good. But what about the recipients? So we need to focus on the recipients. This is very, very important. And another important point is that import duties. So there is high import duty on the embryos. Similarly, in the FSH, we have a high import duty. So the government may decrease the import duty on this one. So this is our ULDB model, but I am working in the Uttarakhand Livestock Development Board. So we sell the fresh and frozen embryos at 4,500 rupees per embryo. We then sale of sorted embryo pregnant heifers post 3 months of embryo transfer, that is 1 lakh rupees. Sale of embryo born bulls for the semen station, it ranges from 2 lakh rupees to 4 lakh rupees per bull at 6 months. Then sale of embryo transfer born heifers, rupees 5,000 per heifer non-pregnant and 75,000 rupees pregnant heifer. Then we are also charging consultancy charges. Some institutions are coming to us, we are charging. And then we have a OPU IVF training program which we regularly run. So we charge 1 lakh rupees and plus GST extra. So this is our revenue model. So typically, what should be the profitable model for this embryo transfer program? Because we cannot alone take the embryo transfer program. That's why we could not do very good in this country. So what we need is that the first bull mother farm and embryo transfer facility. Second is the heifer farm, because every time we cannot rely on the farmers. So we need to have a farm where we can do the embryo transfer program. Third, we should have a genomic lab or its interface. Semen production unit with both sex sorted and conventional semen, because as we produce the male or female, we have to get the money back. So semen is the model where we can get the money back. Yes. So then we need to have inseminators, then farmers and AI training centers, milk recording system with servers for the data, disease screening facility, and human resource for continuous training. So everything is required for a successful program. So this is our farm. We have Red Sindhi, Sahiwal, Gir, Thar, Parker in our farm, along with the Holstein and Jersey, which every time we import the embryos and we transfer the embryos and we sell the male calves to the semen stations. So this is our facility, we have farms. So earlier we used to do the MOET program. So this is our MOET program. Also we are every time importing the embryos for the bull production program, and we are selling these embryos to the semen stations. Then we have this MOET, this OPU IVF program in our farm, and our main purpose is to produce the bulls for the semen stations. And then various model, as I was talking, this is a farmer's cow, and we have hired and we have given 80% female calves back to the farmer. So we got the genetics, we got the bull for our semen station, and females we have given back to the farmer. So this is the model. Another model is this: we have hired one cow from the outside farmer, and then we produce calves, and then again sharing was there for the calves. Then this is the training program we are providing to the almost every states. We are providing the training programs. So this is the consultancy. Again, we are providing the consultancies to the universities, to the other institutions, and getting the money back. So we have a field embryo transfer program also. And this is about the semen station. So we have our semen facility. We have a conventional semen as well as the sorted semen facility. So we are the bulls under the collection, and we are producing the sorted semen since 2017. It was the first lab in the country to have a sex sorted semen. Yes.
Thank you.
So thank you very much, sir.
Okay, thank you very much. I have to leave for GB meeting also, and my flight is also there. So I'll just give just 2 minutes. I will give my comments on this roundtable meeting or other academia industry interface. The title of this interface has been very aptly chosen: transforming face of livestock and poultry industry, leveraging the emerging biotechnologies. We have been listening a lot of science from yesterday to now also, and the very purpose of this interface is how these sciences can be converted into technologies and transferred to the industry, so that industry can be helped, and in the way our country should also progress. The ultimate aim is to increase the production at low cost, reducing the cost and improving the production of livestock and poultry. So industry has a major role. Academia, of course, takes the lead in developing methodologies, technologies, or the way forward, how such interventions can be made that can lead to higher production at a lower cost. So in that way, our scientists, SAUs and institutions, research labs, they are working hard. And from my FAGB, I'll urge all my friends from livestock development boards or from industry to be part of this association, ISAGB, because if you strengthen this association, in turn this association will strengthen you, provide you the platform, include you in their discussion, and also help you in resolving your problems through doing research on those aspects which are raising problem, or the industry is facing problem on those aspects that can be very much taken up at institutes or at university level. So that I remember I had been in Wisconsin. This mastitis was simply resolved by taking a protocol of sanitation. No more, medicaments are applied. Only sanitation procedures they given to the farmers, and this mastitis problem was resolved quite a lot. So such intervention, simple and easily operable at level or industry level are required. Yeah. So thank you very much.
Can I make a comment? Yeah. In fact, the very purpose for this particular session was to see that we have chosen 3 topics. One is the OPU IVF, one is the genomic selection, another is the poultry.
So we do many things in the research lab out of our excitement or euphoria or whatever it is. So when you start doing it in the field, then how it has done. And I think currently in India, there is no other better person than Ajay, who has lived with the mandate of the IVF and first ET and then IVF, ETT and all that. But the person who is sitting to your left, he has seen the world. And I would like to get some comment from Alfred about how do you feel the India's data vis-a-vis the other countries' data, and what's your experience? You take lead, so I will replace you.
Yes, Narula, Dr. H.K. Narula, there was a misunderstanding. If you are here, please come up with the dais, please. Please. I missed your name. Please come. आ जाइए प्लीज सर आपके पास है There was a misunderstanding. I didn't call your name.
I wanted to ask you that Ajay has given you a very exhaustive data about how we are doing it in India, and this is a story of last 7-8 years or maybe last 10 years or so. So you roam around the world, you see others doing it. So can you please comment on how we are doing it?
Yes, so I can comment on that. It was mentioned that there are 30 labs that can do this across India. So then if you, every year I look at the reporting from government or from NEDB to find out how much semen, how much embryos, and those numbers are low in India. So then I think that's a very costly infrastructure to have this relatively small amount of embryos that are transferred. And then I know that there is subsidies by government under the RGM. So I think if that would be taken out, then there would be hardly anything. So government, and that's, I'm always very careful here what to say, but sometimes I see that a lot of infrastructure is provided, but then it's not used much unless
exactly the same thing he said.
Yeah. So, and you think, and then if you look at other countries, Brazil is a very good example. It's very well commercialized there. Then also I sometimes think they do too much because also they have a lot of costs. But it is a lot also there with the meat animals, that they have some animals that they have done a lower, because it's done a lot on the lower in Brazil, not so much in the dairy. So why do they do it in the lower? Because it's also a status symbol. So there are farmers who just want to have from a certain very nice bull or from a very nice cow, they just want to have more of those. They even do cloning of those things. So that's a very costly procedure. So there are some strange situations economically. And if I look at Europe and the US, that is mainly in the dairy that it happens. It's not in the meat sector, it's in the dairy, and that is fully for the bull program. So embryos are sold, but that's usually the surplus that they have. So I also work for a cattle breeding company in the Netherlands, and we produced embryos just for having the best bulls possible. And then sometimes we didn't have enough recipients. So then we sold those embryos to farmers, and then we'd buy back for if a good bull would be born. So then it is really there as a byproduct, this whole embryo transfer. Okay, there are also farmers who like to have faster upgrading, and they pay it as commercial service. But they pay the full cost. There's no subsidy, but that's very limited. Thank you.
Thank you. Anybody others? Any other comment? If not, then
I just want to stress, but then very shortly, that cost is one important thing. I touched that, and you also, I think all of us are aware of that. So hopefully research will enable a lower cost price, that it is more affordable. But the biggest issue is the pregnancy rate that is too low, especially if you go to frozen embryos. You have, and you showed it, it's again 10% low pregnancy rate. That's not what the commercial farmer likes. So it should be comparable to when you use seed.
next speaker, Dr. Nilesh Naeem. He will be, that his topic is genomic selection. How industry will be benefited by the process of genomic selection. This is about.
Thank you, sir. Nilesh, your funder is here and the ground is here and you are enjoying the hit of genomic selection. So we thought that there would be none other better than you to talk about us. Tell us that what we are doing at the field.
Thank you, sir, for your faith, and I will touch upon some of the, like, I was a bit confused, what is this industry and academia interaction? So I thought, let me focus how genomics has been used world over and how we can benefit and what is practically right now being used in our country. So not much repetition from previous talk. So what is genomic selection current status and then benefits both at farmer level and industry level, and how industry, and by industry what I mean is now many genomic service labs are coming up in India, so how they can participate with us. So that's how I have focused. And last presentation has this slide, and this gives a beautiful meaning. Like now anything you want to improve in livestock, dairy, cattle or buffalo or anything. Just now we have got a tool like for pedigree based selection process we used to wait for years together to build up a program in this. But here now as soon as you start building up your phenotype recording system, genomics is a tool which can leverage and give immediate results for affecting improvement. So this slide I like very much. You can improve efficiencies, you can improve animal welfare if you wish to, and if you want to impact on environment inside that also. Only key is phenotype. So I explained this and I'm not going in deep detail for benefit of students. I made this slide, so it is not really scientific slide, but genomic selection is comparing with reference and then testing this test animal where it fits, and then you can get its genomic breeding value. So this is not but as I told, whatever you want to improve as an academicians or as an industry, we all together have to focus on phenotype. Phenotype is still a king in any kind of improvement program. And not only collecting animal-wise or individual animal-wise record, collecting accurate record is the key. And that's where everybody has to work. And then I have seen in our country, there are many programs, like ICR is running one program, government is running one program, some of the state institutions are running programs, some of the agencies are running programs for recording, but they are not standardized or they are not at one platform. So those records, when you want to combine, it is a challenge. And that's where we have to focus. We have to develop countrywide system of standardizing recording. And probably government of India has provided platform in terms of Bharat Pashudan, which gives opportunity, at least in cattle and buffalo, to record almost any aspects of the production as well as health and reproduction. So that platform can be used for this kind of work. In our lab, because I thought there will be some industry people also, so I thought to put up this slide, how we work actually. So for creating reference, we record animals that I discussed yesterday. Blood samples are collected, their DNA is extracted, DNA is tested for its quality, and then it is hybridized with genomic chips that I will show, and then it is processing for 2-3 days for amplification of those probes and then washing and all those, and then finally stained and scanned in specific machines like iScan or Gene Titan or what platform it is. And then this data is used for further analysis for genomic breeding value estimation or other use which I will show. So we have for specific for Indian cattle and buffaloes, we have now 2 unified genotyping chips. Go chip for cattle, suitable for both crossbreds as well as indicus breeds. So here, like most of the breeds, we get 50,000 plus informative SNPs. Informatives, when I say, is for our internal criteria was at least 10% polymorphism in that specific breed. So 50,000 plus informative SNPs in any of these breeds we will get. And then we have like genetic conditions, like whatever genetic conditions we want to screen, those SNPs are available in this chip for Holstein and Jersey, which are reported open source SNPs are there, those we have taken. And then ISAGB parentage discovery panel is part of this chip. For buffaloes, we don't have that big research on identifying those SNPs for specific genetic defects and all those, so we haven't yet included, but we are in process of screening databases and getting those SNPs in our. And this buffalo chip is suitable for both riverine as well as farm buffaloes, so it works well in both kind of buffaloes. As far as reference, because genomic selection doesn't work without reference, so reference is recorded female-based reference, and then some part of proven bulls are also, and now all the bulls in semen collection are part of our reference, and these are the methods which we use. These are number of reference and current reliabilities, what we get for average reliability for unrelated sample which we get. These are the reliabilities for unrelated samples. Like unrelated means not in the reference, and we don't have other information for this sample. So this, like, we do a lot of validation studies with how it is working with that animal's performance or the daughter of the bull's performance. So this is the validation what we have done, and this improves with increasing reference population, and we are still beginning this process, and that's how it will increase in future also. So how at population, definitely we will get benefit of increasing productivities, improving quality, reproduction, disease incidences. Another part is like yesterday we were discussing about inbreeding. With genomics, you can avoid inbreeding also, because you can predetermine whether this mating is increasing inbreeding or not, and then you can either avoid or promote those kind of breedings. So this is what reducing, and whatever you think, whatever you can record precisely in a reference, you can improve through genomics. Another is like which breed composition works well in my particular area or particular, like a lot of crossbreeding we are doing. So we say whether 60% is okay or we should go up to 90%. So genomics can give you that answer, because you have now breed proportion based on your genotype data. We don't have deep pedigree, but breed proportions can be worked out, and we can see which area we can get better productivity from particular. And at state level, suppose you have breeding policy that in my state 50% works well, so I will say we should not have exotic inheritance more than 50%. In many of breeding policies they write 62.5%, but how to monitor that? Do we have any tool? Then genomic is the tool available for us. And of course inbreeding can be monitored. And you see, like an Australian example, after genomics they could lot many traits which were not possible before genomics. So similarly we can do, only thing is we should as a whole, as a collective efforts, industry, academia, everything has to focus on record those traits precisely. At industry level, like breeding industry, I will say, not dairy industry as such, semen stations, they will be able to get bulls with more accuracy, because then farmers will be actually putting faith in semen industry that yes, what they claim can be delivered. Because till now we were not very I will say farmer was using semen for reproduction purpose, not actual breeding purpose as a whole. So this can be increased confidence of farmer in our industry. Second is same genotype can be used for lot many things like parentage, genetic defects, inbreeding, genomic mating design, that which mating can give us probably better bulls. And of course now there are methods that only genotype can tell you whether this particular sample has chromosomal abnormalities or not. So no need to do separate karyotyping, and this can help. So, and then many more use cases are being applied now. So a lot of research in that. Farmer level, when specifically indigenous breeds farmer wants to maintain purity of their herd, then they can test for breed purity with genotype data. Of course, they can select heifers for replacement at a very early age, and now costs are coming at reasonable level. As I informed yesterday, it is now 2,200 rupees per sample testing for getting genotype. So that costs are coming very down. Accurate parentage information, genetic defects, and purchase decision. Now, very interesting use case I have found in industry. Some of the industry people who are selling animals to different farmers, now they send samples, and based on genomic breeding value, they charge farmers what is the cost of this particular animal. Of course, production data and everything is there, but to gain more confidence from farmer, they give. And then there were inquiries from outside, like Kenya and Sri Lanka and some countries who wants to purchase our crossbred or our indicus animals. They also demand what is the basis of selecting this animal, and that's where genomic breeding values can give them confidence. So these are the cases where we can use this particular thing. How farmer gets service in our lab, like they send either blood or tissue punch or semen straw, and they can courier it in cold conditions. We have made a portal for anybody to avail genomic services, and with this portal we are now mapping different labs who can provide services. Labs can analyze sample, generate genotype, and submit us, and then we can give genomic breeding value. That kind of model also we are trying to develop. So this is the report which we give to an individual farmer or the person who is asking services with us. So these are the things which we give. Genotype service provider can participate in this program by this way. They can have genotype through GoChip and MyChip. Government of India has already dedicated this chip and agreed that any agency or institution in India can use this chip. So that is there, and NDDB has reference data for genome breeding value estimation. Any lab can submit genotype data, and we can give genomic breeding value for whatever reference we have based on that. And we don't have to charge anything because it's anyway part of national database, so we don't charge. Only permission you will have to give is this genotype, if found suitable, we will use in national database. So that is the benefit of sharing and samples, and then it will increase accuracies in future. So this is what, like practically how industry can be benefited with this. Thank you.
Thank you very much. Thank you very much. And, that's what it is. I was expecting a little bit from your lecture about the cost of it also. You said something.
So it is 2,200 per sample.
Fine. Then one is cost, another is accuracy.
So I gave some.
I think this is a very tricky game. And when you select the genes to decide the destiny, then you have to be very, very careful that you are selecting something for a useful purpose. Because there, if we do a mistake, then we take the population entirely to a different kind.
Yeah.
I mean, you know that when the government of India decided to launch this program, I was the national head. Then due to something, I thought that I call it a day. But I always say one thing, that my animals in India, in spite of so
nutritionally deprived, but some of the animals, they have performed so well. So it must be something. You know? The condition in Europe and America is very different from that of us. Their animals are well fed, they are doing well. But my animals, they are less fed, sometimes hungry, but still some of them are doing well. So that is the ray of hope probably. But anyway, I think Alfred, some comment.
Yeah, well, I have some questions also related to what you were saying, the different traditions in the US and Europe versus India. So if you buy Holstein embryos, or maybe you buy semen from certain bulls in the US, how do you choose? Do you ask for DNA and then that DNA is going through your predictions and then based on that you make? You were laughing now, so I expected an interesting answer.
A very different and very interesting question. Ideally, I should be doing that. I should be asking genotype data and then comparing with my base. But unfortunately for pure Holstein, I don't have database. And of course, like argument is, I am importing germplasm for crossbreeding program. So shall I be comparing with pure Holstein with my crossbred database? So these are all tricky and technical issues which doesn't allow to do right now. But yes, in future, our goal is to develop some kind of Holstein and Jersey database within country.
Nilesh, I think there is some dichotomy. Dichotomy is, for example, in ICR institutes, we have gathered the data over the last 50 years. And we see that an animal is fantastic, doing well. But the so-called GEBV that you give, that does not qualify you to be included in a national AI program. So I take it with a pinch of salt that when the government of India and the NDDB or whoever, they were making these algorithms, you never ask for the data from ICAR. And whereas ICAR, if you look at IBRI, NDRI, CIRB, and so many other institutes, we are sitting idle with that Excel sheet, having the data. So where is my data? And I have the semen samples from those animals, and the DNA is there. So I don't really need I had a grudge, so I shared it to you. But I really don't want anything. But please think about that. Whatever the very gold mine we have, it should get a representation.
I can just put a piece of information. Last month or maybe 2 months back, we had a meeting, ICR, NDDB and Government of India, DHDP,
Very good.
sitting together
at last
to work at a mechanism where we can include these on
I'm
our
very happy to
records.
see. I'm very happy to see.
Can I add one thing? Because indeed you don't have the Holstein as a reference population here in India, so you showed what you have. But I was thinking the crossbreds you have, so the Holstein with Gier and Holstein with Sahiwal, that could be a source that can tell you more about the durability of certain Holstein genes, what it brings here to the population in India. Is that correct, or is it the wrong assumption?
It has like background noise of indica's genome. It has background noise, but still something is better than nothing.
Yeah, because you have this, there's this publication together with NDRI, I think, where you show that either Gier or Sahiwal, it helped the small breed kangaroo. So that's a very nice example already where one type of reference population can help another breed. Yeah.
Thank you.
Thank you for nice presentation. So major thing is about the reference population. So we have very diverse breeds. So how much it will be accurate?
So yes, we try to go and measure in various environments, not only like specific area or something. Like if you see our Mura reference is from 4 geographical areas. Of course, Gier I can do in Saurashtra itself. Sahiwal I can do in specific pocket, but for breeds which are available across, we try to put recording program on those areas. So it's diverse kind of reference.
next, we will be switch on to the same genomic selection, but the poultry industry. How this genomic selection will be benefited to the poultry industry. For that, the speaker is Dr. Sudipto Haldar. Please come up on the stage.
Sudipto remains a student of my university, and here is the man. He is a very courageous speaker, and he has lived with the industry and living with the industry and making us all proud. Sudipto.
He is now agri-vet research
He is a nutritionist.
advisor.
Yes, I'm a nutritionist. You are a nutritionist, I know, but I have put you on a challenge of speaking on genetics.
That's great, sir. In fact, I'm a nutritionist.
Don't take these jargons of genomic selection.
No,
You restrict to genetics. That's it.
Basically, I'm a nutritionist, so I really don't know anything about genetics. I can just remember those Mendelian laws and all these things, but nothing else, to be honest. But in fact, this is a challenge, and what I can do right at this moment, that I can share something of my ideas. And since this, I thought that this is going to be an industry academy interaction, so I didn't come up with a PPT, because I thought making it a bit of that interactive should be a better option. Now, since this is a poultry genomics, the discussion is on poultry genomics, and as I said that I'm not at all a geneticist, so probably I can start with the conservation of the breeds. And if you don't mind, sir, then, and if I ask you a question, or rather a request, to update me about the present status of the conservation of the indigenous poultry breeds in India, then can I get the answer from any one of you, from the audience or from the dais? Is it possible that I can get the answer that what is the status of the conservation of the indigenous poultry breeds in India? Yeah, anybody? If it is, yeah.
Oh, sorry, I have. So, the most authentic data set comes from the NBAGR, the National Bureau of Animal Genetic Resources, where we have this kind of, but let's not make it a question-answer session. Please.
No, actually, my question, it is not a question, rather. Means I'm interested to know that this conservation of the indigenous breeds, so poultry breeds, what is the motto of this conservation process? Is it just to make a museum, or just to make a future asset?
No, we want to make it, we want to improve upon it and give it to the farmers. First of all, we want to identify. Before conservation, I always ask two questions. One is, what to conserve and how to conserve, something like that.
Yeah.
So if something is useful, then only I should spend my time, energy and money to conserve it. If something is not so useful, why should I bother for it? So I think one of the objectives is to characterize them and to know their production features and their phenotypic features and all that.
Okay.
If we find it useful, then we take initiative, we take it as a project to improve on this.
Perfect. So what sir said, what the Vice Chancellor has said, that this is the objective of the conservation is to identify the usefulness of the conservation process. Am I correct? Yeah. The conservation process. So in that case, I'm just trying to bring up certain features of the indigenous breeds as well as the commercial poultry breeds which are available in India, or maybe globally. Now the commercial poultry breeds, the broiler chickens or maybe the layer chickens, it is all known that we all know that this is something which are prolific producers. We can say that these are simply production machines. We just put some feed before the chickens and they eat them up and they grow. In 35 days they produce almost 3 kilograms by consuming only about, say, how much? Maximum, say, 4.5 kilograms of feed, which is just fantastic. We could not even think about this situation even 20 years back. So if I consider the feed conversion ratio, that means how much of the weight is being produced by consuming 1 kilograms of feed, or 1 kilogram of feed, how much meat can be produced. So the figure was something around maybe 2 in 2000 or 2002 or 2003. But nowadays we are getting the feed conversion ratio of around 1.3, 1.4, which is something fascinating. And if this is the case, in that case, I should say that this has come at a cost. It is not really, it has not come without any cost. Because as we see that the broiler chickens or the laying hens, they are producing a lot of meat, a lot of eggs, but at the same time they have become disease susceptible, hugely disease susceptible. The susceptibility of to disease has increased enormously. Means really, if those who are dealing with the chickens or the poultry industry, they can know that this has become a menace nowadays. The mortality of the chickens, different types of diseases, different types of metabolic disorders, everything. So this is a problem nowadays. Now, why this is so? The reason is very simple. The reason is simple because the birds are growing fabulously, means they are growing just like a machine, and while doing so, the birds are getting deprived of their immunity because the selection pressure has been put on certain production traits. For example, breast meat yield. This is the primary criteria for a broiler chicken. And if the selection pressure is on the breast meat yield, in that case, definitely the pressure cannot be put on the disease resistance. If I take an example of this particular breed, Ross 308, probably those who are familiar with this chicken industry, they might be knowing the name of Ross 308. This 308, it is a product from Aviagen, and this breed is a prolific producer. This breed can produce almost 3.5 kilograms in just 35-36 days, consuming just by about, say, 4.5 kilograms of feed. That means at a feed conversion ratio of 1.3, 1.4, which is fantastic, just fantastic. But while doing so, this breed cannot survive a condition, an environment which is a bit harsh. The environment which is prevailing in the Indian subcontinent is really too harsh for this particular breed. The same situation the company, Aviagen, was facing in many other countries in the Latin America. For example, in Brazil, the Brazilian condition is somewhat similar to what we are experiencing here in India. And there was huge mortality. The breed is so fantastic when they are put in the environmentally controlled house, they are producing good, they are behaving nicely, but whenever they are put in the open houses, the birds become a menace for the farmer. So why this is so? The only reason is that the birds were selected only for production and not for any disease resistance or any metabolic disorders. So just to prevent this situation, or just to rectify, the breed company has decided to insert some local genes from the local chickens, and they inserted a Brazilian male line which are characterized by high disease resistance or withstanding some metabolic disorders under harsh conditions into the female line. And they have come up with a product which is termed as the Aviagen product, and this was developed in the year 1995, and it is termed as AP95. Nowadays, whatever chicken we are getting in the Indian market, a good number of chickens are these AP95. That means the main female line, it is enriched with some local breeds having some disease resistance or certain criteria which can withstand the metabolic disorder. So this is called something, I should say, that genetic modification or genetic editing, I don't know. You might be knowing better than me, but this is something which is very unique, very unique and quite interesting. Now my point of observation is that I have observed that whatever the native breeds are there, the desi chicken, we call them as desi chicken. Now these breeds, they are not prolific producer. We cannot say that they are very prolific producer, but at the same time, they go on scavenging. They pick up something leftovers. They are not on regular medicines or even vaccines. They miss even the vaccines. And at the same time, what we find that their mortality rate is very low. In case of commercial poultry production, the most crucial part of the productive life cycle is the initial 7 or 10 days, because it is during this particular period of time the flock suffers the maximum mortality. And interestingly, these native chickens, the desi chickens, they do not show that much of mortality or even very low mortality during the initial phase. Now, if this is a trait we consider which is important and can be inserted into the commercial poultry line, then probably it is going to bring some sort of, the kranti. Am I correct? Because mortality, the early chick mortality is a very, very important, a commercial trait that every farmer is looking for. And if I find that a particular breed is being developed or modified in a way that imparts the ability of the birds to survive the challenges imparted on them during the initial 7 days, then the mortality which the flock is going to experience at the end of say 35 or 40 days of life cycle, then there is, I should say that this is simply fantastic, and this is something which can be thought about. Now there is another point. Nowadays,
Sudipto,
yes sir?
I could see the clock that you are left with 8 minutes.
Yes sir,
So please save 3 minutes for me.
Okay.
Alright, so finish it 5 minutes.
Oh, so you need 3 minutes from me.
Yeah.
Okay, sir, no problem.
I have some useful things to discuss.
Okay. So since I need to cut it short in 5 minutes, so I'll just talk another way. I mean to say that nowadays we are thinking or we are talking a lot about the gut health, antimicrobial resistance. And you will find that these indigenous or the desi breeds of chicken, they can survive on anything. And probably you won't find this problem of this gut health in these particular breeds. If you put these desi breeds here and the commercial breeds on two farms side by side, then you will find that on the same diet, probably under a bit of critical condition, these desi breeds will not show any sort of gut problem. What should I say, that this diarrhea or something like that, or bacterial enteritis, dysbiosis, blah, blah, blah. But these commercial breeds will show. Now, why this is so? This is a very interesting situation. Is there any difference in terms of their major histocompatibility complex? Or is there any difference in the whole microbiome which is present in the gut of these particular birds? If the microbiome present in these desi breeds of chicken is completely different or having some unique feature, I mean to say that there are certain unique bacteria which are capable of withstanding the pathogens present in the environment, is it possible that we can culture, identify and translocate those bacteria into the commercial breeds at the very beginning? If it is so, in that case probably the reliance on antibiotics can be prevented or can be reduced or can be minimized to a great extent, because it is this particular gut health that the producers have to depend on antibiotics to keep their flock healthy. And if this is the situation that the desi breeds, they are having an indigenous property to withstand this enteritis or the dysbiosis problem, then probably culturing those particular microbiome, identifying the microbiome, that which particular candidate is there that is actually preventing this dysbiosis to set in these birds. Is it possible that those particular microbiome components is shifted to the commercial breeds? I don't know where, maybe during the hatching period, in over-injection, or during the early rearing period, that they can save the life. So these are certain areas actually, to be honest, that the work can be done, and to be honest, certain works are being conducted, are being. One thing we should keep in mind, that while working with this desi breed of chicken as well as this commercial breed, I find that the desi breeds are not disease resistant. I must say that they are not disease resistant. They need vaccines, they need medicines, they need everything. They are actually disease tolerant. The main issue is to find out that which imparts this tolerance to those particular breeds. And if this disease tolerance factor can be identified and can be imparted, or something similar can be developed in the commercial breeds, then probably the commercial breeds will become more profitable. The way the poultry industry is growing in India and the way the turnover is increasing every day, then probably this can be a very good initiative to start work on this particular subject. Sir, I'm giving you 4 minutes and 36 seconds.
Thank you very much. You raised really very important issues. But this whole session is about the industry academia. Now, I have a question. Question is, what is the industry's responsibility for this? I know that this poultry industry currently in India has, it's a giant industry. I find many business houses who are worth 1,000 crores, 600 crores, etc., etc. Now, but unfortunately, I don't find a good appetite of the companies to collaborate with the academia in sorting out the issues. Whereas, sometimes I feel that we academicians and you industry, we are living in two different silos and we hardly talk. And when it comes to doing research, of course the funding matters. And maybe when the industry and the academia, they join together, we can really do something very good and give answer to the solution. I mean, fortunately, you represent both. You are from this academia, you are doing good money by doing business. But I think more and more of these things, other day, I was sitting in a meeting at Lonavala, and a lot of poultry industry people were there, Poultry India and all that. So I was asking them that, can you have a chair in our poultry science department, which will be paid by the industry? So the industry man says, yeah, why not? Tomorrow I will deal. But what you are going to do for me, you Vice Chancellor? I said, you tell me, what can I do for you? He says, go and tell government not to make so hue and cry about the AMR and the antimicrobial resistance. And maybe government is shouting too much about the AMR. And if we follow their guidelines, then the poultry industry will be, ruined. And the data says that there is nothing to worry so much. But I think this is a give and take kind of a proposition. And any comment that, do you think the industry should collaborate more with the academia?
Right, sir. Industry should collaborate more with the academics, as well as the academics should also collaborate more with industry. The thing is that
academicians should collaborate.
Yes, because the academicians, if you ask me honestly, I'm your student. I should be honest with you that the academicians, they have a certain what should I say, that
ego.
Yes, we are in a superior position.
No, you are correct. So I think this is a very valid.
The point is, I just want
that when we decide a research topic, then to do research, we hardly consult the industry. Then what is the industry's problem?
Actually, sir, the need of the hour is that whenever the research program is designed, then that should be directed towards the need of the industry, the society.
In that meeting, in fact, Sudipto, there is a suggestion comes, is that for every student's advisory committee, there has to be a representation from the industry. Because the industry knows what is the problem.
Exactly.
We live in a kind of a magic box. I know, engrossed with my papers and internet and doing that, and we have very poor idea about what is the problem in the industry. So we should come closer, probably.
Now, honestly speaking, I must say that the barrier is probably it is getting thinned up. Recently, we have tied up with this NICE, National Institute of Cholera and Enteric Diseases, and we have developed certain the unique solutions for the industry, and you will find that the same product is going to be launched in this Poultry India Expo Hyderabad on 26th of this month. So a particular scientist who is involved.
Comment from my colleagues.
Yeah, sure.
No, I just wanted to comment on what you mentioned about if those very commercial animals, the broilers, I think what you normally talk about, if they go into the more challenging conditions and having, for example, scavenging then you get into a lot of problems. You have a higher mortality. That is very logical because they are very clean animals and then they go out immediately in very bad conditions. A good way of solving that issue is to bring those animals first into rearing with quite good farmers that make sure that they have good feed and also that they get vaccinated for NCD and things like that. That's a model that is used now in African countries quite widely by, in fact, local hatcheries that support that. They train their local agents that keep them for 4 or 5 weeks, and that model that has been the solution for the issues that you were just saying. So that's also, you think about the microbiome, it also gradually gets acquainted with what is in the environment if you do it in that way. The other thing that can help you is that you said you have either the commercial or you have the indigenous. There are also products that are slower growing, which are still having a very good productivity in meat, but also in eggs. So they call them in Africa now quite widely dual-purpose poultry, but also in other continents, these type of animals are also there. You have companies like Hubbard, Sasso, is a brand. And those animals are very successful now under those African conditions. They need to mix. They have a lot of lines available and they just need to make the right combination of lines. So I think there are more solutions than you have mentioned.
Yeah. Thank you so much. Thank you.
Professor Narula, any comment? Actually, I'm chasing the lunch time. This is 2:30. So yeah.
Thank you, sir. You have raised very valid points, and I think there should be very good collaboration with the industry people. But sometimes some of your procedure and other industry people, they are not able to cope up with us. Yeah, otherwise our side. But our two issues are working very good research project. You must be knowing DPR and carry, and they are taking the care of the new problems and emerging problem of industry. Maybe not.
Same thing is happening in Namakkal as well.
Maybe not 100%, but they are trying to cope up. And I hope so. Best if these type of interaction are there, we can do good work. Thank you.
Excellent.
Thank you for your information. So we are having a BTech poultry program, sir, where we are having a very good collaboration with industry, sir. So last one semester they will go for industry, sir. They will work in the industry and they will get stipend also, and they will come up with their own projects according to the industry needs also, sir. So we are a small group of people every year they are going to the industry, and we have an association, sir. We are developing that one also, sir.
Thank you. Thank you. I see Professor Unandu Biswas in the audience. Professor Biswas, any comment from you about the discussion so far? The issue is the industry-academia collaboration.
Thank you. Actually, this is very much required. It's true, as Sudipto said. But again, the problem, it is not the only, what he explained, that not only that ego of the academicians.
Vice versa, sir, actually, vice versa.
But also the business policy of the industry. They want to keep it hidden. So that is also a reason. But now, what I think this research collaboration is very much required for all, dairy cattle, and because poultry, because our final motto is to generate any technology
and serve people.
To serve people, for the benefit of the farmers. Right. So that is there, and regarding this deshi chicken and this commercial birds. Yes, now of course, if we consider the productivity issues, then of course for large scale production, commercial production, we need these commercial birds. But again, there's a market, that what we call premium market. It's like you know that from CISAGB, how people, local people are coming for purchasing this raw meat from CISAGB, and even from the goat institute, they come for the goat meat and that's a very high cost. So that we must explore that kind of premium product marketing.
Exactly.
That
niche market, what we call a niche market.
For the people, all types of products should be available in the market. For the premium market, okay, this we can promote this chicken with a little bit of improving it. Of course, ICR, they have already produced some, developed some strains of like Bonraja, Crampia, and others. So many. So they are also very popular now. So that way, that can be explored.
I think there is some kind of a mistrust between the industry and the academia.
No, sir.
In the industry, this I have seen. So the industry feels that if I dump money on there, so a kind of a refined information will not come out of it. The academicians will do work in their own way. They will publish papers, this and that, and I have no interest in that. Whereas academia feels that, well, I have a well thought of project and I need $1 million for implementing this project. And the industry will then go for a bargain to say, why do you need this kind of money? You reduce the money doing this, doing that, doing that. And as we say always, that the cheap things are not good and good things are not cheap. It's not that. So I think the one message that you can take it in the proceedings is that from this ISAGB platform, we are recommending that industry should come closer to the academia, or I say academia should go and beg to the industry that at least tell us your problem so that we can work on this.
So basically, I must say that industry doesn't have any inhibition. Inhibition in working with the academia, because when I was working as associate professor in the university, then I personally brought a good number of projects from the giants like AB Vista, Novus, DSA, and we did a lot of work, sir knows. And we developed a small poultry house as well. We could purchase some machines as well. We published a good number of papers in British Poultry Science, Animal Feed Science and Technology. So animals. So these are all our achievements during our work period. So it is not true that the industry is hesitant. The only thing is that the deliverables, that how the academia will deliver the results, with what efficiency. That is the thing. So once that is there, once the professionalism is imbibed into the academia, and I'm at the head of the contract research organization, I'm doing the same thing. I'm carrying the same legacy, what I used to do in the university, but with more professionalism. That's it.
Very well spoken, very well spoken. I think we conclude this session with that particular note. And thank you very much, Sudipto, for acceding to my request. And you are the only speaker in this conference who spoke non-PowerPoint.
Yes, non-PowerPoint. I'm a non-geneticist and a non-PowerPoint.
Now I request Dr. Datta to our Honorable Vice Chancellor to hand over the memento to Dr. Sudipto Haldar. And again I request Dr. Datta, Honorable Vice Chancellor for the University to hand over the mementos to our chairperson. Now we are concluding, yes. So with that, we conclude the business of this ISAGB, and let's proceed for lunch. And we are rejoining at 3:30. 3:30 means 3.30, and for a valedictory function over there. Please be on time and come back. Thank you. See you.
a lot of data. I think those data they get from
going to start with the valedictory function soon, and I think to sit somewhere in the hall and seeing an empty audience is always very boring. It's not that. So the best way to do that is to come closer so that you don't have to look in the front. And what is happening in the back of you, hell with it. So it is better, I request all of you to come closer and make it a very tight audience so that you enjoy the program. And from the dais, those who sit in the dais, it is very torturing for them to see an empty audience. But we can't help it. But at least you get yourself saved. Don't sit and keep on looking at the empty audience. So come closer. Those the backbenchers, please come. Come forward. You are students? Students, no? Yeah? Why you are so shy? Come. मैडम आप भी आ जाइए सामने साक्षी अंदर यहाँ आ जाओ सामने आ जाओ अभी सब वैज्ञानिक बनिए अभी वैज्ञानिक अच्छी बात
Light, please, on the dais. Namaskar. Very good afternoon, distinguished dignitaries, esteemed speakers, delegates, ladies, gentlemen. Once again, we are here and we would like to welcome you all on behalf of the West Bengal University of Animal and Fishery Sciences and Indian Society of Animal Genetics and Breeding to the valedictory session of their 19th convention and international conference. Myself, Dr. Lopamutra, and my colleague, Dr. Reshma Sinharai, is here today.
There is a beautiful poem by Rabindranath Tagore. চিত্ত যেথা ভয় শূন্য উচ্চ যেথা শির জ্ঞান যেথা মুক্ত গৃহের প্রাচীর. Where the mind is without fear and head held high, where the knowledge is free, where the world has not been broken up into fragments of narrow domestic wars. We trust that the enriching scientific deliberation, thought-provoking presentation, insightful discussions, and transformative knowledge exchange will truly show a way forward in the domain of animal genetics and breeding. Hope the serene evening through the river Ganges have left you with memorable experience too.
Ladies and gentlemen, today, on the auspicious day of the birthday of first Prime Minister of India, Pandit Jawaharlal Nehru, wishing all our loving children, especially yesterday I have seen few are there, especially to those, a very happy Children's Day. May the future flag bearers grow gracefully, delicately, with all human qualities, intellects, innovation for making India the best place in the globe in all respect.
And the best place to survive for the future generation. Towards the sustainable development we are moving. So that, ladies and gentlemen, it's an honor to invite our delighted guest onto the dais. So I would like to invite Professor Purnendu Biswas, our former Vice Chancellor of West Bengal University of Animal and Fishery Sciences, onto the dais. Dr. T.J. Rasool, Director Camel Biotechnology, United Arab Emirates. Dr. B.P. Mishra, the President, ISAGB, and the Honorable Vice Chancellor, Dr. T.K. Datta, to grace this valedictory dais. Sir, please be onto the dais. Now I would like to request Dr. Uttam Sarkar, Organizing Secretary, and his colleague, Dr. Sanjay Datta, Office Secretary, ISAGBCON 2025, to accompany all the dignitaries onto the dais. It's our honor to have all the significant dignitaries among us, and we are really grateful to have all of you among us.
May I now request our volunteers to come forward, and I would like to request the Honorable Vice Chancellor, Professor Dr. T.K. Datta, to greet our dignitaries on the dais. Today we are lucky enough to have Professor Purnendu Biswas, the former Vice Chancellor, West Bengal University of Animal and Fishery Sciences, amongst us. Sir, thank you, sir. Dr. Datta is also requested to greet Dr. T.J. Rasool. Thank you, sir. The Director General, Camel Biotechnology Center. Thank you, sir. Sir, you are also requested to greet once again the President of ISAGB, Dr. B.P. Mishra. Thank you, sir. So, Mr. Sir, you are now requested just to counteract. Please greet our beloved Honorable Vice Chancellor, Dr. T.K. Datta. Thank you, sir. Sir, also you are requested to greet the two soldiers of this great event, Dr. Uttam Sarkar, the Organizing Secretary, ISAGBCON 2025. And at last, but definitely not the least, Dr. Sanjay Datta, the Office Secretary, ISAGBCON 2025.
I think the man who communicates with you much more. Thank you, sir. May I now request the Honorable Vice Chancellor, West Bengal University of Animal and Fishery Sciences, Dr. T.K. Datta, to welcome you once again with his warm words.
Yesterday morning, I welcomed you with a good morning note, and today it's good afternoon for all of you. Once again, welcome at this very auspicious function of the ISAGBCON 2025. To tell you frankly, I have experience of organizing this kind of seminars before also, small, medium, large. But I must tell you, this particular conference remained one of the special. And special because of you. I have never seen such a kind of a cohesive audience. And all these sessions, scientific sessions were full. And it was like that you got some substance to chew. And probably thanks to the speakers also, they could ignite your mind, and they will all carry home a very enriched knowledge and new enthusiasm, and ultimately progressing the science of genetics and the science of breeding and the cause of the livestock owners and the poultry and livestock owners for the prosperity of not only India but throughout the world. I must especially thank, this is becoming a Thanksgiving speech. They asked me to give a welcome speech, but I will not get opportunity again. The mic will be taken by Uttam and Sanjay, so I am doing my duty. And welcome to all our international speakers. I could see two of them here, Alfred and Katkar Sahab, and quite a few of them joined online from different parts of the world. And I always believe that it is the speakers who make a conference, and followed by the audience and how you respond to it, and with what kind of a mindset you came to attend this. What was your expectation? And we put ourselves in front of you to judge us that whether we could meet your expectation or not. But if we could do something useful and you like the discussion, this conference will be a success. This is a big city, and we try to make all of you comfortable with your transport, food, etc., etc. And to tell you frankly, it is about a team of more than 100 persons who worked day and night, and since last 6 months, arranging for every small bit of the logistics that is required, be the academic logistics or the physical logistics and all. So I hope you enjoyed. For any inconvenience, you are free to blame me as the head of the institution. And if you like something, this goes to my team. And I also must thank the organizer, the ISAGB, Professor Rasool and Dr. Biswa, for offering us to conduct this very important meeting. Last but not the least, I am grateful to my senior, Professor Biswa, for accepting our request. Sir, you always remain a kind of inspiration for us, a driving force for us. And for any small or large problem, we lean back on you to seek guidance and help. And you never kind of denied it. So for today's function also, we thank you for gracing this particular dais. And thank you once again, and thank you all. And let us wait. I am waiting anxiously to see the award winners for whom this particular function is. But let me tell you that some of you will win awards and many of you will not win awards, but you are already a winner to participate in this particular scientific deliberation. And I wish you all the best, particularly the youngsters. In my yesterday's address, I tell
I already said that this particular meeting, we kept the young and the students as a prominence because that is the investment for the future. This is how we look at it. And you have done excellent in this conference. I was looking at the poster sessions. They were jam-packed. And usually poster session used to be a thin attendance. But here I find that people are taking so keen interest in doing that. So thank you very much once again for coming over there. And as I say that please carry home good memories and please message us back after you reach home that you have reached over there, and keep our WhatsApp group active for some more time, post photos and write your comments on that. That will remain an inspiration for us. Thank you very much.
Heartfelt gratitude to our sir for his valuable words and for this conference. Ladies and gentlemen, we have witnessed excellent deliberation as well as productive interactions. Now, the organizing secretary, Dr. Uttam Sarkar, will place the comprehensive report of the presentation and enriching outcome of this 2 days international conference. I would like to request Dr. Sarkar, please be here.
Thank you all. We had planning the total conference in 2 plenary sessions, 3 technical sessions and 3 booster sessions. In first plenary sessions, Dr. Subir S. Majumdar, Director General, GBU, presented on the topic Generation of High Density SNP chip using indigenous cattle for genomic selection. In his presentation, he showed the development of the world's most densely SNP chip, Go chip, using approx 788K SNPs. This chip was launched in 2024 in indigenous cattle. In plenary session 2, the speaker was Dr. Meher Singh Khatkar. His topic was Beyond Genomics, Phenomics and Artificial Intelligence to Transform Economic Trade Evaluation in Livestock. He has highlighted the importance of how the genetic improvement can achieve through the integrative approach of genomics, phenomics and artificial intelligence in a same platform. This integrative approach can transform economic trade evaluation in a more productive with better economic output. We have 3 technical sessions. First technical session, there were 4 lead speakers, followed by 2 oral presentations and 24 poster presentations. Dr. Srinivas has highlighted the advances in aquatic genetics, where polyploidy and species hybridization is common that lead to better genetic gain, probably due to shorter generation interval and application of genomic selection. He claimed Jayanti Rohu and Maha Maghu were their greatest achievement. Another lead speaker, Dr. Molla, has claimed to evolve a better gene editing tool involving TNPB plant, genome editor, gene activation, and lastly highlighted AI designed genome editor. Another speaker, Dr. Kathiravan, has explained the every details of application of artificial intelligence and ICT. Our 4th lead speaker, Dr. Shana, has presented in online mode from Denmark, highlighting the breeding strategies involving better microbiome evolve in reduced methane emissions. Our two oral presentation from best abstract. First one is Dr. Ravi Kumar has highlighted the detailed insights of cattle genome with particular reference to Indian Zebu cattle, and other speakers, Dr. Jain, has explained in detail the impact of cathepsin in the oocyte maturation journey. In first technical session, recommendation was in precision genome breeding, AI needs to be included in decision making with development of suitable infrastructure involving IoT and computer to generate different phenotypes and integrate with genomics. Alternative gene editing tools may be developed to overcome the limitation of CRISPR-Cas9. Indigo chips need to be used for indigenous cattle rather than that developed for exotic cattle. In technical session 2, next generation breeding, integrating genomics and phenomics. These sessions are chaired by Dr. P.P. Misra sir and Dr. Triveni Dutt, while Dr. Subodh Kumar acted as rapporteur. Their recommendation based on technical sessions: more number of improved pools are required for AI program, more number of improved cows were required for ETT program, the use of sex-sorted semen should be promoted, genomic selection should be coupled with IVF ETT program, a single-step genomic blood was promising outcomes, novel traits should be phenotyped and included in GS program, fitness traits should be taken into account in the genomic selection program. In technical session 3, the topic was exploring IoT and artificial intelligence in animal food system. There are 4 lead speakers, followed by 2 oral presentations. Dr. Mohanty has detailed how the sensors may be effectively employed for analysis through artificial intelligence in a dairy. The use of AI may evolve for optimizing resources and productivity. Other emerging AI and machine learning approaches discussed where AI, IoT powered wearables as applicable for cattle and buffalo production, health, AI and IoT were equally applicable for conventional and climate-ready livestock system. The oral presentation highlighted the comparative analysis for breeding value estimation and predictive of milk yield. Recommendation was AI coupled with IoT need to be implemented with conventional breeding system for better output. We had 3 poster sessions. In first poster session, participants were 15, out of students was 4 and faculty was 3. In both cases, Dr. Devarko Pal was first in case of students and Dr. Varsha Jain is second. In case of faculty members, Neeraj Kashyap was first and Prem Dubey was second. In poster session 2, total participants was 24, out of 11 was student and faculty was 13. In case of student, Akshata Patil was first and second was Kankan Baro. In case of faculty or scientist, Dr. Nihar Ranjan Shah is first and Dr. Somen Nashkar was second. In poster session 3, total participants is 23, out of 10 is student and 13 is faculty or teachers. In case of students, first was Ranjit Ji, and second are Hemlata Valmiki and Rohit Barawar jointly. In case of faculty and scientists, Dr. Kanaka KK was first and Hemantu Kumar Maiti is second. This is the poster session. The last session was Roundtable Industry Academia Interfaces. Dr. Ajay Aswal, Dr. Nilesh Nai, and Dr. Sudito Haldar was the speakers. The common recommendation of these three speakers was industry require research that is both innovative and practical, adequate genetic scheme into field of applicable solution for enhancement of productivity, health and sustainability with Indian specific environment and required framework. Second was, there is need of industry-institute collaboration for identification and problems and effective solution benefits both scientific and industry. Third recommendation is genomic selection is more than specific advancement. It is practical solution for future and Indian dairy industry. Efforts should be made into create more diverse reference population and effective data coding mechanism. Fourth recommendation is the number of embryo production in India is very low compared to developed countries, and embryo success rate also very poor. Industry, academia, people should collaborate to develop strategic policies for implementation of developmental performance. Thank you.
Thank you, Dr. Roy, for your valuable inputs to all of us. Now, ladies and gentlemen, last 2 days, the scientific workers from different corners of the country and beyond presented many thought-provoking ideas to justify the theme of ISAGBCON 2025. They have shown us really how the most sophisticated technologies of artificial intelligence and machine learning could promisingly guide the new era. Especially India, with the highest livestock population, towards transformative future to ensure better growth. Finally, the most awaited moment.
So the most awaited moment has come to honor those brilliant, innovative presentations and presenters who push the boundaries of what is impossible and what is possible. But before that, we must congratulate each one of the presentations for their oral or poster presentation. We want to congratulate them. There is no winner, there is no loser, we all are at the same page. So every presentation, every poster, every effort is visible in this presentation, in this conference. So thank you all of you for joining us. But this is the time to present the award towards the paper as well as the poster. So this is the time.
Actually, we received initially more than 130 abstracts, out of which a screening committee constituted with competent experts have chosen only 6 best abstracts to be presented orally. So ladies and gentlemen, could you understand the competition begins that time only. Here, all 6 presenters out of more than 130 submissions are already super winners of this event in their respective technical session.
I would like to call upon the stage the best abstract presentations from different sessions. But as you have already received the memento in your technical sessions, here only we are giving you our heartfelt thanks as well as certificates. So I would like to invite Dr. Rani Alex, SS, NTRI Colonel, for technical session 1.
Please,
Dr. Rani Alex.
So, Dr. Rani is going to illuminate, going to come with her illuminating presence, and that's why I think the light got off, put off. So Dr. Rani, please huge round of applause for Dr. Rani.
Congratulations, Dr. Rani, for your kind work, for your beautiful work, beautiful presentation. Second, I would like to invite Professor Ashish Kumar Ghosh, GUPUAT, Pantnagar, sorry. Dr. Ashish Kumar Ghosh, Professor Ashish Kumar Ghosh.
So anybody on behalf of him may please receive the prize.
Teammate, friends.
So technical session 2, tools for accelerated genetic improvement, applications in plants, animals and aquatic system. And the best abstract winner are first Dr. Ravi Kumar Gandham, Head Animal Biotechnology, ICAR NBAGR Karnal. His topic of lecture was Cattle Genome, Insights into Indian Zebu. Huge round of applause for Dr. Ravi Kumar. Congratulations. I'd like to request our Honorable Vice Chancellor to hand over the certificate. Next is Dr. Ashit Jain, Associate Professor, NDBSU, Jabalpur. His lecture was Cathaspin inhibition by E64 during oocyte maturation enhances developmental competence of buffalo embryos via apoptosis suppression. Please put your hands together for Dr. Ashit Jain.
Congratulations, sir. Now it's the time for technical session 3. And here I would like to invite Dr. Bikash Divakaran NTRI Colonel for comparative analysis of artificial intelligence based on prediction algorithms for breeding value estimate in Mura buffalo. Dr. Bikash Divakar, NTRI. May I request Dr. B.P. Mishra, the President of ISAGB and former Director of ICAR NBAGR, to confer the award on them. Now, it's the time to invite Professor B.N. Sahai, GBUAT, Pantnagar, for comparative evaluation of machine learning models for prediction of fast lactation 305-day milk yield in crossbreed cattle. Dr. B.N. Sahi is not here, so on behalf of his colleague, he is taking this award.
Congratulations. So ladies and gentlemen, as per our jury, remaining submissions were allowed to present posters. It is understood how tough it was and how challenging it was for the evaluators to screen out the best presentation. Finally, those wise members of the evaluation committee have clustered
whole presentation into two bags. One for the students, the upcoming researchers, and another for the teachers, faculty members, and established scientists. So, now onwards in poster session, for each technical session, we are going to hand over to first and second in students category, and also in the second category, that is professor, faculty members, scientists or researcher. So I would like to request Dr. Reshma to please spell out those names for the poster session. So, So from the first technical session, Dr. Debar Gopal, Dr. Debar Gopal, and then Dr. Varsha Jain. Dr. Varsha Jain, please come first. And any friend or teammate of Varsha? Next, for the faculty category, Dr. Neeraj Kashyap. I'd like to request VP Mr. Sir to hand over the prize. Next one, Dr. Prem Prakash Dubey. Dr. Prem Prakash Dubey, yeah, he's there. Now, I think Dr. Neeraj Kashyap is coming, yeah. Dr. Neeraj Kashyap is here. Dr. Prem Prakash Dubey. Poster session 2, next generation breeding integrating genomics and phenomics. I am requesting teacher Rasul sir to hand over the prizes. Students category, Dr. Akshata Patil. Dr. Akshata Patil, and second one, Dr. Kankan Baro. Dr. Akshata Patil and Dr. Kankan Baro. And in the faculty category, please get ready, Dr. Nihar Ranjan Sahu and Dr. Shaumen Nashkar. Please get ready to receive your awards. Now, Dr. Akshata Patil taken. Please move forward. Okay. This is for this session 2, poster session 2, Dr. Akshata Patil. And Dr. Nihar Ranjan Sahu. Oh, Kankan Baro, sorry. Dr. Kankan Baro. Dr. Kankan Baro. In faculty category, Dr. Nihar Ranjan Sahu. In faculty category, Dr. Nihar Ranjan Sahu. Please see. The volunteers, you please. Dr. Nihar Ranjan Sahu. Yeah. And Dr. Shaumen Nashkar. Poster session 3. Dr. Shaumen Nashkar, yeah, please come forward. Okay, just wait, we are looking at it. Yeah, it is there, sir. Yeah. So on behalf of Dr. Nihar Ranjan Sahu, his colleague is taking his award. Now it's the time for Dr. Shaumen Nashkar. Dr. Shaumen Nashkar. No. His colleague. Okay, Dr. Shaumen Nashkar is not here. His colleague is taking his prize on behalf of him. So let us move towards poster session 3, Biological Systems, Reproductive and Adaptive Genomics. Students category, Dr. Ranjit G, Dr. Ranjit G and Dr. Hemlata Valmiki. Please come forward. Students category, Dr. Ranjit G. Please come forward first with Dr. Ranjit G. Ranjit, okay, and Hemlata. And in faculty category, Dr. Rohit Barwal, Dr. Rohit Barwal. And the next one, oh, I see there are two joint second winner. One is Dr. Just a minute, Dr. Hemlata Valmiki and Dr. Rohit Barwal. So I'd like to invite Dr. Rohit also on the stage for receiving joint second winner prize in this category. And in faculty category, ma'am, please. Yeah. Dr. Kankana KK. I would like to invite Dr. Kankana KK. Dr. Kanaka KK. Dr. Kanaka KK, sorry, excuse me. Dr. Kanaka KK. Anyone on behalf of her? Then I would like to invite Dr. Hemanta Kumar Maiti, please onto the dais. Dr. Hemanta Kumar Maiti. Dr. Hemanta and Dr. Kanaka. Kanaka, please come forward. Okay. Oh, he is also. Congratulations, sir. Now, Dr. Hemanta Kumar Maiti. So again, I think they have allocated themselves the duties. I will present and he will take the award because they are confident enough that award will be there. So, Dr. Shumon is taking the award on behalf of Dr. Hemanta Kumar Maiti. So now, another exciting event, the Young ISAGB Young Researcher Award. This year, the ISAGB Young Researcher Award is given only to one, and that goes to B. Jaya Madhuri, Tanuvas, Chennai. So please give a huge round of applause to B. Jaya Madhuri from Tanuvas for winning ISAGB Young Researcher Award 2025. It's a great moment for a teacher to receive the award on behalf of a student. So sir is taking this award. Thank you, sir. Thank you. So, yeah. So we have completed our award ceremony of this conference. Now it's the time to hear some blissful things. So may I now request to invite Professor Purnendu Biswas, the former Vice Chancellor, West Bengal University of Animal and Fishery Sciences, and one of the most pragmatic visionary leaders to address the learned congregation. So please.
So first of all, I like to convey my thanks and gratitude to Dr. Datta, Honorable Vice Chancellor of our university, and the organizers for inviting me here. I am really delighted to be here with you for the last, today and study. This is one sort of what I think that this association conference, is, along with the scientific deliberations, it's also a kind of reunion. Many of you, I know, you also know me same, irrespective of the subject. I always like this kind of conferences, and when Dr. Datta told me that they are going to organize this in Kolkata, so initially he is also new to Kolkata, although he is from here, but after long service life he came here, and a little bit hesitant. But once our team in the university, we work well, they are very dynamic, and any kind of this conference they can organize. So under the leadership, and nowadays it is also become very easier because of the social media. You have your own group. I am also a part of this group. They have made me a member. So I saw that even yesterday night also, up to late night, we got many videos, pictures of this quiz dinner. So anyway, I have some suggestion. This other than subject, I have some suggestion. I was discussing with Biswa that for betterment of this kind of conference, but this is exclusively my opinion, so you can think over it. That number one is what I saw here, that number of retired person is very less. I don't know who are retired here because I am from nutrition fraternity, so I don't know who are retired. But what I feel that retired persons, I suggest that this ISAGB should sponsor the participation of the retired teachers, retired personals here. Number two is for the medals and awards. Some sponsorship is required because these awards and medals, this is basically, to me, it's meant for the students to inspire them for further better work, because the participation of the students in the conference, this is required. This is required because they should know how to prepare a paper, how to prepare a poster, how to present, how the seniors are presenting. So this is required. They should learn it from the seniors. So these are the few, because again, you see, for category-wise award, particularly if you ask or request the retired persons of your organization to sponsor some awards and medals, so lifetime achievement and all this. So I think that will not be very hard to gather. You can collect it. And when every year you are organizing this, in animal nutrition, we organize this biannual, and you are organizing this annually. So, and I am also very much delighted that the first this kind of international conference held in Kolkata. So I am really very glad, and I hope that for the last 2 days, other than these scientific deliberations, you have enjoyed and visited some part of Kolkata, that places of interest you have visited and enjoyed the food, because Bengal is made for food. Particularly, I don't know how many of you is non-vegetarian, because Bengali peoples we are fond of vegetarian, non-vegetarian foods. So and to enjoy Kolkata, you'll have to visit the street food corners in Kolkata, this tea corners and tea stalls in street where you will get 3 rupees, 5 rupees in small cup, clay cup or paper cup. This tea is still available in Kolkata. So you'll have to enjoy that by walking through the roads in Kolkata. Although we also miss these vintage, the tram company, tram CTC, Calcutta Tram Company. So it's missing. But again, Kolkata is the first city where this metro started. So that's the thing, we are all proud of our city, and I think you also have enjoyed a lot, and I was, how was the people enjoyed yesterday night? So I thought that he told me that no, everybody they enjoyed as per condition. So once again, I like to congratulate all of you for participating here and coming to Kolkata, and hope that in the near future also again ISAGB conference will be held at Kolkata. Thank you all.
Thank you so much, sir, for your wonderful words. We are delighted to have Dr. B.P. Mishra, the President ISAGB and former Director ICAR NBA GR Karnal. I would like to invite sir onto the dais.
Good afternoon. I think I see a delightful picture that at the end of the valedictory session, normally we would have 40 to 60. That's the ideal size at the end of today. But today in this hall, I was having a rough estimate. It's more than 100. So that speaks that truly the conference was good, and it speaks that the valedictory session has got a quite a decent representative. On the dais, I welcome the chief guest of today's function, our patron, Dr. T.J. Rasool, the Director of Center for Biotechnology in Dubai, and the patron of ISAGB being the chief guest, and Professor Purnendu Biswa, Honorable former Vice Chancellor of this very university. My friend, Honorable Vice Chancellor Dr. T.K. Datta, the two dynamic soldiers for the university who have spearheaded painstakingly, Dr. Sarkar, the organizing secretary, and the secretary for the office, Dr. Sanjay Datta. Of the dais, the two international speakers still present in this very valedictory session, Dr. Alfred DeFrage from Gates Foundation, Dr. Meher Singh Khatkar, professor from Australia. We are delighted that you are still here. The First Lady, Madam Datta of this university, Dr. Mrs. Misra, Dr. Dave, the distinguished Dean of Veterinary Science, Dr. Niltut Bose, our Vice President of ISAGB, Dr. Vineet Vasil, my Joint Secretary, Dr. Raut has to move, so therefore he is not there. All the distinguished ISAGB fellows, the faculty of this very university, the delegates, the heads of division of various centers of ICR Institute situated over here, students, ladies and gentlemen, as everything comes down to an end, that this very first ever international conference of ISAGB, Conference 2025, has come to an end. I, on behalf of the society, and being the president of the society, it's my duty to acknowledge with heartfelt thanks the painstaking effort of Honorable Vice Chancellor, Dr. Tirtha Kumar Datta, and the entire team of West Bengal University of Animal and Fishery Science. A big round of applause to the university and to the department that they made it possible that in the Biswa Bangla Convention Center, I hope animal genetics breeding today has got a new dimension. In terms of the international conference, I could feel myself, after attending so many in my 36 years of service, I feel that being a genetics and breeding fraternity and also being in the society, we could have an international standard conference at this venue. Thanks to you and your entire team. I know every conference to organize requires painstaking effort quite for some time. And this particular one, which we involved from ISAGB, myself, my core team of Vice President and Joint Secretary, and Secretary with the team here, working through online interactions, I know how difficult it was not only to mobilize logistics, but first and foremost, the funds to organize such a big and huge conference. Again, those all painstaking effort, all who are involved, we are saying only the front runners, but there are all the committees. I think I was looking at the yesterday night cultural extravaganza over Ganges. I think that was something different for the delegates to experience. So all these requires mindful thinking, and particularly so when we decided on the theme of this conference, we had painstaking effort of deciding it, but we at one time thought that this would be much more advanced and we are not right away working on these areas. But I remember Dr. Datta said that we have to start somewhere. Like we have started in 2017, this conference was started on genomic selection. When we could see nowhere we are near to that, but still 2017, IBRA hosted a national workshop one day, chaired by Honorable Director General Dr. Tirlochan Mahapatra, a plant breeder. He only told that if you want to do something, you should have a scientific session one day. That was the starting point. And then followed by that, we had a national conference at IBRA. So from there, the journey started. And every year we are having something. But this year, the theme is extraordinary. In terms of what you saw, the selection of lectures, the selection of speakers, and cross-learning from plant fisheries. I'm thankful to the professors of plant, the professor of engineering, right? And all those speakers who have spoken about AI, about genomics, which we have been doing, but this cross-learning across disciplines where genetics did not find barrier, whether it's plant, animal. Being animal, we are, the principle of breeding starts long before than other fields. But we are now sharing knowledge, and I hope this journey begins from here. We'll have next round of what you call, somebody says breeding 4.0, breeding 5.0, and all that. This will be a launching pad, and I foresee that the next conference, when it will be happening in November or December next, in either central India or the southern part of India, that will be deciding soon, and you all are welcome to the next version of ISAGB conference 2026 with a different perspective, and hope the organizers who have seen it here would raise the bar of ISAGB. I, on behalf of the society, thanks all the delegates and the host, and my patron, my colleague, Vice President, Joint Secretary, who have painstakingly worked to convene and make this international conference, ISAGB 2025, a grand success. So therefore, I once again thank to the host, and thanks everyone from the ISAGB and all the life members, fellows and the delegates, and wish you all a very pleasant return journey, safe journey back home. Thank you. Jai Hind, Jai Bharat.
Thank you, sir. We are gratefully acknowledge your kind words of appreciation and encouragement. I think definitely it's a great compliment for all of us. Now, I take the opportunity to invite another stalwarts in the field of animal genetics and breeding, Dr. T.J. Rasool, Honorable Director, Camel Biotechnology Center, United Arab Emirates. Sir, please share your impression and valuable view regarding today's brainstorm session. Sir, please.
Good afternoon. Professor P.K. Datta, the Honorable Vice Chancellor of West Bengal University of Animal Science and Fisheries, Professor Biswa, ex-VC of this university, Dr. B.P. Mishra, President of ISAGB, Dr. Uttam Sarkar, the local organizing secretary, and his colleague, Dr. Vineet Basi, the Vice President of ISAGB, Dr. Raut, Vice Joint Secretary of ISAGB, Dr. V.K. Saxena is not here, I think Secretary of this society, Dr. Alfred Divris, Dr. Meher Super Kekar, who just like me took pains to come here all the way and to attend this meeting. And Mrs. P.K. Datta, the First Lady of the campus, and Mrs. Veena Misra, the ex-First Lady of NBAGR campus and the Principal Scientist of in NBAGR, the rest of the ladies, and my colleagues, my friends, Dr. Dev, my colleague, Dr. Sandatta, my colleague, old friend, Dr. Pushpender. I mean, if I have left anybody, please excuse me. Now, the least energy or lowest energy session in any scientific seminar or technical seminar is the valedictory function. So I know all your energy out now. So I don't want to take much of time. And already people have already explained everything. And within that also, within the valedictory low energy, this one, the last speaker would be the lowest energy vendor. So I will not trouble you much more than what they have already told. But mind you, with all kudos and appreciation to Professor Datta and his team, this was one of the best ISAGB conference we have organized. We are organizing these conferences from 1984, and I am a person from the beginning, and it had ups and downs in its course of history. But when 2007 I moved to council, I totally rejuvenated and brought really young blood, and from that Dr. Mishra, Dr. Vineet Basir, my colleague, Dr. V.K. Saxena, and a battery of young people, and since then this society is going smoothly without any problem, and today it has come to this level, and I am very happy for the West Bengal Agriculture and Animal Science and Fisheries University to have done this yeoman service. So I have nothing more to add. Appreciation is there, but I have some, no appreciation will be complete without some criticism also. I have two criticism I felt. Professor Datta, you have, should not mind because you have done an excellent, this one you have given. One thing is I felt the poster session, see, how much encouragement we give the children, that is more important, students. Posters should be given more prominence. I would prefer a poster session should be immediately after the inaugural session. And enough time should be given so that the youngsters will get enough encouragement. I hope in future, Dr. Biswa, this will be taken care of. Another trivial issue I want to talk is not on a society or organizers, but we, the members. The applause level in the auditorium is very low. You go to film fraternity, you go to any other technical sessions or the applause level when a colleague is getting an award, when somebody is appreciated, is very high. And that shows the high energy level among us. And here, I do not know why the journalists are miserly in applauding. A clapping is an energy transfer mechanism. And it's a sense of appreciation. We should be free to applause and give appreciation to all our colleagues whenever they are honored for some of their merits. This is my humble request. Now, regarding the subject matter, definitely the topic of symposium is totally different from. We are hearing for artificial intelligence and machine learning from many times, but we did not know what exactly it is. And today, at least we are better educated, better informed what exactly it is, and most of our confusions are clear. And I definitely once again appreciate the organizers for taking such an innovative idea, bringing AI into animal genetics. And but one thing I want to add is This is nothing but totally data based. And the data is my sequencing. The entire thing is sequence only, NGS only. Around 20 years back, when human genome mapping project started, we thought, oh, God's blueprint is in our hand, we can do anything. But nothing happened. Human is there only, genetics is there only. We are improving. So don't think genetics by sequencing or NGS and the data alone. You have to convert it into a practical utility, or in other ways, no research can sustain unless you convert into economic unit. So every one of my colleagues, my advice is that please plan a economic end, where a commercial output from the research. Earlier, because we believed that basic research, applied research and all this one. But due course of time, basic research definitely is required. But when we plan, and even if long term, there should be an economic value to the research project we are taking, and we should not ignore that point. Another issue I would like to add is that again, we thought sequencing is everything in entire nucleotide sequence in all the chromosomes is everything. But now we realize in our own experience, we wanted to improve the efficiency of raising by cloning the best animals. So it is not repeating exactly. So sequence alone is not the ultimate decisive factor in genetics. There are much more than that. Before, 150 years back, when we heard about Darwin, there was a person called Lamarck. Lamarckism, at that time, nobody gave that importance, and Lamarckism died a natural way. But after 1.5 centuries, what we realize is now, genetics, sequence alone is not forming the genetics. We have something more than that. The most important is, we know now we have cloning, our lab is cloning camels like a Xerox machine. Professor Huan from Korea, who is the father of the Snoopy, the second animal cloned in the world, the dog, after Dolly, they are making so many clones of each top champions. But in the field, they are not able to reproduce the same as their original donor camel. So what I want to emphasize is, there should be enough importance to epigenetics, methylation differences, also along with sequence information. I have already requested Dr. Mishra to have a separate session on epigenetics in between before our next annual meet. I have nothing more to add. It was a very interesting symposium. As we part ways, let us carry forward the spirit of inquiry and the contacts we built here. May the seeds of ideas planted here grow into new researches that benefit science and humanity alike. Thank you all, and I wish you all continued success in your research journey forever. Jai Hind.
Thank you so much, sir, for your wonderful words. We are mesmerized by your words. Now this is the time to felicitate our significant dignitaries. Actually, any kind of conference is an event of exchange, is an event of collaboration, and a story of growing old together, growing together for a better world. On the Rabindranath Thakurer kothay, dibe ar dibe, milabe milibe, jabe na phire. When a conference is in an old city like Kolkata, which has the history of 1893, when the veterinary college was first initiated as the first veterinary institute in our country. So obviously that conference makes a sense, that has a significant impact historically as well as on the basis of the theme of the conference. So now it's the time to felicitate Professor Purnendu Biswas, our former Vice Chancellor. I would like to request our Honorable Vice Chancellor, Dr. T.K. Datto, to felicitate Sir. Thank you so much, sir. Now it's the time to felicitate our most significant guest, Dr. T.J. Rasool. I would like to invite our Vice Chancellor, sir, to felicitate, sir. Thank you so much, sir. But end is not an end. It's the beginning of something new. In order to end this session positively with a great vibe, I would like to invite Dr. Datta to giving the vote of thanks.
Respected dignitaries on the dais, Dr. T.K. Datta, Honorable Vice Chancellor, West Bengal University of Animal and Fishery Sciences, and Chief Patron, ISAGBCON 2025, Dr. T.J. Rasool, Honorable Director, Camel Biotechnology Center, Management of Scientific Centers and Presidential Camels, Department of Presidential Affairs, Al Ain UAE, Dr. B.P. Mishra, President ISAGB and former Director ICAR NBR Colonel, Professor Purnendu Biswas, former Vice Chancellor, West Bengal University of Animal and Fishery Sciences, distinguished guests, invited speakers, respected delegates, colleagues, and dear students. Good evening to all of you. As we conclude the 19th Annual Convention of Indian Society of Animal Genetics and Breeding and the International Conference on Precision Animal Breeding through Genomics, Artificial Intelligence and Machine Learning, it is my privilege on behalf of the entire organizing committee to express our deepest gratitude to each and everyone who made these 3 days of knowledge, collaboration and friendship truly memorable. We are profoundly grateful to our chief guests of the inaugural sessions, Sri Pradeep Majumdar, Minister in charge of Panchayat and Rural Development, Government of West Bengal, and Sri Swapan Devnath, Minister in charge of Animal Resources Development Department, Government of West Bengal, for gracing the inaugural session and delivering an enlightening address. Your words have truly motivated us to harness science and technology for the transformation of Indian livestock sector. Our deepest regards goes to the former Vice Chancellors of West Bengal University of Animal and Fishery Sciences, namely Professor B.B. Mollik, Professor Chanchal Guho, Professor Purnendu Biswas, Professor Shamsundar Dana, Professor Chandrasekhar Chakraborty, for adding prestige and honor to this gathering with their esteemed presence. We express our deepest respect to the former teachers of Animal Genetics and Breeding and Livestock Production and Management Departments of West Bengal University of Animal and Fishery Sciences, Professor A.K. Sahu, Professor A.K. Samanto, and Professor D.N. Moitro, for gracing us with their distinguished presence and guiding us with their wisdom. It is indeed my profound privilege to thank Dr. Triveni Dutt, Director ICAR IVRI, Dr. T.K. Bhattacharya, Director ICAR NRC, Dr. N.H. Mohan, Director ICAR NBAGR Colonel, Dr. Yes Pal, Director ICAR CIRB, Dr. Mihir Sarkar, Director ICAR NRC Y, and Dr. Tapos Ghoshal, Head CIFE, for taking time out of their busy schedules and inspired us with their valuable presence. Your presence not only elevated the stature of this event, but also reaffirmed our shared commitment for advancing animal science and innovation for a sustainable future. Our sincere acknowledgement goes to all the invited guests from different institutes, colleges, universities, organizations for gracing us with their distinguished presence and active participation and enthusiasm that brought life to the event. We sincerely thank our invited lead speakers, Dr. Aruna T. Kumar, former editor, Indian Journal of Animal Sciences, Dr. Alfred D. Vries, Senior Program Officer, Gates Foundation, Dr. Subir S. Mojumdar, DG, Gujarat Biotechnology University, and an excellent singer, Dr. Somen Mondal, Principal Scientist, CSIR, Central Mechanical Engineering Research Institute, Dr. T.K. Mohanty, former Principal Scientist, NDRI Karnal, Sri Srinivas Jagirdar, Principal Scientist, ICR CIFE, Dr. Gopal Guane, Principal Scientist, ICR NDRI Karnal, Dr. Pediyasamy Kathiravan, Principal Scientist, ICR NRCM, Dr. Meher Singh Khatkar, Adelaide University, Dr. Nilesh Naim, Deputy General Manager, NDDB, Dr. Kutubuddin Ali Mullah, Senior Scientist, ICR NRRI, Dr. Srinivas K. Ambatipudi, IIT Roorkee, Dr. Ajay Pal Singh Aswal, Uttarakhand Livestock Development Board, Dr. Sudipto Haldar, Director at AgriVet Research Advisory Private Limited, Dr. Ositabo Sood, renowned dairy consultant, Dr. Aditya Konar, Chief Scientist, CSIR IICB, Professor Arunashis Goswami, West Bengal University of Animal and Fishery Sciences, Professor Gautam Sahana, Arahas University, Denmark, Professor Mintu Nath, University of Aberdeen, and Professor Suresh Raja Nitirajan, Dalhousie University, Canada, for stimulating thought-provoking discussions and academic exchange during the conference. I wish to extend our heartfelt thanks to all the chairpersons, rapporteurs, and session coordinators for their wholehearted support and smooth running of the technical sessions. A special thanks and gratitude to Dr. B.K. Taneja, Chief Patron ISAGB, Dr. T.J. Rasul, Patron ISAGB, Dr. B.P. Mishra, President ISAGB, Dr. B.K. Saxena, Secretary ISAGB, Dr. P.K. Raut, and Dr. Vinit Bhasin, Joint Secretaries ISAGB, and Dr. M.S. Tatia, Treasurer ISAGB, and all the distinguished executive and editorial members of ISAGB for their constant guidance, wholehearted cooperation, and invaluable suggestions that shaped this conference into success. I wish to extend our heartfelt thanks to our Honorable Vice Chancellor Dr. T.K. Dotto, Chief Patron of this conference, for his inspiring leadership, guidance, and constant encouragement. Sir, your confidence in the organizing team and your vision in positioning the university on the global academic map have been cornerstone of this success. We sincerely acknowledge all the patrons and chairpersons for guiding us with wisdom and grace at every step. I place on record my sincere thanks to all faculty members, officers, staffs, and student volunteers of West Bengal University of Animal and Fishery Sciences, and for acting as the chairperson, co-chairpersons, conveners, and members of various committees, and for their unseen hours of hard work, dedication, and their collective effort behind every detail. I would also like to acknowledge the invaluable support of the university administration, whose cooperation and behind-the-scene assistance have been instrumental for smooth execution of this mega event. A very special note of appreciation goes to our student volunteers, the real driving force behind the registration desk, travel and accommodation logistics setup, and technical sessions. Your dedication, energy, and smiles kept the spirit of ISAGB conference alive. Our deepest gratitude to all the delegates, scientists, academicians, industry professionals, and research scholars from India and abroad for bringing diverse ideas, experiences, and research to the table, and for their active participation and thought-provoking discussions. Your enthusiasm and contributions gave true meaning to the instrumental gathering. Our sincere appreciation to Dr. T.J. Rasul, Honorable Director, Camel Biotechnology Center, UAE, Dr. B.P. Mishra, President ISAGB, former Director ICR NBHR Karnal, Professor Pundendu Biswas, former Vice Chancellor, West Bengal University of Animal and Fishery Sciences, for accepting our invitation to serve as the chief guest of the valedictory function. We also acknowledge the generous cooperation of the Animal Resources Development Department, Government of West Bengal, and associated institutions like West Bengal Poultry Federation for their encouragement and presence. Our gratitude extends to the sponsors and the funding partners, namely ICAR, ANRF, Gates Foundation, Indian Immunologicals Limited, West Bengal Poultry Federation, Bengal Animal Health and Nutrition, ICR NRC-Y, ICR NRC-M, ICR NRC-E, ICR CIRB, ICI CIRC, GIS College of Veterinary and Animal Sciences, Mogra, Hubli, Das Dag Center Private Limited, BioLink, IMB India Private Limited, Hube Pharma Private Limited, Zeus Biotech, Rosary Biotech, Bhaksindhu Animal Health and Private Limited, Edison Life Science, Nucleum Informatics Private Limited, Feedback Private Limited, whose generous financial and logistic support helped us to turn this conference into reality. Our heartfelt thanks to different media partners, Radium Management Private Limited for the technical team of the BBCC, West Bengal Surface Transport Corporation Limited, Kolkata Police, Aheli Caterer, Bhoj Caterer, and different hotels and guest house providers for accommodation and hospitality for their professional assistance throughout. Last but not the least, I would like to thank all the contributors who, namely not mentioned here individually, but for your support, effort and presence have been deeply valued. Every small act, silent contribution and unseen dedication has played a vital role in making this event possible. Thank you for standing with us and being an integral part of ISAGBCON 2025. As we draw the curtains on ISAGBCON 2025, let us carry home the shared commitment to scientific excellence, collaboration and innovation that define the legacy of both ISAGB and West Bengal University of Animal and Fishery Sciences. On behalf of the entire organizing committee, I thank each one of you once again and wish you all the safe journey back home. May we meet again in the next ISAGB convention with renewed spirit and stronger partnership. Thank you. Thank you all.
Thank you, Dr. Sanjay Datta, for your heart-touching note of gratitude. The valedictory session will be closed officially with the national anthem. May I request you all to please stand up for national anthem. Let us start ourselves. Yeah. Thank you all. With deep appreciation and warm regards, we will say adieu to each one of you. May I request, as our Vice Chancellor wishes, please come forward for a group photograph. This memory will be kept forever. I think, please don't hesitate. All the participants, organizers, friends, Ma'am Mishra and Ma'am Datta, all are please requested to come on the dais to, I think in front of the dais we can have the photograph. And I wish my team, my reception and our team also go forward for the memorable testimony. And every ending carries within it promise of a new beginning.
Take photos. Yes.
Yeah. So I feel that you will move back and definitely with a very enriched take-home messages. Ladies and gentlemen, after these photographs, we all will move together towards Hall 2, where high tea has been arranged for